Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Symposium. Good day, good night, and greetings from wherever you are around the world. My name is Ray Ford, and I'm with the Small Business and Industry Assistance Program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communication. It's my pleasure, along with my CEDAR colleagues, to be with you here today. This slide provides QR codes for our social media avenues. You can scan them now with your phones and be taken to the appropriate platform. They'll also be advertised across the screen banner during the breaks if you miss it now. We do have some logistics reminders. This is a hybrid event with online attendees, and as such, there may be some internet issues, but please bear with us and we'll resolve them as quickly as possible. Speaking of tech issues, here is our one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. That's the same page you used this morning for your online access to the event, so please bookmark that page. We also invite you to submit questions so that we have a vibrant and productive coveted Q&A session. When you do submit your question by selecting the appropriate session, please identify the individual presenter by referencing the question with the speaker's name. Also, during each presentation, there is a link on the right side of the pod where you see the speakers. You may see an open headshot, and it says Slides and Resources. Note this link's only available during the real-time presentation. Also, if you need closed captioning, please look at the top of your Adobe screen to the right, click on the CC drop-down menu, and select Show Captions. There will be a link to the speaker evaluation available for you, and we encourage you to complete the individual evaluation for each speaker. In addition to the speaker evaluation, we remind you of the final evaluation. The final survey is the mechanism by which you'll receive the certificate of attendance. When an attendee completes the survey evaluation, they can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credit. The certificate must be downloaded immediately after completing the survey, otherwise this opportunity is lost. Even if you don't want or need the certificate, we'd appreciate it if you complete the evaluation to help guide the design of our future events. Note, the evaluation and the certificate are only available for 2 weeks, which means the deadline is February the 29th. The survey link will be posted on our one-stop shop page this afternoon at the end of the day's event. All of our sessions are being recorded. It'll take us roughly about 10 business days to process the recordings, but you'll soon be able to access them from fda.gov/CEDAR-SBIA. For on-site attendees, here is the passcode and the link to the available Wi-Fi. And we ask you also to please silence your phone, try to use the internet sparingly. And again, once a day, you're on your own for lunch. Just to remind you of our upcoming events on February the 22nd. Join us for the joint US FDA Health Canada ICH public meeting. On February the 28th, join us to expand generic drug access through International Engagements webinar, and on March the 7th, we're having the Integrated Safety Analysis and Drug Marketing Applications, Avoiding Common Mistakes. There's more details at fda.gov/CEDAR-SBI. So please bookmark those events. We would love to see you there. We do have a special announcement on clinical trial approaches, on enhancing the adoption of innovative clinical trial approaches to understand the state of innovation in clinical trial design and conduct. FDA is asking for perspectives via comments to our public docket, FDA-2023-N-4489, by April the 19th, and/or participation in the public workshop hosted in partnership with Duke Margolis Center for Health Policy on March the 19th and 20th. For more information, please see the contacts listed here on the slide. We'd like to welcome, for opening remarks and a keynote address, Dr. Julia Cho, Division Director in the Division of Generic Drugs Study Integrity, Office of Study Integrity and Surveillance, and the Office of Translational Sciences in CEDAR. Please join me to welcome Dr. Cho.
Good morning, everyone. How are you guys doing? I'd like to welcome you all to the third day of the FDA-MHRA Health Canada Joint Symposium. It is my great pleasure to kick off the bioequivalence session today, and I highlight some of the work that we have been doing in the areas of bioavailability and bioequivalence. Today's presentations are a continuation of the discussions on important GCP topics that were discussed in the past 2 days of the symposium. However, this morning's focus will be on a specific GCP area, which is for bioequivalence studies. And that is slightly different than the conventional efficacy trial that we've heard for the past days. As you can see, this is a joint symposium with the MHRA and Health Canada and FDA. And as such, what we aim to achieve from this symposium is to share with you the collective knowledge and experiences from these agencies on inspections and regulatory oversight on bioequivalent studies, and to convey harmonized message with the hope that it helps you guys to do a facilitated drug development, global drug development. The international collaboration is one of the important strategic goals of our office, the Office of Study Integrity and Surveillance, as well as for the FDA overall. The OSIS Bioequivalence International Collaboration, specifically with the MHRA, has started more than 8 years ago. And as you can see from this symposium, the collaboration now has expanded to include Health Canada, which we started a few years ago. This international collaboration has been very successful because we all work together with a common goal of making sure that the regulatory data submitted to our respective agencies are robust and reliable, and the safety of subject and participant who goes into the clinical trials are well protected. As part of the collaboration, we conduct joint or observational inspections, exchange information about the inspection details and outcomes, clarify expectations to industry on study conduct when it's necessary, and share any tools that might help the regulatory oversight. Again, by working together, we hope these efforts convey a harmonized message to industry on expectations on study conduct of bioequivalent studies across the agencies, and help us streamline the planning and execution of your own studies. As for the topics of the presentation this morning during the bioequivalence session, we have 3 very exciting sessions planned. In the first session, we will present an overview on our experience on regulatory remote evaluations. When COVID-19 hit the world at the beginning of 2020, and essentially every aspect of our lives was impacted by that, our office in FDA led an innovation and did a really groundbreaking work, which was to develop a remote evaluation approach. And it was these efforts that allowed us to continue our work and to accomplish FDA's mission of protecting public health throughout the pandemic. Among other things, we crafted a communication plan with the industry, made sure that the remote approach that we are developing is as flexible as possible given the very fluid at the time, and made very best use of all the technologies and communication platform available, which, by the way, weren't that much at the time, as you know. Yet, we try to make sure that we maximize the efficiency and the evaluation capability so that we can make adequate review of the study conduct and study information and make robust conclusions about the reliability of study data. Initially, we called this remote tool as a remote regulatory review, in short, RRR. Then in April 2021, FDA issued a guidance for industry entitled Remote Interactive Evaluation, RIE, for drug manufacturing and bioresearch monitoring facility during COVID-19 public health emergency. And that was the first public announcement that explained FDA use of a remote tool. Since then, the agency recognized the need for formalizing a procedure for remote evaluation across a variety of compliance programs. In July 2022, FDA issued a draft guidance titled Conducting Remote Regulatory Assessment Question and Answers. And in there, it also explained that the terminology, the Remote Regulatory Assessment, RRA, that you heard of yesterday, is a category of activities that FDA uses for remote evaluation. And it encompasses the remote regulatory review and the remote interactive evaluation as types of RRAs. And as some of you may know, just a few weeks ago, FDA issued an updated draft guidance, which is now open for public comment for the 60-day comment period. In parallel to FDA's efforts, our colleagues in MHRA also developed a remote inspection paradigm. And through the collaboration that I mentioned earlier among MHRA, Health Canada, and FDA, we exchanged the tools and approaches, advantages and challenges, and notes on similarities and differences in how we conduct remote evaluations. It has been extremely valuable to learn from each other, and now I'm very excited to see how the two presentations this morning, one each from MHRA and the FDA, in the first session will inform you about the two agencies' remote approaches and provide you with the important take-home messages so that it helps you to get prepared for your future engagement and inspections by these agencies. So the next two sessions that follow, the RRA presentations, will touch on the actual bioanalytical and clinical aspects of bioequivalence studies and the perspectives from the regulatory inspections. Based on the current business models and industry trend, we see that it is generally the case that the sites that conduct the clinical phase of bioequivalence studies and those that test and analyze the biological samples from study participants are separate entities. And depending on the business relationship between those two sites, there could be more or less of communication between those sites about study design, sample collections, sample transfer logistics, and etc. And in some cases, there could be additional sites in addition to the bioanalytical and clinical sites that play various roles and functions during the study conduct. And as more entities get involved, it could easily get complicated when one tries to reconstruct all the study activities and to verify the integrity of study samples. And if there happened to be an information gap, there could be a delay in application review or even approvals. So given the pivotal nature of the bioequivalence study in generic drug approvals, you can appreciate how important it is for us to verify if subjects receive the right sequence of drug treatment and the drug concentration data submitted to the agency are reliable. So for these reasons, FDA, MHRA, and Health Canada conducts inspections at both clinical facilities and bioanalytical laboratories. As for the clinical phase, we expect the bioequivalence studies are conducted per GCP principles, and documentation is complete and contemporaneous. Studies should be conducted per protocol, and all deviations should be reported. Importantly, the rights and welfare of study subject or study participant should be appropriately protected. For the bioanalytical phase, we want to ensure that the method that was used to analyze the study samples is fully validated and works robustly during sample analysis. The tools and the processes that measure drug concentration should be clearly established and properly controlled. Throughout all the phase of the study, from the subject screening and enrollment to sample analysis to data reporting, we expect that a robust quality management system be in place. So I know many of you working in the pharmaceutical field put the patient in the center of everything you do and try to do the right thing. And we and the public really appreciate that. In some cases, however, we also have seen some concerns with data from study sites. I feel that we as regulators need to let the industry know of our expectations on study conduct and best practices and pitfalls we have seen. And that is exactly why we are holding a conference like this today. We perform a number of bioequivalence inspections each year, and the presentations from the 4 speakers in the 2 sessions this morning will highlight some of the common or unique observations from our inspections that are worth noting, so that you can pay attention to those aspects when you're conducting your own study. So we have a morning that is full of important and informative presentation, as well as opportunities for you to ask questions and discuss with the esteemed panelists. I would like to express my sincere appreciation to all the speakers and moderators who worked so hard to make today's event informative and impactful. I hope that you are as excited as I am, and I look forward to having engaging interactions and lots of great discussions. Thank you so much. So I'd like to hand off the podium to the moderator of the first session, Dr. Sean Kasim.
Jeff warned me about that. Good morning, everybody. Good afternoon and good evening. Really glad for everyone who could meet us here in the room and everybody online virtually. My name is Sean Kasim. Thank you to Julia for the keynote opening and welcoming me. I'm the director of the Office of Study Integrity and Surveillance at the Center for Drugs and FDA, and I'll be your moderator for our first 2 sessions this morning. Today we're in for a real treat. We have 2 presentations in our first session, as Julia alluded to, focused on the use of and the development of the remotes in bioequivalence oversight, both by US FDA and the UK MHRA. Our first speaker is Dr. May O, who plays a critical role in OSIS. She's the lead pharmacologist of our clinically focused BA/BE team in OSIS's division of generic drug study integrity. After May provides an update on how OSIS developed and has been using RRAs, our second speaker is Michael McGinnis, who will provide a perspective on remotes developed and used by the UK. Michael is the head of GLP and laboratories within MHRA's Compliance Team 1 and the head of UK's Good Laboratory Practice Monitoring Authority. I'm really looking forward to their presentations and our Q&A panels after session 2. Just to remind everybody, we've switched the order of the agenda today. We're going to have our break before our first question session, so it'll be announced from the podium at the time, but just to let you know that this morning we'll take a break after session 2 and then come back and have our session with the 3 speakers that we've had from sessions 1 and 2. All right, well now please join me in welcoming Dr. O.
Thank you, Sean. Thank you. My name is May O, and I really want to thank this opportunity to give a presentation today. It works. Okay, here we go. So my presentation is remote regulatory assessment, a valuable tool for OSIS to support drug application review in FDA. So this is my disclaimer. And for the overview, first, I'm going to briefly introduce who we are and what we are doing. And then OSIS oversee in vivo BAB studies, what we're doing for the remote regulatory assessments, and what we are looking forward for the future. So we are Office of Study Integrity and Surveillance, OSIS. So we are in OTS in Cedar. There are 2 divisions in our office. Division of New Drug Study Integrity, and Division of Generic Drug Study Integrity. Other than the two divisions, we also have immediate office, project manager team, as well as cross team. So all the team work together to support OSA's fundamental inspectional activities. Because of that, we do have several stakeholders in CEDAR, which we call review divisions. So we support Office of Clinical Pharmacology and Office of Generic Drug. We also have lots of collaboration of Office of Compliance, Office of New Drug, etc. Other than that, we do have lots of collaboration with Office of Regulatory Affairs, ORAs. So we do lots of inspections, ORAs together. So here is our office vision and mission. So for the vision, OSAS improves the public health by protecting study subjects and promoting properly conducted studies. For the mission, OSAS promote the public health by ensuring the welfare of study subjects and by verifying the quality, study integrity, and regulatory compliance of the BAB studies, GLP, and animal rule studies. So for what we are doing, simply say, we do select, evaluate, review, and support. For the select, based on review divisions consult as well as our Quest team site assessment, we do select the sites that need to be conducted inspections or RAs. After that, OSAS reviewers will perform the inspection or RAs, which is to ensure the integrity of the study data generated by those sites. So after inspection or RAs, OSAS reviewers will, based on all the evidence we collected, we generated EIRs or RA reports, as well as the OSAS review. So for all the activities, we support CEDAR review process and by give the data reliable recommendations as well as the compliance evaluation. For the typical in vivo BAB studies that we conduct in either inspections or RAs, usually there are two components. The first is the clinical component. For the clinical inspections or RAs, they are mainly conducted by ORA staff. Sometimes OSAS reviewers also will join them for inspections or RAs. For the clinical component evaluation, we evaluate the adequacy of the site, the facilities, the personnel, procedure, subject screening, enrollment, investigational drug administration, subject safety monitoring, protocol adherence, biological sample collection, and so on. The second component of the in vivo BAB studies for inspection RA is analytical component. So for this component, usually it's OSAS reviewers conduct the inspections or RAs. And sometimes ORA staff will join us. For this component, OSAS reviewer will also evaluate the adequacy of the site, the equipment, personnel, biological sample processing, method validation, study sample analysis, documentation, data security, and so on. So overall, OSAS oversee both of the clinical and analytical components of the in vivo BAB studies conducted. For the remote regulatory assessments that OSAS conducted, it's a virtual interaction between OSAS and the sites. So as we know that OSAS implement RA start from June 2020, right after pandemic started. In July 2022, a draft guidance was published, and as Julia mentioned, last month, an updated draft guidance also published. So there are several characteristics of the RAs conducted by OSIS. First is voluntarily participation. If the site agreed to participate in RA voluntarily, usually OSIS reviewer will request all the documents, records, or information being submitted prior to the virtual interaction. If there are some observations being, usually we call objectionable conditions, being observed during the RAs, those observations will be emailed to the site at the closeout meeting. So for the RA activities, we'll also provide the data reliability and subject safety recommendation to the review divisions. So RA activities actually allow OSIS to continue to oversee the conduct of the BAB studies, GLP, and animal rule studies during and beyond the pandemic, which is support CDER review for NDAs, BLAs, INDAs, etc., and also support FDA's mission to protect public health. So here I'm just briefly introduce the RA process and inspection. For the RA preparation, OSIS reviewers will review all the documents and information submitted to FDA during the preparation. Meanwhile, we will send an email to the site, ask for voluntary participation. If the site agrees to participate voluntarily, usually we just ask the site to provide all the documents prior the virtual interaction. Currently, FDA use the cloud filing sharing solution, which is box.com. For the inspection preparation, very similar, OSIS reviewer will review all the documents and information have been submitted to FDA. Opening meeting. Because RA is virtual interaction, so OSIS reviewer will schedule the meeting with the site through Zoom. For the inspection, it is in person. So OSIS reviewer will arrive at the site, present the credentials to the site. If it's a domestic, then FDA Form 482 will also be presented, and then have the opening meeting on site. For the facility tour, RA, usually we would like to ask the site to give us a virtual tour, so they can use any type of technology device. For example, any smartphones or smart tablets or camera. So they are going to show us the facilities in the Zoom meeting. For the inspection, then the facility tour is in person. OSIS reviewers will follow the site management or staff to visit the whole facility. Daily meeting. For RAs, usually OSIS reviewer will schedule 2 to 3 hours daily meeting with the site. So in that Zoom meeting, we will discuss any questions or concerns with the site management or staff. For inspection, the OSIS reviewers will stay in the site, review all the documents, as well as have the meeting with the site management and the staff. Closeout meeting. So if there are some observations in the RA, those observations will be written in email and sent to the site. But for RAs, we do not issue FDA Form 483. And for the closeout meeting of inspection, if there are some observations, then FDA Form 483 will be issued. So we can see from the side-by-side comparison of these two processes, there are some difference, but there are still lots of similarities between those two processes. Here's the studies that OSIS have conducted for the RAs. We do have lots of in vivo BAB studies, PK studies, ligand binding assays, immunogenicity studies that we conducted through RAs. And you can see we have even more variety of the in vitro BE studies that have been conducted during the RA. So all the activities actually is to support the regulatory decision regarding the drug application safety, efficacy, and labeling. So here I'm going to briefly talk 2 case studies that we conducted during the RAs. They are more focused on the analytical components of the RAs. So for the first case, here I just put what we observed during the RA. That OSIS observed that the site did not store the reserve samples for the reference product in an area segregated from the area where the in vitro BE study was conducted. So the story is that during the RA, after the opening meeting, we had a virtual tour. So when the site passing the in vitro study sample preparation and processing bench, we saw a small refrigerator standing just next to the bench. And then the site explained that refrigerator was used to store some chemical reagents and the reserve samples for reference product. So we did ask the site management to open that refrigerator for us. And then we did see in the upper shelf, there are some chemical reagents stored there. And in the lower shelf, the reserve sample of the reference product was stored there. And the interesting thing is that the site did have the reserve sample of test drug product stored in a designated refrigerator, which is located in a designated storage room. But for some reason, the reserve sample for the reference product stored in that small refrigerator next to the study bench. So we do collect the evidence that, and as well as the photos when the site is submitted to us, we do collect the evidence that those reserve sample for reference product was stored not appropriately. But in the meanwhile, we audit lots of other documents, like, for example, drug accountability documents, shipment receipt, and we also audit the method validation study report. And for us, we see those, even though the site did have a regulatory violation, but not appropriately store the reserve sample for the reference product. But that misplaced of the reserve sample did not impact the study data generated from the site. So for us, our recommendation to the review division is that the data are still reliable, generated by the site. So when we give our recommendation to the review division, that will allow the review division continue the review. The second case study is a pivotal in vivo PK studies to support a BLA. So during the RA, OSIS finds that there are about 36 samples drug concentration documented as higher than LLOQ in the source data, but they are documented as lower than LLOQ in the submission to FDA. So we observe this discrepancy, and then we alert the review division, say, hey, there is a discrepancy between the source data as well as the submission. So that will allow the review division to send the information request to the sponsor and also allow the sponsor to give the explanation. So after that, the review division decides that explanation is appropriate and acceptable, and that little discrepancy did not impact the final PK analysis. So our RA recommendation also allow the review division to continue their review for that BLA. For the future, what we are looking for. OSIS will continue to improve and refine RA for its remote process and effectiveness. And RA is a critical and valuable tool that OSIS will continue to use to assist FDA in accomplishing its mission of ensuring public health and safety. Last but not least, the data submitted to FDA should be complete, accurate, and reliable to ensure safety, efficacy, and quality of drug products. Thank you very much. Okay. So I'll give this platform to Michael. Thank you.
I'm going to apologise for making this noise. Oh, that wasn't bad. Good morning, everyone. To those joining us online, good afternoon, good night. And that is the highlight of this talk. So Sean said, my name is Michael McGuinness. I am joining you from the UK MHRA. I have been well warned to slow down, and that will happen during the two presentations that you have from me today. I'm going to give you the MHRA's perspective on remote and hybrid inspections, to build on some of the sessions that you would have heard from Dr. Seller, Jennifer Adams, and Rachel Mead from the MHRA yesterday. What I'm going to do is talk about a specific bioequivalence slant, talk about some of the differences, and then provide some tips, logistics, feedback around the development. So without too much delay, we'll give you an overview of the bioequivalence program at the MHRA, talk about who conducts the inspections. We'll go through the inspection types. There is a small amount of audience participation. We'll talk about when the MHRA may make use of these types of inspections, and then we'll give you those, hopefully, hints, tips, things to think about. So, in terms of the inspection team, and contrary to what you'll have seen is the MHRA bioequivalence inspections are performed by the laboratories and GLP team. The team that I head up, that is a multi-GXP team. So we have, as the name suggests, GLP inspectors, but also those that conduct GMP quality control laboratory analysis, inspections, GCP, and as you would expect, bioequivalence and biosimilar inspections. We do have invaluable support by our GXP colleagues. Some of them are joining us today, joining from the GCP team. On the right, you can see my colleagues who obviously can't be with us today. We will hear from Emma Whale later on, and just to the development of the MHRA's hybrid inspection program, Emma was crucial to that. So I want to just acknowledge that. And Emma, if you're watching, hello. The one thing that, again, just to mirror what we talked about with Dr. O, was the MHRA's inspection program is both by the clinical element and also the bioanalytical element coming together. Now, you will see this slide again, but there are a couple of different elements I want to pull out. So you will see on the right-hand side the deficiencies that we have raised and pulled together for metrics between 2019 and 2003. But we'll come to that in the next session. We've conducted 24 bioequivalence inspections since 2019. Now, obviously, there is the downturn from the pandemic where we adapted. That also doesn't include any of the UK inspections that may be classed as bioequivalence, but would be considered a GCP laboratory inspection or a straight GCP inspection. And then the date that you can see there, so remote or office-based inspections were conducted between October 2020. We weren't quite as quick as the FDA. And that ended in June 2022. The reason for the delay we will allude to as we go through the presentation, but we recognise the crucial role in data review and weren't willing to compromise the outcome of the inspection until we had the correct tools. So, audience participation. I apologise to those online. We are going to talk about each inspection type as we go through, but for those in the room, I imagine if we asked you who had done an on-site inspection or who has hosted an on-site inspection, hopefully many hands, has anyone been through a remote inspection? Okay. Anyone been through a hybrid inspection? And then keep your hand raised if it was a good inspection. The FDA aren't watching. So yeah, we'll take these each in turn and talk about the development at the UK MHRA and how we brought that about. So this slide I call an inspection in a nutshell. Across the top in the lovely red, orange and blue, you will see the inspection flow. It's very deliberate that you will see that on every of the subsequent 3 slides. So you can see that the remote, hybrid and on-site inspections will follow the same process. So if we take it in turn, the notification and data requests, what we imagine is when that email lands in your inbox, there's a calm, considered response to what you're going to expect. At that point, it doesn't feel any different. You should know who your inspectors are, you will know the dates, you will have some information about what we intend to do, and also there'll be a long or short list depending on the scope of the inspection of requested information. We move into the inspection, which we will talk about, and then report and closure. The point to note here is the MHRA don't distinguish between remote, hybrid or on-site. We will produce an inspection report, and in GCP you will get a statement of inspection detailing how it went, whether there were criticals, majors or anything else. The part below that is a very high-level review of what we consider to be a labs or a bioequivalence inspection. So at the centre, we've got 4 big buckets, if you like. So we have the facility review, classically called the tour. You'll see that is the smallest of the 4 components before we move into interviews, discussions. Then we go up to systems reviews, so looking at quality management systems, quality assurance, quality control, document control, the list goes on and on. But the largest component, and we'll talk about this in a second, is for data review. Crucial to the bioequivalence inspection conduct is our ability to verify and reconstruct the information that you have generated during the conduct of the trial. We have heard from colleagues from GCP about real-world data, decentralized trials. A bioequivalence study, in its essence, is not that complex. It's usually open label, 2-product, crossover, and done in a relatively short period of time. The data that you generate during it, whether that's the clinical or bioanalytical, is crucial to our assessment when we're looking at it. So, the trained observers amongst you will see that my title says hybrid and remote, but we're talking about on-site. The reason I've put this here is just to enforce and highlight the development of the tool. You'll see that, as you imagine, all activity is performed on-site. But what we've done in moving through the pandemic is realised that it is worthwhile us taking use and investing in our staff in terms of training, but also bringing in the analytical data systems. We won't go through them here because I can't endorse any of them. But we hold a number of software that are prevalent within bioequivalence studies. We request that data through Microsoft Teams, and we work within our own systems. This is a couple of highlights on our side. One, the training. It means we don't need to bother you on site. It helps with license management from your side, and then means that we can share in real time, but allows the development of this process. Remote inspections. As you can imagine, all activity will be performed remotely. So this was from October 2020. What we did here, and to highlight, is exactly as Dr. O talked about, is the use of visual technology to bring back that facility tour or a facility walkthrough. Now, we've trialed quite a lot of systems. We've used Microsoft Teams, Zoom, the list goes on and on. We've even made use of some augmented reality to test whether or not that would be worthwhile. it was mostly dizzy. But in terms of the remote inspections, again, it should feel like an inspection. You will see the boxes will definitely take longer. Referencing Jennifer's slides from earlier this week, an on-site inspection, the actual conduct generally is shorter, not factoring in travel. But where you start to be into remote inspections, and we'll come to it as we go to the tips, that element takes longer to pull together. What would normally be put a lab coat on, straight through, find the stairs, go there, turns into, right, we have someone that is possibly on the right side of the air shower, someone that is ready to go, multiple cameras. And those are a lot of hurdles that delayed our deployment of the remote inspections. Then moving into hybrid. So as again, you can imagine a hybrid is a cross between a remote and an on-site. These are not mutually exclusive, and the one thing to say is that during the pandemic, and I hope none of you have done this, but if at any point there was a reason to turn a a remote inspection into a hybrid inspection, that option is open to us. So we can escalate, so you go from remote to hybrid, potentially, and it brings the on-site component to it. One example was, as soon as we mentioned the word critical finding, the camera went off, the microphone didn't. And it was interesting. So the hybrid inspection, again, pulls together all of those components. So we are working within Microsoft Teams. We'll invite nominated colleagues from your sites into our Active Directory. We will deal with all the communication. We will request documents. We will host meetings with you, provide constant feedback. We're there on Instant Messenger if you need us. Video calls, tours. And it should, again, feel like a real inspection. There are segregation of activities. So in the nutshell part, you can imagine the facility walkthrough will be done on site. The systems review will be split. Data review will be a large part done remotely in the United Kingdom before we turn up on site. So the idea is that we would run through all of the analytical data and turn up with questions, therefore making best use of your time and ours. Interviews, again, possibly split. So when will the different types be used? And again, this is dependent on several factors. The first one is compliance and inspection history of the site. So we take each component in turn. The compliance, whether or not that was good, bad, or indifferent, will determine whether we're there. So if we're looking to confirm closure of a critical finding and that related to a facilities and equipment issue, it makes sense to be on site. If that related to a software validation issue, which we could bottom out prior, then you might see us as a hybrid or remote inspection. The inspection history, the point to note here is we will predominantly, if you are the first time you've had an inspection from the MHRA, we want to see what the facility looks like, walk through, for want of a better term, make sure all the lab coats are in order, and then continue there. So you are likely to see an on-site inspection. The nature of the inspection is the second part, so whether it's routine or triggered. So the MHRA conducts systems-style inspections for, if you like, frequent flyers of bioequivalence generating sites. Whereas if it's triggered, looking at a specific issue, what we might want to make use of is the effective use of your time and our time. So possibly lends itself to an on-site inspection. And then the last one, and we'll talk about this as part of the review, but the suitability of the inspection site. If your Wi-Fi is so terrible that you can't download a PDF, it is unlikely that you will be able to support our Microsoft Teams video tour. So we will ask those questions up front. We possibly switch to a hybrid inspection after that notification, and then we move forward. So this section is related to genuine feedback that we've received as part of the development of the tool. And as we continue to do this, I would encourage you, if you are subject to a remote MHRA inspection, a hybrid MHRA inspection, please do continue that feedback loop. So the use of visual technology. This one is crucial as we work through, but ensure sufficient connectivity through the facility, whether that's through Wi-Fi, 4G. What typically happens is when you approach an LCMS, all of the systems go down. It doesn't like the interference. Consider the movement between areas. It's very easy just to consider that an air shower is normal, a water shower is very difficult for a computer, for a phone or a camera to go through. So yeah, bear that in mind. Possibly multiple devices is what we've seen people do. Time zone challenges. So as I mentioned, the bioequivalence inspections we're talking about in our metric here are our overseas inspections conducted, whether that's in India, whether that's in the US, whether that's in Canada, to name a few. But the UK obviously is not in that time zone. So you may find that you can see us for part of your normal working day. So again, discuss this as part of your planning process with your lead inspector. We are used to this and well practiced. It may mean that we'd have 4 hours with you, and then when that goes down, we will work on our own, submit document requests, and then arrange a meeting to discuss that with you the next morning. There is not an expectation for you to work through the night, nor turn documents around at you or 12 o'clock. So agree with colleagues when they will be available to support the inspection. Again, your lead inspector will walk you through this. The second part is in providing the analytical data. So one of the things that we learned is that everybody's experience with analytical data varies. How you can upload it, whether or not you zip it, all of that we have walked through. So we have come up with a guide for the majority of the systems that we have, and that will be provided to you to try and take some of the thought process out. So if you follow that, you should be all right. If you are encountering any challenges, please let us know. It's a crucial part of the inspection for us. And we have found on occasion where we've started the inspection, think that we've got everything, and then look, lo and behold, all the whiff and injection files are missing. And what that does is it raises the inspector's blood pressure and you start off on the wrong foot. So avoid that. Do share. So in inspection progress, what we'll do is we will try to arrange this as much as possible, but agree feedback sessions. As we said, depending on your time zone, that might be better the day after. We will share our concerns. If there is anything we perceive to be a deficiency, certainly at the critical or major end, then we'll do that there. We'll give you time to understand our thinking, potentially provide a rationale for why that might be the case, and then we move forward. Inspectors should share feedback as they go through. In terms of the last one, so during the inspection, we've talked about a number of systems, so whether that's Microsoft Teams, Active Directories, whether that's the analytical data, if the use of a system or process is causing you a hindrance or administratively burdensome, let us know. It's not designed to be a stress test for you. Most of us have been on the other side and received the exact same treatment. So we are trying to get as full an understanding of your systems, your procedures, in order to make a relevant compliance assessment to our assessors. It is not designed to put you through the ringer. So come back to us, liaise with your lead inspector and provide that feedback to us. There are a number of avenues open to us to change document requests, to switch systems within reason. If you tell us you don't want to give us your analytical data, that's going to be a red flag. So in summary, we've embedded the new tools during the pandemic into routine processes. You can see that with the on-site inspections. That analytical data upload took us a long time to put right. It is something that we have worked hard on and will continue to improve and expand the number of systems that we have, but it's definitely been a big plus for us. We continue to look for new ways to improve and our remote and hybrid inspections. I do have a Disney quote, but I'll come back to that. Your feedback on the inspection experience is, as always, welcomed. As with anything, the MHRA prides itself on its transparency and pragmatism. We talk about it during our training and accreditation, and it's something that we would encourage. There is no stupid question during an inspection, within reason. And then the last one, before I give you the quote, is the aim of the inspection, regardless of the type, remains the same. We want to understand your systems, your procedures, your quality of data, your protection of the subject, it's with the study participant and the well-being. And that, before I finish, is the quote that I have for you is, find the right tool and you'll make magic happen, from Toy Story, where Woody tries to encourage teamwork. And with that, Sean, I'll pass back to you.
Thank you so much, May and Michael. Really appreciate your presentations this morning. Now, for our next session, we are taking a focused look at the inspections of analytical conduct of bioequivalence studies. So in addition to another presentation from Michael, which you all get to look forward to, we are also pleased to introduce another essential member of OSIS, Dr. Igwe Cynthia Zhang. Dr. Zhang is a senior staff fellow in OSIS. She plays the integral parts of conducting, coordinating, and reviewing comprehensive FDA BIMO clinical, analytical BE inspections, as well as supporting our GLP team. So please join me in welcoming Cynthia this morning to speak on bioanalytical issues. Cynthia.
Thank you, Sean, for the kind introduction. Good morning to everyone in the room, and good afternoon, good evening to the online audience. I am thrilled to be here today to talk about some bioanalytical issues found from the recent FDA BIMO inspections and remote regulatory assessments, RA. Here is my FDA disclaimer. The learning objectives of my talk today include to get familiar with the relevant US laws and regulations, to understand the basis for demonstrating bioequivalence, BE, and also to make you aware of some bioanalytical issues from recent FDA BIMO inspections and RAs. And here is the outline of my talk today. In 1984, the Hatch-Waxman amendments of the Federal Food, Drug, and Cosmetic Act introduced the new abbreviated approval pathways of marketing applications. Specifically, a 505(b)(2) application is an NDA containing full reports of investigations of safety and effectiveness, where at least some of the required information comes from studies not conducted by or for the applicant, and for which the applicant has not obtained the right of reference or use. Meanwhile, a 505J application is an ANDA for a drug product that is a duplicate of a previously approved drug product, and generally must contain information to show that the proposed generic product is pharmaceutical equivalent and bioequivalent to the reference listed drug, RLD. Additionally, the Code of Federal Regulations, CFR, Title 21, also includes relevant sections. For example, I listed here, there are Part 312, the Investigational New Drug Application, Part 31454, Part 31492, as well as Part 320, Bioavailability and Bioequivalence. Here, I am showing you all the table of content for CFR 21, Part 320, Bioavailability and Bioequivalence, which lays out the legal foundations for requirements of bioavailability and bioequivalence studies. You may find the details online if interested. In Part 320, the basis of demonstrating BE is defined. Specifically, two drug products, test and reference listed drugs, RLD, are considered as bioequivalent if they are pharmaceutical equivalent or pharmaceutical alternative. And the rate and extent of absorption do not show a significant difference when administered at the same molar dose of the active moiety under similar experimental conditions, either single dose or multiple dose. And if the rate of absorption is different, the difference would be intentional and reflected in labeling. And the extent of absorption should not show a significant difference between the test and the reference, or the RLD. For drug products that are not intended to be absorbed into the bloodstream, BE may be demonstrated by using scientifically valid methods that are expected to detect a significant difference between the test and the reference products in safety and therapeutic effect. In addition, FDA also publishes guidance documents to describe the agency's current thinking on certain topics. The guidances do not establish legally enforceable responsibilities and should be reviewed only as recommendations. For example, here I'm showing you the guidance M10 bioanalytical method validation and study sample analysis published in November of 2022. This guidance describes recommendations of the bioanalytical methods used to measure concentration of chemical and/or biological drugs in biological matrices obtained in all phases of clinical trials in regulatory submissions, including comparative BA or BE studies. And the table of content displayed here is showing some expected critical elements during method validation, including selectivity, specificity, matrix effect, calibration curve and range, accuracy and precision, carryover, dilution integrity, and stability. Last but not least, the FDA also has the Bioresearch Monitoring Compliance Program, BIMO. It's a comprehensive program of on-site inspections and data audits designed to monitor all aspects of the conduct and the reporting of FDA-regulated research. The BIMO was established to assure the quality and integrity of data submitted to the agency, FDA, in support of marketing applications, as well as to provide protections of the rights and the welfare of human subjects and animals involved in FDA research, FDA-regulated research. Among several BIMO compliance programs, the program 7348004 describes the coverage of a typical in vivo BABE analytical inspection, like I'm showing you here. Again, you may find it online if you're interested for more details. Now, let's switch gear, and I'm going to talk a few case studies. The first case study, this one, is a BE study with PK endpoint submitted under ANDA. The drug product was a small molecule and also an endogenous compound. The study was designed as a randomized 2-treatment, 2-sequence, 4-period, crossover, single dose, and full replicate trial. And the bioanalysis of the samples was conducted using LC-MS. During the FDA inspection or RA, objectionable conditions were observed. Specifically, there were higher pre-dose analyte concentrations observed in the subjects that's receiving test products compared to the reference listed drugs. Not all the subjects, but only some of them. And also the pre-dose analyte concentrations did not exhibit expected physiological variability in the subjects. The firm being inspected could not provide adequate explanation or justification of the above observation. No discrepancy was identified during sample analysis. However, like I mentioned before, the firm could not offer any explanation or justification would support the observed pre-dose discrepancies between the test and the reference products in this subsets of the subjects. Therefore, OSIS concluded that the analytical from the clinical study are not reliable for agency review. Case study number 2. This was an in vivo BA study with PK endpoint submitted under a NDA. The product was a large molecule and was analyzed using ligand binding assay, LBA. During the inspection or the RA, objectionable conditions were observed. Specifically, the assay actually did not demonstrate sufficient accuracy to measure the analyte concentrations. Specifically, the calibration standards and quality control samples, they were not prepared using a reference standards of known purity. Nonetheless, the calibration standards were used to back calculate, assign, and report specific analyte concentration for the subject samples. This subject's concentrations were later on used to evaluate and support the endpoint in the clinical studies. OSIS concluded that this observation impacts the study data reliability because the site did not use analyte reference material of known purity to prepare the calibration standards and QC samples. Thus, it is impossible to calculate and report accurate analyte concentration of the study subject samples. However, the percent change from baseline analyte data should be reliable because the method actually demonstrated adequate precision when measuring instrument signals from the QC samples. That is, the method consistently measured higher signals in high QC samples and low signals in low QC samples with adequate reproducibility. OSIS inspectors also verified that the instrument was properly maintained and calibrated according to the firm's SOP and CLIA requirements. Thus, the method showed adequate precision and the percent change from the baseline analyte data were reliable. All right, now it's time for some challenge questions. Challenge question number 1, true or false? You must follow all the recommendations listed in the FDA's guidance for industry for bioanalytical method validation and sample analysis. A, true, B, false. Can I get a show of hands? Who's picking A, true? Half, maybe 0. Okay. Option B, oh, sorry, I gave up. Yeah, so the answer is false. Like I mentioned before, earlier in my talk, the FDA guidance documents, they are not legally enforceable and they should be viewed as the recommendations only, and it reflects the agency's current thinking on certain topics. All right, challenge question number 2. Which type of evidence may be used to establish bioequivalence, BE? A, comparative PK study. B, comparative PD. C, comparative clinical endpoint. D, in vitro in vivo correlation, IVIVC. Or E, all of the above. Can you guys just shout out what you would choose? Perfect. So all those about 4 study types could be used as evidence to demonstrate bioequivalence. And here are some references that I mentioned during my talk. And if you are interested, you should be able to find them on the FDA websites. With that, I would like to thank my OSIS colleagues and management support to make this presentation possible. Thank you so much for your attention, and I'm happy to answer any questions you may have during the panel discussion. Thank you. All right, now let's welcome Michael to the stage, and he's going to talk about MHRA's perspective in their findings from recent inspections. Welcome, Michael.
Hello again. So as Cynthia said, I'm going to take a UK slant and talk about bioanalytical observations and findings from recent inspections. Again, just to bring this slide back up, we'll talk about something different. So concentrating on the very well put together graph, 3D image you can see on your right, distribution of findings. We'll go through not in a lot of detail, and you will pick up some of the clinical ones from Emma's presentation later on. But there you can see the number of critical findings, major findings, and other findings that we've raised since 2019. The aim of this presentation is slightly different. We have a slightly mixed audience. We've got pharmacovigilance later. We've got bioequivalence in the morning, both clinical and analytical. So the findings that we talk about here have been deliberately selected to try and give you food for thought, and with the idea in mind that if you take care of these, you won't end up in the critical finding list. Just in terms of, I said there'll be 2 audience participations, but one of them includes a prize. For this one, by show of hands, can I ask those that are directly connected with regulated bioanalysis in the room? If you're online, please feel free to raise your hand, I just can't see you. So anyone directly connected with regulatory bioanalysis? Hannah, thank you for your hand. Okay, that's great. There is no problem. We will walk through all of these together. So as I said, here's a breakdown. You will find this again in Emma's slides. So in the red or orange box, depending on what you can see, that is what we've classed as our clinical findings. And Emma Whale will go through that in session number 3 later today. What we are going to talk about is the major findings in the blue box. So as you can see there, method validation, data integrity controls, and clinical sample analysis. As I said, the intention here is to stop those findings turning into these findings. It has been very deliberately ordered that you can see method validation, data integrity, which is the, if you like, big brother or sister of data integrity controls, and clinical sample analysis. So as I said, we've got 3 examples to talk you through. Just in terms to give those context, some of you may have experienced an MHRA inspection in the past. We classify our findings in those categories. So a critical finding is where a significant departure from the principles of GCP have been identified, which either put the subject, clinical trial subject's well-being at risk, or the integrity of the data cannot be relied upon. A major is something that, if action is not taken immediately, will result in a critical finding. So if you like, it's a warning shot. So just to take that down, we'll break this into the method validation example I've selected is around stability. The clinical sample analysis will be on run acceptance, so how you decide whether or not an analytical run is valid. And in terms of data integrity controls, we are going to talk about dynamic data. So what I want you to think about as we go through these examples is making effective use of regulatory guidance. Cynthia's talked about that. Guidance is exactly there. It is not a thou shall, but if you're going to do something different, you need to think about it, document it, and go through. The idea of transparency and how crucial that is, particularly where you're going to do something innovative or novel. We want to be able to reconstruct your thinking, your processes. We don't want to rely on your colleagues' memory when you've won the lottery. And the last part there is data security and control. It is unfortunate that a number of inspections in my 9 years at the agency have resulted in negative outcomes, not necessarily because the method doesn't work, not necessarily because they weren't diligent, but because the system which you had acquired the information to leaked information. It was open to abuse, and it's one of the things that is incredibly frustrating as an inspector. So, to go into method validation, on the picture you can see there is a lovely -80 freezer with some samples. What I want to think about is long-term stability is the assessment of stressing the samples by putting them in to a freezer designed to mimic subject samples during the clinical trial and pulling them out at defined points. If you think about it like a lottery ticket and waiting for the draw, you put them in hope. And then as each number comes up during the draw, you get a little bit closer to the jackpot. You get to design your subject samples. The bit I want to talk about, and what we've seen on inspection, is that a number of laboratories have failed to analyse the prepared samples, stability samples, immediately after generation. So if you like, you're your lab scientist, you've got your lab coat on, you've got all of your samples in front, you are spiking your low quality control samples and high quality control samples ready to freeze. We'll go to the guidance part in a second, but at that stage, you have a huge amount of variables that can affect the outcome of the assay. Whether the analyst's pipetting technique, if that's what you're doing, is on point that day, whether the tip has got a manufacturer's defect, whether or not there is something else at play within the method. The idea of doing the analysis up front is to give you as clear an indication of what you can see later on, that you knew it was all right when it left there. Now, again, I'm going to share some ICHM 10 references, and Cynthia's links are there because I tried to hyperlink this and it didn't work. So what I've done is taken the direct wording from ICHM 10. So this is specifically section 3.2.8, handily titled stability. And the part in bold is unambiguous. It talks about aliquots of low and high QCs are analysed at time 0. Going back to my hamstrung lottery example, it is the one thing that you can do to improve your chances of the jackpot. It gives you future information about it was okay at the time. And it's a missed it's a missed opportunity if you don't take it. So what we'll do there, the text you can see currently is a slimmed down version of the deficiency. So laboratory procedures did not contain a requirement to analyse stability samples immediately following preparation. Therefore, there is no assurance that stability samples met acceptance criteria prior to storage. Now, this has a number of outcomes as we work through it. But at this stage, long-term stability is something that you do right at the start. It's done in method validation so that you can lay your samples down because you have to wait for them to age. It is not something that if you get to the end of your clinical trial that you can jump back. You have lost that opportunity. So what can you do if this is your facility? And this is a very common finding that we're seeing currently. We've presented on it in the past, and I'm hoping in talking about it today, I can stop writing that finding. So the first thing is to recognise that you have a problem. Look at whether or not you have time zero samples. And I will find most of you that work with laboratory scientists, they are quite thrifty. They will turn one experimental procedure into many. So you might find, although it wasn't designed to be time zero, that your low and high QCs might actually have been analysed by luck. And some of the cases we find on inspection, that's exactly what's happened. So that's the first case scenario, what I would call the best outcome. The next part is if you don't have that, then you're starting to look at the reliability of the long-term stability, how it's deteriorating over time, the percentage of nominal for your low and high QCs. And that's a really difficult thing to do because what you're doing is replacing an experiment with a human's opinion. And as all inspectors will tell you, we would much rather have data. Now, it's not the end there though. You might have seen that your time point 1 to time point 5 all come out beautifully in line with nominal, plus or minus 6%. So well within acceptance criteria. But you might also find that time point 1 passed and time point 3 passed, but time point 2 didn't. And you're left in that horrible situation where you can't say it was okay when it went in. And that situation crops up more than you would think. So again, try to incorporate this into your procedures as early as possible. If you don't have it, it's not the end of the world, but you should start documenting that now. The next one we're going to talk about is clinical sample analysis. And again, this is run acceptance criteria. So I know a number of people in the room don't work directly in regulated bioanalysis, but this is the part when you get to the end of the assay, you've got your calibration curve, you've got your QCs, and what you're doing is looking to see if you can trust the unknown concentrations that you've got as a result. So those are incredibly well designed. They're to take away any subjectivity or ambiguity that it shouldn't be my decision or your decision. It should be, that's what we've agreed up front and that's what we're doing. So what are we seeing on inspection? The inclusion of company-specific criteria. So we've mentioned ICH M10, FDA have their guidance as well. In addition to that, previous to that, you have the European Medicines Agency's Bioanalytical Method Validation Guidance 2012. And what those talk about is a defined list. So you're looking for assay accuracy precision, you're looking specifically at the calibration curve, whether or not you can regress lines against it, you're looking at your extraction method through QCs, you're gathering a wealth of information and then putting that back in. So that's one thing. If you as a company decide we want the best data, we want our gold-plated analytical results that are irrefutable to a regulator, and we're going to insist on this. That is, as we've said, a possibility. Document it in your SOPs. The second part and bullet point 2 is that these additional criteria are not always applied consistently. This is where you end up in a problem with the inspector. Because as we said, bioequivalents are slightly different to some trials. The design is relatively simple, 2-period crossover, open label. The outcome for a comparative PK assay is very well defined. You know what a pass-fail is. Most regulators, all regulators I would say, will be able to tell you that, a confidence interval of 85 to 110, or 90 to 110. So now you've got this real fear that bias, whether it's conscious or unconscious, can be introduced by the decisions that you make. So something that would normally have been regulatory guidance, unambiguous acceptance criteria, suddenly becomes, that's just an SOP deviation. And that's the dangerous place to put an analyst or a scientist in, which might mean that they have to work a double shift at the weekend. So take the subjectivity out. What I would say, if we go into, again, it wouldn't be a regulator's presentation without where we're taking this from. So I've slimmed this down from ICH M10. But again, it's really clear. It talks about rejected Cal and QC calibration curve. This is a specific example for a comparative PK assay, where the company were including a criteria. If I go back. Nope, apologies. Where the company had included their own criteria that if they lost the lower or the higher calibration curve, then the run would be rejected. Regulatory guidance incorporates the reason why you might not want to do that, so long as your QCs are covered within that run. And for the example that we're talking about here, we saw a selective use of this criteria being applied. So, again, on the right-hand side, from there you can see the deficiency that we would raise. So laboratory procedures were found to contain issues with respect to analytical run acceptance criteria, where requirements were not in accordance with guidance. Consequently, and this is the part where we're talking about bias, the policy for analytical run acceptance would have otherwise been, as a consequence, the policy for analytical runs, which would have otherwise been considered acceptable, were repeated and the original analysis not considered for pharmacokinetic output at the end of the trial. Now again, in terms of considerations, one thing that I would challenge, whether you're working with a bioanalytical lab, you're a study monitor, is one, are these additions essential? Are we talking about gold plating, or is there a scientific reason for why this needs to be in your comparative BE study? And if they can't answer that question, my suggestion is that you ask them to work to the guidance. You take away any form of guidance. They might have a very well-documented rationale. Again, when we've seen this, where we've seen that it being applied inconsistently, again, one thing that we've struggled with is that documented rationale. Why are you doing it? Why is it in place? What can be done? Okay. So, the last example that I want to talk about is data integrity controls, which seems like I haven't stopped talking about it since 2018, and the use of dynamic data. So what are we seeing? And again, this is in the major category. We have examples where this has failed and we're talking about criticals, but this is the precursor to there. This is where you have the opportunity to affect change. So what are we seeing on inspection? We're continuing to see static data being used for review of bioanalytical data, which has been acquired in a dynamic format. Just skipping ahead to the MHRA's guidance and a plug, other guidances are available, including the FDA's. Raw data must permit full reconstruction of the activities. Where this has been captured in a dynamic state and generated electronically, paper copies cannot be considered as raw data. What I'm going to do is take you through an example, and this is where the prize and audience participation comes in. What we've got here in the picture is a chromatogram, and it's been used for stability assessment. It is generated in a system with well-defined and what I would call broadly GCP and GXP data integrity controls intact. If we go slightly closer, let's do audience participation. This study was from a comparative PK study where you had LCMS, MS, but not to mislead you, but this chromatogram better shows my example, but it is not from an LCMS, MS. Anyone in the room want to offer a potential, what method this might have been from, what machine? And I can offer you a carbonated Scottish soft drink. Feel free to shout out in the room. Again, online, I'm really sorry. Not LCMS, MS, anything else that would be in that chromatogram generating sphere before I open it up to my colleagues on the right-hand side. Okay, that is fine. Anyone from the inspectorate, whether UK, FDA or Health Canada, want to offer? No, I'm running ahead of time. No. Other soft drinks are available. Okay, so this again, we're talking about dynamic data. What this company were doing was printing out the chromatograms and presenting it for review. Now, again, everybody in the room, I imagine, has some experience in quality assurance. There's something that sticks out about this, something that doesn't appear right. That is a lovely gap down the bottom. So the chromatogram printout was modified prior to the review stage. And what I would say here is the removal of the acquisition date and time sticks out like a sore thumb. when you highlight it. Now, the reason this is a problem is we're talking about stability. It's not the chromatogram itself. The chromatogram actually was acquired very well. It was within acceptance criteria. It can be used. But the timing, the metadata when you acquired that injection is incredibly important, because what it does is show that I can rely on your data. What they'd done is forgot to take the sample at the right time. So the scientist, in some misguided attempt, said that's fine. And they waited that out and submitted it to QA and QC, like all the way through until we turned up on inspection. And we are used to looking at gaps, so we found this. So in the interest of time, I'm not going to read the deficiency. But again, it's there. You can see that the dynamic state is crucial to it. One, you've spent time, money, generating a system that can generate all of your analytical data with audit trails. It's got a Watchman directory that moves it out of a modifiable format. You might have Active Directory to link it directly to an individual. There is no way of changing it. But yet you print it out and you let them submit it. So in terms of considerations for this, I want you to think about why would that be necessary. One, it's easier. I can take a piece of paper, I can go and sit at my desk. It takes a bit more thought to understand why you might want to then go to the system, go to the lab, have the yellow light, have the noise. That's not a conducive environment for review, but it's not the reason to not do it. The removal of the opportunities is only part of the puzzle. And I think I've gone through 3 examples, and what I would say is, if you remember nothing else, it's the second part of this. Creating a system that is tightly secured and tracked by audit trails, the modifiable data is moved by Watchman directory to a safe, secure location, the archive is all intact as one piece. Humans are relatively resourceful, and again, in 9 years, I've found that people have found a way if they want to do this. So what you need to do is look to achieve the right environment where they can make mistakes. Not doing the stability sample at the correct time is not the end of the world. Removing the acquisition date and time is. That is the one that breaks regulators' trust. We then talk about where else have the modifications come. And going back to that, that is a very difficult crack to fill. So, I want to pull this up. The idea of the talk was to try and take you from that wiggly blue box to remove them completely and avoid getting to this grey black box. What I want to leave you with is a short summary. Look at regulatory guidance. It is our current thinking. If you do have a good reason for doing something different, make sure you document it. Your decision making and the documentation around that is crucial. We don't want to rely on hearsay when we turn up to do an inspection. Exercising control of your data is important. Don't let all of your hard work with the CSV, the periodic reviews go to waste. And act before issues escalate. And with that, I'd like to thank you for your attention and pass back to Sean.
Once again, a big thanks to Cynthia and Michael. We are now going into a break stage. We have 20 minutes, so aim to get back at 10:20. And for those online, appreciate your flexibility with the change in the agenda. And we'll have our question and answer session for the first 2 sessions at 10:20. See you in a second. Thanks. On. Thanks for everybody. Starting our session 1 and 2 discussion panel with our presenters. Again, thank you for everyone for submitting your questions, and we've got a good set to discuss. So jumping right into it, we have an interesting question about our India office. And so in the FDA has an office in India, and the question is, do FDA personnel in India conduct FDA inspections? So Maid, would you like to answer that?
So would you mind to say which question?
This is, we have the FDA India office.
Oh, okay. So before I answer the question, can I say the disclaimer is still usable right now? So all the answer is on my own, not represent FDA's policy. Okay, so if that works, so I would like to answer this question. So the question is, what is the purpose of FDA office in India, and does FDA personnel conduct FDA inspection in India? Yes, we do. FDA do have Indian office, so there are inspectors present on site when we conduct the inspection in India. Meanwhile, our OSIS reviewers also travel to India to conduct the inspection on site. Thank you.
Thank you.
And,
Mike, you wanted to add anything?
Yeah, the second part of the question talks about, does the MHRA have an office in India? No, we don't. Our inspectors travel from the UK to wherever the bioequivalence site is located.
Excellent, thank you. Okay, well, we have another question for, I think, for May from your presentation. Can you just clarify the differences between the RIE, the RRR, and the RRA acronyms we've been
yes, for this question, I will have to defer to FDA guidance because we understand there are several names have been used for remote. So guidance already have very clear description regarding those names and the function. Thank you.
All right. Now, here's a question I think, Cynthia, maybe you could take. A question about the performance of RRAs, and is it performed, what time within the BA/BE study conduct do the RRAs get performed?
Thanks, Sean. That's a great question. Usually we would perform the RAs or inspections after the completion of the study, and the sponsor or applicant submitted the application to the FDA. However, during the RA or inspections, sometimes if the study was conducted, let's say 10, 15 years ago, we would also like to have a look of the firm's current practice. So we usually require or request like a master list of the BAB studies conducted during the past 5 years. And sometimes we would pick some more recent studies, sometimes even ongoing BAB studies, to evaluate the firm's current general practice of bioanalysis. So I think that helps.
Thank you, Cynthia. Any additions from our other panelists? All right. Now, here's a question about remotes in general, and we've discussed how they're voluntary, but what happens if a site refuses? Sort of a question for everybody. Anyone have a thought to share there? What are the consequences?
I think if I start off from an MHRA perspective, obviously we will ask the reason that you're refusing. We consider remote, hybrid, and inspections to be inspections, on-site, sorry. And I think with this question, it's the same as anything else. Our reason for inspecting is there to review the data and ensure it's suitable for an application. It is unlikely to shorten the application's length. It's more likely to prolong it, but we would engage with you at that point.
All right. Thank you, Michael.
Yes, similar to FDA. So if we meet the refusal for the RAs, at that moment, maybe there is no consequence, but FDA still have the authority to issue the inspection. If inspection would not be happen, hypothetically. So the review process could be delayed. So I think that's the consequence we can think of. Thank you.
And I think another example I can think of is some of the sites where they are located is under impossible circumstances for them to host the RA. For example, some are experiencing wars or there's some technical difficulties. So in that case, we understand, the difficulty, and we would try to do some remote data audit without the participation of those site staff.
All right, thank you very much. I think, the message of flexibility and the use of the remote tool really, as needed, is an important one. A question about observations and the inspection reports. Are observations included in inspection reports? I don't know. This is probably for everybody. Mike, do you want to?
So again, just from whether observations are included within inspection reports, yes. So whether it's hybrid, remote, we will incorporate everything into that inspection report and issue it to you, but that's our position. In terms of, we use observations in a slightly different way. So we have observations at the bottom, which may not necessarily be a deficiency. So just in case there's a terminology issue there, they might be things of note during the inspection, which are for your information.
Excellent. Yeah, good distinction. Here's a good one that we hear quite a bit is what's the reason for not issuing 483s with an RRA?
I can answer that because RA is not an inspection. So 483, the full name is Notice of Inspectional Observations. So that is why we don't issue 483 for RAs. Thank you.
All right. Now, question about feedback. How do we, as regulators, or do we accept feedback from a site post a remote? And is there an evaluation form being provided to help sites provide guidance feedback?
Okay, I can answer that. So we do welcome feedback. Currently, every time when we finish the RAs, we provide the email box, which is cedar-ocs-beq@fda.hhs.gov. So we welcome any feedback. And if you have any form you like to use, please also email to us. Thank you.
Yes, please.
So specifically in the idea of a form or template, the MHRA don't make use of one. The one thing I would stress is not to wait till the end of an inspection or a remote observation to raise anything. What your lead inspector should do is try and tweak the inspection to remove any burdensome conditions and work with you. And we do that in real time. So the feedback that I described during my first presentation was related to real-time feedback that we received during the inspection. Equally, we are fully contactable after the inspection, and we will take anything that you just didn't think of at the time but want to let us know.
Thank you. I think that's a really good point, that the more feedback we get, the better the tool can be developed over time. And so it's really important to have your thoughts shared as they come up. Now, regarding remotes and the discussion of virtual tours by video, is it required to have a live tour, or is it okay to have a photo tour or a pre-recorded tour? Anybody want to answer that one?
That's a great question. I would say that the live tour is highly recommended. Especially during the RA, you're welcome to upload the, photos or pre-recorded tour to box.com as you wish. But during the RA, we still hope to see your facility live, because a lot of things could have been changed since the recording of your tour or the photos being taken. And the live one would be, I guess, a better reflection of the current situation of your facility. And that will give us a better idea of how your facility operates. Thank you.
Excellent. Michael, go ahead.
Yep, just to say in terms of, we do receive photo tours, what I would consider a business development video at the start. One of the key things for an inspection is the interaction between the inspection team and the inspectee, and a video tour doesn't allow us that chance for collaboration, discussion and questions. So although you might provide it, we would always insist for a remote inspection that we do a live tour. If that proves cumbersome, then we might move to hybrid.
Excellent, excellent. Now, regarding, again, a question on remotes, and I know this came up in the past sessions yesterday. When do we see remotes being used in the future, past COVID?
I'll say when needed. So we'll still continue using RA as a tool past COVID. Thank you.
Excellent. Michael?
I think Dr. Sellers said yesterday, whenever. And it's true, the remote inspection is a tool. If there's a reason that we want to take a specific look at something, we will make use of that. It's not restricted to COVID. We've spent a lot of time developing these tools, as the colleagues in FDA have. And if it's to answer a specific question, then that might be very well the best way and route to do that.
Excellent, thank you. Let's see. Wrapping up a couple of there's a few more remote questions. One of them is, in terms of, written observations, and as you mentioned, written deficiencies really of a remote following an inspection. In terms of at least the FDA side of things, what's the status on classifications?
Again, because RA is not considering inspection currently, so FDA do not classify the observation. Thank you.
Thank you, May. And also, let's see, in terms of in conducting of the remote, what kind of information is asked for from the site?
For the BAB remote RAs conducted by OSIS, we really follow our own SOPs. Usually there will be communication letter 1 going out to the firm, asking if they are willing and able. If they say yes, we will continuously sending out communication letter number 2, which would identify the studies. And we will also include attachment A, which is the detailed list of the request of the documents. Usually those documents would include, I would say 2 to 3 parts. The first being the general administrative information, like the overview of your firm, your SOP index, your flow plan, your like the list of personnel involved in the studies and their training records, the list of equipment used and their calibration records. And then going on to the study-specific information, we would request for the all raw data. For example, if you use like a hard copy of the lab notebooks, we would like to see those, I don't know, either scanned copy or the electronic copy you already have. And then there would be, for example, the preparation sheets of your calibration standards and QCs, audit trails. And we may also asking for some electronic file, for example, the individual run, the results and acceptance criteria, and your sample repeat. Also the correspondence with the sponsor or clinical sites, if you're an analytical firm. And regarding sample receipt and the tracking, handling, chain of custody. In addition, if this is in vitro B study, there will be reserve sample requirements per 320 regulations. So we would asking for information related to the reserve samples for the IP investigational products, and also the storage conditions, again, chain of custody. And any deviations you have documented, or any investigation of failed runs, or justification of repeats. So just a few examples.
Excellent.
Yeah, but if you happen to be selected for RA, you will definitely see more. Thank you.
Thank you, Cynthia. Michael, do you want to add anything from UK's perspective?
The only thing to make a specific reference to, because I think Cynthia's list was very good, was just specifically on the analytical files we request. You will get notification of what that looks like. Traditionally, it's all analytical files associated with method validation, sample analysis, and audit trails that bridge the gap the month before, the month after. So month before validation and the month after sample analysis. But that could be subject to change.
Yes. And that actually leads into questions focused on session 2, is what are the specific software that MHRA holds and uses for review of analytical data?
And this is as of the current time. We're currently looking to install and embed others, but the current list is SciEx Analyst, SciEx OS, Waters MassLinks, and Empower 3. That list is growing. We are looking at seeing what's the most prevalent software within industry. We're looking at adding LBA assays, for example. But yeah, as of this time, that's the current list. But it's fair to say that we have now worked through that process and can add to it.
Excellent, excellent. All right. Now, of course, something that's on everybody's mind is the use of AI. And so one of the questions that we got was, what is the use of AI in bioanalysis as we've seen it, and any thoughts on acceptable, unacceptable uses? Yeah.
So yeah, I can start with this one. In terms of have we seen it, the simple answer is not yet. We are expecting, if you imagine, you can automate certain aspects of whether that's bioanalysis, the run validations, etc., interpretations and integrations. But in terms of what is acceptable and unacceptable use, is we're still very much at the stage of treating it like any other computer system, and the applications that you would see there from a GCP point of view apply here. It's worthwhile mentioning that artificial intelligence is being used broadly as a catch-all term for machine learning, generative AI. So the functionality is key to what it's doing for you. And with anything else computer systems on inspection, we want you to demonstrate that it's fit for intended use, and you can demonstrate that through documentation.
Excellent. Well, I think that is a nice way to wrap up all of the questions that we've received. I want to appreciate and thank all of the panelists again. Thank you for your time and your presentations, and please join me in a little welcome for them. Thank you. Next up, we are going to move on to our BABE clinical session. I think they will be joining us on the stage, and then we will move on. And Jason Wakeland-Smith will be the moderator for the next session. Thank you very much.
So yes, I'm back again, but luckily this time around I don't have to give a presentation. So it's my great pleasure to introduce a couple of colleagues. You'll notice there's only one on the stage. I haven't lost somebody. Emma's joining us via a pre-recorded presentation, but I'll come back to that in a little bit. But just to introduce my colleagues then. So Doug Pham is the Associate Director for Clinical Policy in the Office of Study Integrity and Surveillance in the FDA's Center for Drug Evaluation and Research. He received a pharmacy degree from Oregon State University and his law degree from the University of Oregon. Doug recently joined OSIS after 10 years in CEDA's Office of Compliance and is currently focused on developing and improving the clinical BABE inspection program. The topic today is about the clinical study conduct side of things, so that's the focus for this session. In terms of Emma, I'll introduce her now. So Emma Whale is a Senior GCP and GLP Inspector for the MHRA in the UK. In her role as a senior inspector, Emma conducts inspections of all sorts of things, both within the GCP team, but also within the laboratories team. So that includes both commercial and non-commercial organisations in the UK and overseas inspections for bioequivalence and biosimilars. And at that point, I'd like to introduce Doug to the stage.
All right. Thank you, Jason, for the introduction. Good morning, everybody. It's an absolute honor and privilege to be here with you. As mentioned, my name is Doug Pham. Currently serve as the Associate Director for Clinical Policy within OSIS. After a number of years on the enforcement side of things, my current role allows me to kind of take a step back and really look at how we can improve the clinical trial conduct within the BE/BA space. More specifically, we want to be able to see if we can do it more efficiently, do it better, while also adhering to the kind of the pillars of what we want to do, especially in ensuring quality data, and then as well as protecting the human subjects that participate in these trials. With my brief time today, I just want to cover some of the basics about GCP principles, especially focused on documentation as well as informed consents, why they're important, and some of the concerning things that we've been seeing most recently. I know that in the past 2 days, we've been talking about the future of clinical trials, and what kind of things we can do to innovate. But I'm going to be that guy on the last day of the conference to kind of pump the brakes a little bit and say, we're going to get back to basics and see if we can do things better today. Okay, so keep in mind that today's conversation is not about who did what, or, oh man, I hope that wasn't me. It's just more of a, let's start a conversation and see if we can improve all of this together. This one. Okay. Okay. Disclaimer slide. I know that some of my colleagues have kind of passed through this, but with my legal background, I'm going to pause longer, especially with my inevitable oops sometimes, right? So, okay. So for today, we're just going to talk about a little bit of overview of FDA's BIMO program, talk a bit about source documentation, subject eligibility, with some examples and trends that we've been seeing. And finally, hope to cover some information about basic informed consent content requirements, as well as some examples and trends that we've been seeing. Okay, I know that we've already gone through this slide, but just briefly, OSIS sits within CDER under the umbrella of Office of Translational Science. We're made up of 2 divisions. And we are heavily interconnected with all the other offices within CDER, and we align very closely with Office New Drugs, Office Generic Drugs, Office Compliance, and we also work really closely with our counterparts in the Office of Regulatory Affairs. Okay, from a very simplistic perspective, OSIS performs 4 basic functions. The first, we select sites to inspect. The second, we perform the inspections themselves. Third, we report on those inspections via an established inspection report and EIR reviews. And lastly, we support CDER by providing recommendations on data reliability as well as compliance evaluations. To note, when I talk about the clinical side of things within OSIS, we tend to veer more toward both site selection and then doing the support of CDER with our recommendations, partly because ORA does all of our inspections and RRAs. Okay, for FDA's BIMO program is a comprehensive program. I'm sure that you're all aware of it, right? It includes inspections, data audits, and it's designed to monitor all aspects of FDA-regulated research. The BIMO programs are essentially there to hit 2 primary things. First, to assure the quality of the data that's submitted in any application. And second, to protect the human rights, safety, and welfare of those subjects or participants that participate in the clinical trials. There are current 11 BIMO programs. They span from IRBs to clinical investigators to sponsors, in addition to in vivo BA/BE studies. For today, we're just going to focus on the in vivo BA/BE, specifically Compliance Program 7348.003. Please keep in mind that these CPs or compliance programs are essentially instructions to FDA personnel and compliance activities, but for everybody else, it's a great source of information about what we're looking at, what we want to expect to see, and it's really helpful for your clinical sites to kind of take a look at and make sure you're kind of in line with these compliance programs. Okay, before we take a deep dive in the CP, let's talk about why documentation is so important. Much like all our previous speakers and our colleagues, documentation is kind of the cornerstone of how we reconstruct data or studies, how we assess study conduct, and how we determine data integrity as part of data acceptability in any pending application. Without kind of overstating things, documentation is absolutely vital, right? In compliance program 7348.003, there's a really good discussion on informed consent as well as study source records. And specific to the studies or subject records, a clinical site should have documentation that demonstrates that a subject met eligibility criteria. A clinical site should have documentation that accurately or demonstrates accurate and timely reporting of adverse events as well as serious adverse events. And a clinical site should document or have documentation that enrolled subjects are alive, real, and actively participating. One thing about subject eligibility, it's really important to have source documents to show that the subject has met all inclusion criteria. Without records to show that each criteria was met, the assumption is that they haven't been met. For each type of records, here we're looking at record organization, condition, accessibility, and completeness, your typical ALCOA standards. Okay, the first example that's been kind of popping up here and there is this absolute lack of source documentation to show subject eligibility. The example here speaks to a situation where there's no records to indicate a subject had achieved a certain dose of a specific drug within the 2-week period prior to enrollment. You can insert a variety of different factors. Here are subject eligibility requirements or fact scenarios. But in the end, if you have no records to affirm eligibility, then it's really difficult to say the subject is eligible. The second example here speaks to when you have conflicts in documentation. And here the example speaks to where the inclusion criteria requires a grade 3 of a particular acne assessment, but the records indicate both a grade 1 and a grade 3. While the contrast of records doesn't fall in that doomsday scenario like our first example where no records equals no eligibility, here the contrast or discrepancy creates a lot of question, right? To kind of help assess that, we have to dig a little bit deeper, and we consider a lot of different factors. The first is whether the record discrepancy is meaningful or impactful, right? So let's assume for this particular one, the protocol requires that the grade 3 acne diagnoses has to occur for, or must be within 30 days of enrollment in the study, right? And let's say, for example, that the grade 1 record was within a week of the grade 3. So in that scenario, it's kind of meaningful, right? Because you're shifting, the records contradict that need to meet that 30-day requirement. On the flip side though, let's say the grade 1 document was 10 years ago. So at that point, that record doesn't really mean much. Another concern to consider is whether there's an interplay with the study design and the study drug itself, right? There's a greater risk of impact when the study drug is intended to treat acne, but it's less so when it's not. And lastly, something to consider is what did the clinical site do or not do as a result of the discrepancy? So some things to think about that will help to kind of determine the impact of that record discrepancy. Okay, the last example here relates to documentation is when you do have documentation, but the records themselves do not clearly link to a specific subject. It's been described in a variety of different ways, and it typically comes down to where you have source records that are either unreadable, or the source records themselves don't have any link or attribution to a specific subject. While there's lots of factors, different types of explanations, from our perspective, you're kind of left with a lot of questions, right? On the one hand, you do have records, but on the other hand, without that link or attribution to specific subjects, it means you kind of don't have records, or it's not meaningful in that way. Okay, let's switch gears a little bit and let's talk about informed consents. Within the CP 7348.003, there is tons of information on informed consents, and it speaks to a couple of different things, but it includes kind of the timing of when informed consents must be signed, who can sign, and the contents within the ICF form itself, with special reference to ICHE 6 as well as 21 CFR Part 50. And when we talk about 21 CFR Part 50, 5025 discusses some of the basic tenets about what should be included within the informed consent forms itself, right? Amongst kind of a laundry list of things, it should include a statement that you're participating in a clinical trial, the purpose of the study itself, and the expectations while you're in the study. As you'll see with some of the examples, there's a bit of a concerning trend of late where the informed consents used in BA, BE studies are kind of wonky, they're weird, they're a little confusing, right? And these odd statements in the ICF itself creates a lot of confusion as to what the subject is enrolling into, as well as the purposes of the study itself. So let's take a look. Okay, the first example speaks to a trend that we've been seeing a lot in ICFs. The typical language used goes something like this. The study is a clinical research project, but no part of it is of experimental nature. So that's kind of my first reaction too. When you first read it, you're like, it's kind of weird, right? Read it a couple of times and they're like, you know what, that's downright contradictory. But despite kind of that, all that, there's curious rationales for this that we've been getting. That the statement in these ICFs, they range that this is a template we're using, or this is a BE study, so the drug's approved, so it makes sense in a way. But regardless, though, a statement like that in your ICF is kind of confusing. And if you're confused like I am, then we kind of start down that rabbit hole of like, is it even appropriate to be in the ICF? Much like the first, the second example creates a lack of clarity as to the risks associated with the enrollment in the study. The language we've been seeing includes something like this. The drug has been on the market for quite some years, and all aspects of its usage are well studied. There are no unforeseeable risks associated with it. Okay, well, I can understand the statements because, it's a BE study, it may be an approved product that you're looking at, but the statements in itself, in an ICF, creates a lot of confusion and may be interpreted as downplaying the risks. Okay, so seems like we've hit the end of the road. That was a really brief chat on the basics of, source documentation, subject eligibility, as well as informed consents and the contents within it. What I want to highlight, or at least remind folks, is that yes, we want to look toward the future and innovate and do better in our clinical trials, but we do want to make sure that we do the basic things correctly and well. To close, because my house is full boys, and we kind of skip the Disney movies, I'll end with a quote from Big Daddy in the Adam Sandler movie from Sonny Koufax. He says, "Having a kid is great as long as his eyes are closed and he's not moving or speaking." So in that vein, take care of your house. Do all the things you're supposed to do. Thank you.
So the next part of this is to introduce Emma's recorded presentation. So don't worry, Doug and I are going to leave the stage and you're going to see us walk towards the back. We will come back, don't worry, we'll be back after Emma's presentation along with some special guests. But more of that in a minute. Okay, thank you.
Thank you, and good morning, good afternoon, everyone. Thank you for the opportunity to present to you during this event, and apologies that I can't be there in person. I want to use this time to talk through some common issues, inspection findings and case studies identified during recent MHRA inspections of the clinical portion of bioequivalence studies. Michael has already shown this slide earlier today, but it summarises nicely what we've done in terms of bioequivalence inspections since 2019 and the different approaches that we're able to use for our inspections. I wanted to include this here to reiterate that these different inspection approaches apply to both the clinical and the analytical elements of our inspections. We haven't yet had an example of an inspection where the clinical part has been done remotely and the analytical part has been performed on site, or the other way around, but we would be able to adopt this approach if it was deemed to be necessary for a particular inspection or a particular study. This next slide has also been presented earlier today and shows you some additional detail relating to the critical and major findings identified during our inspections. Where Michael spoke to you earlier today about the study activities highlighted in blue on this slide, I'm going to be talking through those highlighted in orange. So the critical findings relating to the clinic, only one of the 4 on the slide relating to subject eligibility, and the major findings presented on the right-hand side of the slide in the orange box. The findings that aren't in a box haven't been forgotten, they relate more to general supporting systems. This next summary slide shows a very colourful breakdown of the other findings identified during our inspections. I don't want to go into too much detail here, but the diagram represents the other finding categories that have been identified on 5 or more occasions during our inspections. And I just wanted to acknowledge that there's a pretty even distribution between the different areas. It isn't that we have one particular problem area. And the list on the right-hand side of the slide shows the other finding categories that have been identified but less frequently. Okay, moving on now, I want to use the rest of my time in this presentation to talk through some of the areas in the clinic where we're still seeing recurring issues and where significant findings are still quite common, just to raise awareness of the types of things that we're encountering. Hopefully, if these are things that you're familiar with in your organisation, you may be able to put measures in place to avoid some of these findings in the future, or to avoid some of the business and compliance challenges that these situations can present. So these are the main areas that I'll be covering. Let's move on to the first one. Often during our inspections, we encounter clinical pathology labs that are external to the company that we're inspecting. In these situations, we look at the vendor assessment process and the written agreements that are in place that describe the services to be provided, the division of responsibilities and so on. And we often find issues here. Either the contracted lab failed to deliver the services in the contract, or the contract disagrees with the clinical protocol or design for particular studies in the nature of the sample analysis to be performed. We've seen situations where a bioequivalence facility has contracts with multiple labs as a backup plan, which makes sense from a business continuity point of view. But where we've seen issues with this is where multiple labs are used with within the same study, and their approach to sample analysis and the associated reference ranges for lab testing are different. So a result from one subject tested at one laboratory may be acceptable, but if the same sample had been sent to a second laboratory with a different set of reference ranges, the result may have been out of range. In these situations, it makes it very difficult to interpret and to compare the results from subject samples side by side, where they've been assessed using different reference ranges in different laboratories. We still see companies that use an external contract pathology lab with a set of reference ranges, but the Bioequivalence Centre also have a set of reference ranges as an appendix to one of their in-house SOPs. Often these sets of ranges are not the same, which presents issues when there are results that are acceptable according to one, but not acceptable according to another, and it's not clear how these results should be handled. We've also seen stability times applied to samples prior to analysis that haven't been met. So urine samples sent to an external lab and tested outside the established stability period, with the results being used to determine subject eligibility. The issue here isn't just with the potential impact on subject recruitment, but also the relationship between the external lab and the Bioequivalence Centre, as the lab would have been aware of the stability limitations of their own assay and the sample collection times from the clinic, but there's no communication with the clinic or a mechanism put in place to ensure that samples are received with sufficient time to ensure accurate analysis. Also relating to clinical pathology labs, we're still seeing issues with assessment of clinical significance of results. Either these assessments haven't been done for out-of-range results, or they're done for laboratory tests where the protocol doesn't permit any assessment or interpretation. So there's a hard limit on what the results need to be, either between two values or less than a certain limit. But the results are assessed as not clinically significant and the subject is recruited regardless. We also see laboratory testing for parameters that aren't detailed in the trial protocol. Often this is driven by the pathology lab, where they run a panel of tests regardless of what the sample is for. And in this situation, we would look at communication between the bioequivalence centre and the lab to ensure that protocol compliance issues are actively avoided. This can also present a subject welfare issue, as you're potentially testing subject samples for parameters that they've not agreed to via the subject consent process, and how you handle any results that come back indicating there may be something wrong when you didn't have consent to run those tests in the first place, particularly if it's something sensitive. I've also put eligibility issues on the slide, but I think I've covered those already. So moving on. The next area I wanted to talk about is IMP management. We've seen an increase in the number of findings relating to certificates of analysis for products being tested in bioequivalence studies. We still see where there's no expiry date available for the test product, either at the time of dosing, and I think once we saw where this was listed as not applicable in the final report for a study, which presents a significant subject safety concern and potentially ethical issues where subsequent stability is not established. I have seen on a number of occasions where the results listed on the certificate of analysis for product testing versus predefined specifications have indicated out-of-range results, or where testing hasn't been done, but these gaps and discrepancies haven't been questioned or challenged prior to study dosing. We've also seen where provisional data has been included in certificates of analysis, essentially draft data that is subject to change, but this has also not been challenged or addressed prior to dosing. I've seen issues with product verification checks, where these have identified discrepancies between what the product is versus what it should be, but these discrepancies haven't been adequately resolved prior to dosing. I've had many discussions on inspections about the difference between white, off-white, pale yellow tablets and the potential impact of these discrepancies and how to handle them. I think the advice would be, if you are in any doubt that what you've been provided with is not what you're expecting to receive, then do not go ahead with dosing. And then my final 2 points on IMP management relate to data loggers. Where these are included in shipments of products to be tested in bioequivalence studies. You need to ensure that you have access to the data that they hold to assure yourselves that the products have been shipped appropriately and that they're suitable for use in the study. Often the software needed to retrieve the data isn't available at the bioequivalence centre, and the data loggers are sent back to the delivery centre, never to be seen again. And this can present a significant problem where the product has been transported globally over a long duration with specific storage requirements. Also, where you're provided with the downloaded data, you need to be able to link this directly to the shipment that it related to. Being able to approximate that the date range looks about right isn't really adequate for a temperature-sensitive, unlicensed medicine that's about to be administered to humans. And finally, we've seen issues recently for facilities that may have well-established systems and processes for studies involving oral tablet dosing, but when they move into new study types for different IMP formulations, they encounter issues specifically relating to IMP, such as validity of supporting paperwork, verification checks, and accountability, and so on. In these situations, we'd advise that you prospectively look at your existing processes and pull them apart to understand what purpose they serve and how this can be replicated for the new study type, and introduce new steps, as it's highly unlikely your existing ways of working can be directly applied to a new study type without any modifications. Moving on now to talk about issues we've seen relating to computerized systems. These are being introduced more and more in bioequivalence studies, and we welcome this approach where it streamlines things and makes things easier, but they do come with their own set of challenges. The main areas we encounter findings are electronic case report forms and volunteer recruitment databases, so we'll look at these two areas in turn. Starting with ECRFs, we see all different kinds of systems being used, from those developed entirely in-house to those bought off the shelf from a vendor, and the documentation supporting their development, testing and release for each bioequivalence study varies hugely. Some of the more common issues we see relate to audit trail functionality in these systems. Often the data capture side of things is pretty good. It will let you enter the data you need, otherwise you wouldn't have chose to use it. But the ability to see who did what in the system, when they did it, and the ability to track any changes in the data is often lacking. Many systems have little or no audit trail functionality, and comments in the audit trail such as data update or record modified are not particularly useful and wouldn't help your case if that was all that was available to defend a data change, particularly if the changes are also not date and time stamped and can't be attributed to an individual. Essentially, when you're looking for an ECRF solution, we'd encourage you to look into the audit trail functionality in some detail. We also see systems not permitting study-specific builds or historic systems that aren't fit for purpose, where subject data is forced to fit into existing data fields that they're not designed for. These can take a lot of time to understand or interpret, and the data can easily be misrepresented. Barcoding is becoming more and more common. We've seen, we have some clinics in the UK where there's very little manual data entry in the ECRF system, and everything used in the trial has a barcode, so staff, participants, equipment, and everything gets scanned as study activities are occurring. These systems are fine, and this approach reduces the risk of manual data entry errors, but they can be abused, where the relevant barcodes are copied and stuck on the front of a study folder and scanned from there, rather than the system being used as intended. And there are also examples of ECRF study building and testing in a live environment and not a sandbox or a test environment, which can present a risk to live system data integrity where things can be edited in error. Moving on to volunteer databases, the main issues here relate to data migration from old to new systems. We've seen a lot of these recently, and often the migration's incomplete, where not all data has been migrated as intended and the gaps haven't been identified. Also, the detail in documentation relating to how the migration was managed is often lacking and doesn't provide much confidence that the migration effort was comprehensive and successful. And then finally, biometrics. We're seeing these more and more in bioequivalence centres for identifying volunteers at clinic visits using fingerprint scanners. We've had a few issues with these over the years. Often the scanners fail to identify the correct individual, or the same fingerprint scanned multiple times identifies a number of different individuals, suggesting an issue with the specificity of the scanners. This obviously raises significant concerns when this approach is used as the primary mechanism for identifying subjects on arrival at the clinic. More and more often, we're seeing clinics that have the capability for biometrics, but they're not using them, which suggests that they don't trust them. Okay, the next area to talk about is facilities and equipment. Most of the findings we've identified in this area relate to facility washrooms and alarm points. During my time as an inspector, I've been locked in many washrooms in bioequivalence centres and challenged staff to retrieve me from behind locked doors. While this is quite a light-hearted story, the point of the exercise is to demonstrate that washroom design doesn't always allow for access to trial participants behind a locked door in the event of a collapse. In response to this, we've also performed inspections to the soundtrack of electric saws, drilling and hammering as facility upgrades are being made, demonstrating that often these facility issues are easy to fix and commonly are resolved before our inspections are completed. Regarding alarm points, we look at the location of these throughout the BE facility during our inspections, and we look at the testing schedule for them to ensure that this is done regularly and it's done successfully. With bioequivalence trials, there tends to be a feeling that the products being tested are generics with well understood safety profiles, and mechanisms for management of medical emergencies are of lesser importance, but facilities should always be prepared for these to occur, and this is often not the case. Also on this slide, I've mentioned equipment maintenance and calibration, just to highlight a few areas where we've seen issues with the frequency and effectiveness of equipment maintenance. Testing kits such as urine, drugs of abuse and pregnancy testing kits being stored at incorrect temperatures, and communication issues with automated interfaces between equipment and the ECRF where these are in use. Okay, moving on to talk about electronic archiving. We've raised many findings over the years relating to archiving electronic data, and often it isn't the data itself that's the issue, but the metadata that sits behind it. I've listed on the slide some of the systems where consideration should be given to ensuring any related metadata is also archived where there's electronic data available. By metadata, I mean any settings, parameters, supporting information that's held within the system that you need access to in order to make sense of the data itself. So settings on an ECG machine or an X-ray machine, or any supporting information that's not presented as part of the data itself, and where the data would be incomplete without it. And then the final area I want to touch on before I move on is to talk about insurance. During our inspections, we're asked to review a copy of your clinical trials insurance policy. And over the years, we've seen a number of issues with the type of cover in place, ranging from specific populations being excluded from policy cover, specific global regions being excluded, specific medicines being excluded, which could be an issue for future trials where you may not know what products you'd be asked to work on, and specific types of studies being excluded. Also, we've seen gaps between the end of one policy and the start of the next policy where trial work has been ongoing. I haven't yet seen a situation where there's been a need for an insurance claim, where there's been a gap in cover, but I think that's been more fortuitous than anything. So I would urge you to check the terms of your clinical trials insurance, the duration of your policies, and also any exclusions in your policies that you may not be aware of when you next have the opportunity. Okay, the final area I wanted to quickly move on to is to talk about transparency in bioequivalence studies. This issue could apply to any study documentation and any activity in a study, and it really relates to how representative are the study records of what has actually occurred. We see many situations where the way things are recorded do not accurately represent what took place, and the time and resource needed to get to the bottom of things and fully understand what took place is wasteful and unnecessary. It tends to be around unusual events, investigations, interpretation of data and deviations, and I have a few case studies to go through which will help illustrate my point. But when you're involved in study activities, I want you to ask yourself, I know what I mean, but is that what it says? Do any assumptions need to be made regarding what's been written, and could this be misinterpreted? So this first example was from a recent inspection where we were told that QA were not listing all audits for certain types of studies on QA statements. The justification for this was that there were multiple audits that were relevant and the QA statements would be far too lengthy to list them all. So only a sample of audits were routinely included in the report. The issue here is that there was no qualifying comment on the QA statements to explain to the reader that there was additional audit information available. So it would appear to the reader that the listed audits were all that there was to demonstrate QA coverage for a study. Worst case scenario here is that an objective reviewer of the report, so an assessor at a receiving authority, for example, could determine that the QA coverage wasn't sufficient for the study, could ask more questions, which could lead to delays, and in worst case scenario, the data may be rejected. But if the data had been presented in an unambiguous, comprehensive manner, the situation could potentially be avoided. The second case study, paper records for a study activity indicated for several subjects ND, indicating that it hadn't been done, with some additional coding for the same procedure and the same subjects indicating subject non-compliance. On initial review, this suggested that the subjects had been non-compliant during the study, when in effect, the procedure in question hadn't been performed by study staff, and it was nothing to do with the subjects at all. So essentially, the way the coding had been used in the data was misleading and not very transparent. This next example, exclusion of a study subject for other reasons. We see this quite often, and normally we just look into subject withdrawal, potentially for safety reasons that requires maybe further investigation if we think it's safety related. So other reasons could suggest it's a safety issue. On this occasion, only 27 of the planned 28 subjects had been recruited, as the study had sufficient statistical power without the data from the final individual. But justifying this approach in the data as other didn't really explain this in sufficient detail and prompted further questions and investigation that was time consuming and could have been avoided. Then this final example, 2 subjects were shown as not dosed in IMP data, but there was dosing data and subsequent clinical data available for both of them. It turned out that they had been dosed, just not on the date that dosing was originally intended to take place. Again, the time taken to investigate the issue, to arrange meetings with relevant staff to explain the issue was wasteful, and it could have been avoided if the method of documenting the situation had been clearer. So the take-home message from this part of my presentation is really to be clear that the way your studies are documented, your investigations are performed, are truly representative of what took place, to avoid any potential for misinterpretation, confusion, misunderstanding and unnecessary delays. Okay, thank you all for your time and for listening. I will leave it there and hand back to the room. If there are any questions relating to my slides, there are people in the room who will be able to take those during the panel session. So thank you again.
To bring up my special guests, so if I could bring up Jennifer Adams, Doug and Michael McGuinness, that would be great. So we're going to play this one a little bit by ear. We have some questions which have been submitted, but equally a little bit conscious that you've only just finished listening to the last one. So we may end up with a few more as they come through, and we'll see where we go. So the first question I have is to start with Michael. So Michael, how do you decide what studies to perform inspections on?
That's trade secrets. I mean, it's fair to say it's a logical process, but it depends on the study type. So we have in the UK routine and triggered inspections. For routine inspections, we will look at the list of studies that have been submitted to us, evaluate where they are of interest. If you've previously been inspected to us, there might be specific aspects of those study conduct which we find would best show your compliance and a suitable candidate. Triggered is very different, obviously, because it's been raised as part of an application process and it's very specific and targeted. So yeah, it's not one factor. So it's not like a case of the fourth one down on the list or anything like that.
So yes, it's a double secret, trade secret. But I mean, like MHRA, it's a multitude of factors that you consider, including, the study, past history, or, if we have additional information, or there's certain things about the application that we want to take a look at or not. So there's a variety of different things that comes into play as part of that calculus. But yeah, it's double secret.
So then following on from that a little bit, so Doug, can you talk a little bit about how, or does the FDA complete BIMO inspections for all NDAs and ANDAs?
Yeah, so we do not complete all inspections of NDAs and ANDAs. As part of the risk-based assessment, we try to do the best that we can, and we use it to site select, as mentioned before, using all the factors to go into whether it's an NDA or an ANDA we're using a variety of factors to determine whether we go or not. Do we inspect in India? Yes, we do. A lot of our inspections are both NDAs and ANDAs. And there are a variety of different actors that we take a look at as well there too. So.
Thank you. Jenna, did you have anything to add on that one?
Sure, yeah. The follow-up question to that was, do we conduct inspections from sponsors located in India? So we would inspect facilities regardless of the location of the sponsor if it was deemed necessary. That said, the bulk of our bioequivalence work is located in India. It's about a third of Office of Bioresearch Monitoring's total foreign work plan is spent within India. Specifically doing clinical bioequivalence inspections. I think it's fair to say that the analytical inspections, a lot of them are located in India probably as well.
Thank you. You can leave your mic on because I'm going to give the next one to you as well. So what's the role of paper source worksheets when the eligibility of the medical records and research activities are available in the subject's electronic medical records? For the person who asked the question, for their studies, they have the subject's medical record, ECRFs, and paper source worksheet. So they're worried a little bit about duplication of data.
And I think I would agree that this seems like a lot of duplicate entry. And anytime you have a single data point that's being recorded over and over again, you increase the risk of error and having discrepant data, which then requires more effort to explain. I would just say that, if you wanted to eliminate the middleman, in this instance, the paper source worksheets, you just would want to make sure you take a careful look at your EMR and make sure that it has all of the information that you actually want to capture, that it's accurate, that it's complete, and that it has adequate controls on the system. The last thing you would want is for somebody totally uninvolved in your study to be able to delete records that you are relying on for your source. So I would just make sure that you do your due diligence when assessing your EMR, but then I don't see any added value in transcribing that data yet. Again, you're only going to introduce more error.
Thank you. Michael, did you have anything to add on that one?
Nothing, just to echo obviously when you record something more than once, then it's definitely a risk factor. But yeah, I think for the sake of time.
Excellent, thank you. So next question, I'm going to hand it to Doug first of all. Do the inspectors look at the data generated, so things like pre and post study safety assessments by the clinical path labs when you're looking at BE studies?
The short answer is yes, we do. And we'll take a look at a lot of different things, and this is part of that kind of area that we can focus on and take a look at. But it's not going to be the end all be all. We take a lot. We look at that, but then we also weigh it against other things that we're looking at within the source documents or the study records as well. So.
And then Michael, do you want to chip into that one?
Yeah, I think Emma touched on this as part. The clinical laboratory values you usually get back can form part of subject eligibility, and you've seen the example that Emma discussed. So yes, we do review that. Emma mentioned that you do have both inbuilt pathology labs within the bioequivalence centres, but also external. So the level to which we review the data might be dependent on access, but certainly they form a key part of when we're doing that eligibility assessment. It's fundamental to subject safety to make sure you don't have anybody in the trial that's at possible risk.
Thank you. And I'll stick on the theme of clinical labs. This one is certainly something we've seen on inspection. So the question is, can clinical facilities have separate reference values for subject eligibility, which are different than the clinical laboratory reference, whether that's in an investigator's SOP or the facility's own list of values or protocol appendices? So perhaps you can kick that one off.
This one arrived late, so this is more off the cuff. The short answer is it will definitely depend. You can definitely see a scenario where a number of vendors are associated with a bioequivalence site, and the onboarding process will look at the difference between the laboratory reference ranges that those labs have and whether or not they make use of their in-house facilities, the in-house SOP-defined reference ranges. So one of the things that's crucial to consider on this point is inter-study, you're looking to compare apples with apples. So if you change a vendor during it, that is different to if you're using a single vendor for study A than when you might be using a different vendor for study B. We have, with bioequivalence centres, seen a variety of approaches to clinical reference ranges where the investigator's SOP overrides the accredited laboratory, usually expanding the acceptable ranges. And again, this is something that we'll query on inspection to look at what is that? Is that to, ultimate goal is that to extend recruitment and make it easier? But equally, is there any risk to subject safety? So it very much depends on the setup. And if the question was submitted in the room, I'm more than happy to talk in greater detail about the specific aspect, but it's a very good question.
I think the other key bit to that is that you almost have to stick with one. You can't pick and choose depending on what you feel like. So if you use one person's values to get somebody in and then use another set of values to keep somebody in potentially later on. So pick and stick with one. I couldn't help myself but give myself a question. I do apologise. Sorry, gang. So the question is, for the protection of human participants in a clinical trial or a BE study, is an insurance policy compulsory to come from the sponsor? So is it fine if there's no insurance policy, but the sponsor includes a statement in the master service agreement saying they'll cover any related injuries due to the study? In essence, that's a flat no. There's no self-insurance permitted. Doesn't matter who you are or what you have. That doesn't fly. You'll always need an insurance policy to cover your clinical trial. And you also need to make sure that there are no exclusions for each protocol that you run underneath it. Certainly, we've matched them up over the years and found trials which the insurance policy says you either can't be doing trials of this medication or in a particular group of participants. And lo and behold, that's exactly the kind of trials that we've found. The other thing is just to look for dates and that they actually cover the activities and the sites and things that you have listed. So insurance is always an interesting one, not particularly exciting to look through, but there's often plenty of things to find within it. The next question is for Doug. What's expected in terms of proof within source documents of not meeting the exclusion criteria? And in particular studies which don't have chronic diseases, past medical records, those kind of things.
Yeah, so most of the conversation that I had before was talking about inclusion, so where you have to have something before you can come in. When we're talking about exclusion criteria, it's a little bit different perspective. It's what things would take you out of the pool of becoming part of the trial. We would look at the documentation as you screen, right? As what kind of questions are you looking at, or what kind of things are you talking to the subjects around, get a medical history, especially if it's like, let's say an easy one would be an exclusion because you're a diabetic, right? You would expect the CI or the clinical site to pull a history at least to kind of take a look and then start going through the interview and doing the checkoffs to make sure that they are not a diabetic prior to enrollment. So the exclusion side is not as clear cut as having to show something, but it's also part process and part of your self-created documentation.
Thank you. Michael, did you want to add anything to that one?
No, it's just to say that it's very difficult to prove a negative. Within the clinical documentation, we do see, for example, ticks to say that check of exclusion hasn't been met. Particularly for bioequivalence centres, we do see it, but again, the level of documentation that was in the background, it's something that we would check.
Thank you. The next one's to all of you, and it's sort of a link across all types of trials, really. So would you all agree that most observations in BE inspections are similar to investigational clinical trials? I'll give it to you, Jenna, seeing as you've got that puzzled look on your face.
I mean, the objective of the program is the same: human subject protection and data integrity. So I would say that, because those are the focuses, regardless of whether we're doing a new molecular entity study versus a bioequivalence study, we're going to find things in those same two buckets regardless. I don't know if I'll throw it to Doug.
So I would agree. I think those are the primary things, to ensuring the quality of the data that's generated at the site versus, or on the same vein, is protecting those subjects that are enrolled in that site. How you get about that will primarily depend on the functions of the site, the design of the study, and the protocol itself. And those details are going to be kind of driven by that aspect of it. But the overall approach, depending, regardless of a BE study or any other type of study, it's going to be very similar.
Michael?
Yep, so fully agree with my FDA colleagues. It's interesting because we have the same objective. As we said, the inspector's aim is there to find anything that detracts from data validity or anything where the welfare of the subject has been put at risk. It's fair to say, though, to expand that across the GXPs, I have the advantage of being in a multi-GXP team, and we share everything between us, the observations, people's, like we say, whether it's conducted in Glasgow or here in Maryland, people have similar traits, and it's taking those learnings and applying it across the board. I wouldn't restrict it to say the presentation topics we've heard today don't apply to you because you don't do generic applications, but you never stop learning.
I'll chip in a little bit as well. I think BE studies are a little bit different in some respects because they're so compact and they're so intrinsically linked. You have your participants, you dose them, you take some samples, you sit on those samples for a little bit while you finish the rest of them, and then you analyse them all. The trials in the wider world with the patient populations take years to get to that point, and they're huge generally, whereas bioequivalence trials can be 40, 50, 60, 70 participants. So they're very different in terms of a speed kind of thing and that intrinsic link, and both parts are often done by the same organisation. So they do pose questions sometimes around access to data, the ability to look at things in different ways. Unlike some of the monster normal, in inverted commas, clinical trials that we see where there's thousands of vendors involved, multiple sites, multiple investigators, all these kind of things. So there are different things to think about as well. On top of, is the data good? Are the participants safe? Can we trust the data? So there are nuances to all of these things. Again, I'll throw the next one open to the group. Can you tell me a little bit about sort of collaboration for sharing observations across GXPs? And I'll widen the question and say about, is this being about collaboration between us as organisations? So can you talk to me, first of all, about collaboration within your organisation, and then secondly, about collaboration between us as a group?
I can kick us off and then happy for. So yeah, in terms of, if I've got this right, collaboration within our group. So as I said, we share everything that we talk about. We have team meetings. We try to ensure consistency between inspectors, so it doesn't matter whether you get myself, Jason, Emma turning up, that you get a similar approach. So collaboration through training, discussions, professional development is absolutely there. All of our reports are peer reviewed by a colleague that was independent of the inspection itself. So in terms of that aspect, it's something that we encourage. It's well supported by management. In terms of collaboration across the different agencies, if I've not skipped Jason's question, I think it's very true that you've heard almost for the last 3 days, the word collaboration has been the word of the workshop. We work very closely with our partners here in North America, both FDA and Health Canada, and it's fair to say that information is invaluable. Lessons learned, lessons shared. Again, we talked about the similarities of GCP inspections, the specific aspects of BE, and we hold quarterly meetings between ourselves. I'm very familiar with colleagues here at FDA, but also at Health Canada, and that information sharing is an excellent source. And I think if I open it up at that point.
Yep.
So on the FDA side, we are not the same group. We have the disadvantage of being a separate program. FDA aligned our programs, I think, in 2019. So our pharma folks are actually in a separate program from our biome folks. That said, when you conduct an inspection, part of your preparation for the inspection is looking at the inspectional history, and the database of all of our inspectional findings is shared across all programs. And so we have access on a BIMO inspection if I'm inspecting a large bioequivalence facility that is also inspected under GMP. I have access to the full report, all of the observations, the names of the investigators who conduct it, and would have access to them and all of the data that they discovered on their GMP inspection to help inform my upcoming BIMO inspection.
I mean, not much to add, but it's just in the case of FDA, the volume is kind of an ordeal, right? And so we continue to work collaborate with all the various offices, with ORA, within CEDAR, but it's just a huge amount of information and lots of inspections, lots of different types of inspections, but they do get pooled, and it is part of the calculus every time we site select, or when we go out to inspect, or when we do a review post-inspection. We're always looking at the totality of information. So that's a continuing thing, to continue the collaborations across agencies. That's something we do all the time, and it's super fun. I think it's great, and it's a great way to share stories, share information, and like you mentioned, kind of learning moments about what you've seen here, certain types of inspections, or different kind of things. So it's a valuable thing that I think has been going on for a bit of time, and it's something that I think everybody here is looking forward to continuing for the long term.
Thank you very much. I'm going to keep the next one because the answer is a bit of a negative, but the question was, is informed consent required for data taken from a biorepository? And we've taken that as sort of registry data where access to the patient may not be possible. So you don't know when the patient may next be coming in for a checkup, the patient's moved and didn't tell us, these kind of questions. So we've taken that as a BE kind of question, that you've had a volunteer for a while, and perhaps they haven't attended the last visit. And we've taken it as, could you then use registry data to potentially follow them up if they just didn't come back? The answer in some respects is we have no experience of that, of using registry data in bioequivalence trials, really. We'd want that clearly defined if ever you were going to do that. And it seems to be a fairly blunt instrument of, are they still alive? Have they attended a hospital? And quite when you might be able to access that data, and even whether or not you have consent to do that. But just, I guess, be careful, but something to explore potentially in the future. I'm just going to give my poor panel one more question, and then I'm going to release you. The question's suitably broad, so by all means, feel free to go with it. The question is, can you please define oversight, study, and reporting? And so, Doug, I'll start with you.
Okay. So, oversight of study and reporting is, as you can imagine, huge, because it's, you're looking at different actors as part of a study. You're talking about sponsor, you're talking about clinical sites, you're talking about clinical investigators, SSPs, CROs, that are all in between. But what I would reiterate is that at every point or any touches within the study, you should have some overall oversight with what you're doing. And if you're the sponsor, you should have oversight in general of the entire study that's being performed. Those kind of controls should be built in a sense. It'll help prevent a lot of problems. It'll help, if there are problems, you've already pre-planned for them in how you approach them and deal with them. But the answer to this is, it kind of depends, right? Depends who you are and what you have to do. And then the reporting aspect of it will be related to that too. So it's a long-winded answer for me. Michael?
Yeah, I can go next. So as Doug said, it's a huge, huge piece. It's in terms of a bioequivalence study, the oversight, one of the key pieces is, like we talked about, those touch points. It's not, we've got screening, we've got then getting them in, getting subjects or trial participants into the clinic. Linking them up with something like a lab where you might have a light-sensitive molecule. Not having that two-way conversation, it's the same as pretty much any aspect of business. Communication is key. Oversight and control is a different aspect. Demonstrating it works differently in almost every company I've worked in or I've inspected. Some go with what I would call very militant, restrictive. We will meet every day at 9 o'clock. We'll have a toolbox meeting. We will go through this. Some don't. And there are strengths and weaknesses to both parts. The communication element is key, having information. The one thing is just to acknowledge the negative part of that. If you're a sponsor and you have outsourced everything to a BE centre, be aware of the role in which you as the sponsor are exerting your pressure and control. You looked during my bioanalytical presentation, if you'll allow me to jump into it, we saw the negative aspect of pressure on individuals where they pushed, and then we ended up with a data integrity breach, which then resulted in the trial ultimately going down. So what might start off as positive oversight, positive control, can very quickly be turned into a negative, and that's a very slippery slope to try and climb back up from.
Jenna, did you want to add anything?
I mean, I guess I will highlight that this is a great question. We don't spend a lot of time talking about oversight in bioequivalence compared to the first 2 days of this course. But ultimately, the oversight of the study at all of the levels that Doug has discussed, the oversight by the clinical investigator, oversight by the sponsor, oversight by the CRO, all of these different levels of oversight have a direct influence on the integrity of the data and the protection of the human subjects that come out of the study. And so as a result, it's just as important in bioequivalence as it is in a normal non-BE clinical trial.
And I'd like to link it back to Emma's presentation in some respects, picking up the reporting side of things. So it's important that the report bears resemblance to what actually happened on the trial. We've seen plenty of examples in the past where there are an errant cut and paste job with particular issues to this kind of thing, or you find things within the trial data itself which just don't appear in the clinical study report. And even when you're talking to people about it, they just don't necessarily appear. What's really important, and we talked about risk earlier on in the workshop, if things happen, if you tell us, we can think about it and give a considered opinion and try and link it up to how it all fits together and how it works. If you don't tell us and we find it, we've not really got too many options left to us. It might be possible to extend the inspection, but certainly if we're overseas, that's limited. Or the next one is to go back to the assessors and start to say, we have problems here. We found this stuff, it's not in the clinical study report. Over to you, what do you want to do with it? So be open, be honest, discuss, comment on what you found, talk about what you did when you found it and what you then put in place to try and deal with it. And then we can have an active conversation about what that is. That's really helpful to have it up front so we can prepare for it, talk to the assessors potentially if it's in the CSR. If you've omitted to put it in an error and it comes up in inspection, then again, talk to us early. Let us know that there's something we need to work our way through. So I'm going to draw it to a close there. Thank you very much, panel. Thank you very much for coming to join me on stage. If I could ask you to put your hands together and thank the attendees. My final job is the important one, and that's to let you all go for lunch. We're back at 1 o'clock for lunch, so that gives you a bonus 12 minutes to grab a little bit of lunch. So thank you very much. We'll see you for pharmacovigilance this afternoon.
Welcome back from lunch. Hope you had an enjoyable and delicious lunch. Just a few housekeeping reminders. As a matter of fact, one in particular. Remember, the survey and the certificate of attendance are available online until February the 29th. February the 29th, so we don't want you to miss out on that opportunity to download your certificate of attendance immediately after you fill out the survey. Otherwise, that opportunity may be lost. It's my pleasure to introduce for the Pharmacovigilance Compliance Keynote, Stephen Venter, Head of Compliance Team 1, MHRA. Please join me to welcome Stephen Venter.
Oh, good afternoon to everyone in the room, and hello, whatever time zone you may be online at the moment. And really thank you very much to the organisers to give me the opportunity to introduce this session. And I think it's a really great thing now to see the combination across the GXPs, if you like, within this sector of combining it into a single event. So I just thought I'd just cast our minds back a few years now. I'd say I was stood on this very stage, but the venue was different at the time. But I remember back in 2018, myself and colleagues visiting the US to really kick off this collaboration that we now have around symposia and meetings. And the meeting that we held all those years ago before disruption of pandemics, where we really focused on clinical trials and data integrity. But even now, just looking back and the experience of working with our inspectors and in the pharmacovigilance area of just how relevant that data integrity discussion was then and still is now, as PV really does really get involved in that level of data and just the importance of the compliance and reliability just to help inform decisions. So even then, you know, I think we were really touching on these key subjects and these key items for an ever-evolving world of regulation and conduct of all these different activities related towards medicines. So I just thought before we get into what's going to happen this afternoon, I'd just reflect what's been happening over the last few days. And I think these themes are relevant for whichever area that we're looking at and discussing. That, you know, we've talked a great deal already about collaboration, and not only the collaboration between us as regulators, but also that collaboration between us, industry, you, the stakeholders. And also, I think it's been really refreshing that we've heard a lot of talk about patients as we're having these discussions, and how that's the important thing at the end of all of everything that we do. So that whole collaboration theme has really come through, and also how we can improve and get better and working together as a community around the delivery of safe medicines. And of course, all of that is really built on information sharing. So you'll hear about how our agencies work together to be more effective and more efficient, and also how important information is, especially for when we're looking at safety. And I think, again, we'll just touch on how pharmacovigilance really does, it's an information game in effect. And when you start combining all these different themes, it really just shows how all of these three areas are so important. And then this week, we've already looked at the future and new approaches, and pharmacovigilance and compliance is no different in that there have been changes, and there will continue to be changes to how we work, how technology influences how we look at things, how systems and inspection approaches have evolved, and how that will continue to do so. So even though we're now on day 3 and the PV session, I think it just shows how all of these topics intertwine themselves across all of the regulated areas that we look at. So even though we're in a new session, it's really an extension of what we've been talking about all through the week. So I think when I was just pulling together, trying to work out what to say here, and just discussions with some of the team back in the UK, I think this collaboration theme really does underpin what we're talking about with pharmacovigilance, in that it is in effect the ultimate collaboration, in that we have the patients and the healthcare providers, we have the pharma company, the organisations, then us, the regulator. And actually it doesn't work without collaboration, because we're all reliant on this transfer of information. You know, it's a cycle of trying to find the signals, the events, and the organisations and adapting, and the regulator having the oversight. And of course, importantly, the patient plays such a key part here of actually informing and starting that information share, and then obviously being the recipient of advice and changes to guidance to patient information. So without this collaboration, the system doesn't work. And I think it's very easy for someone like myself that spent their regulatory career very much looking at clinical and preclinical and building up to the all-important approval, that it's quite easy for people like myself to forget the importance of where pharmacovigilance steps in, of expanding that data set about safety, of the ability to have a larger population to get the data and the information to inform the safety decisions. Because certainly as working in the development phase, we've got a limited population of where that data comes from. So the power of this system is so important to patient safety. It's really important that we make sure that the systems work, that they're compliant, and that there's good quality data to inform those decision makings. So again, reflecting back to my presentation earlier this week, you know, that the whole decision-making process of having the right information, the capability to make these safety decisions, a pharmacovigilance system is critical to enable us to do that. So I think that really does show the power of communication and collaboration. And without that, these things don't work. And I think the other thing that really comes home as a powerful message here as well, this really is a truly global activity, in that we're not just talking about the FDA is only concerned with data from the US, the MHRA only concerned with data from the UK, Health Canada only from Canada. It really is about sourcing all this information as widely as possible to inform those decisions. And this global activity as well it's not purely our responsibility to have the right systems and regulation and oversight in place. And we're increasingly seeing now that this really is a global regulatory activity, and the opportunities there for us as international regulators to partner with more and more regulators from around the world to make sure we're all have got reliable data and harmonise as much as we can to make sure those systems are in place to really, really build good quality informed decision making. So what we're going to see today, I think this is a great start to this event for pharmacovigilance and coming into this family of events this year. So again, we're going to hear about international collaboration. So we'll hear how our agencies are working together to really maximise all of our experience and resources. As with the rest of the week, we'll look to the future and learnings that we've had and the direction of travel for how these inspections and the oversight will be put into place as we move forward and work together. And then as usual, there will be updates. Part of these events are to enable us to inform you and to the widest audience possible to get our current thinking across and point you in the right direction. But equally, hopefully these presentations will prompt questions, whether it be online or in the room, that can feed back to us now. And then the panels will be able to talk things through, offer advice, or point you in the right direction. And it all builds into this collaborative model and working together to make sure that we all can work with good systems, good data, and work together to the ultimate goal of ensuring that there's safe medicines and that patients are safe. So my last message really, before I hand over to the teams, is please enjoy the rest of this event. I'm sure it's going to prompt some really good discussions, ideas, and we can really share the expertise that we're going to have up on the stage for the rest of the afternoon. So that's me done. So hopefully this set up the day for everyone. And now I'd like to hand over to Carolyn, who will moderate the first session this afternoon. So thank you very much.
Thank you, Stephen. So as he said, I'm Carolyn Volpe, and I'm the team leader for the Pharmacovigilance compliance team at the FDA. And I'm really excited to be moderating the final afternoon of the symposium. It's been great. Now, like Stephen said, we're going to focus on pharmacovigilance in the post-marketing setting. And, I think that this is the point in the drug's life cycle where the drug is now being used in the real world. This is being used by patients of all age groups, races, with concomitant medications, and even used off-label. And this is a critical time when we become aware of the safety issues not seen in the clinical trials. And to make sure that we're all aware of the safety issues as they emerge, the next presenters will discuss how regulatory agencies across the globe are collaborating. First, we have Claire Longman, an expert pharmacovigilance inspector from MHRA. She will discuss several projects focusing on international collaboration and the goals of these projects. Next, we will have Sherry Boyce. She's a director of the Division of Enforcement and Post-Marketing Safety at FDA, and she's pre-recorded her updates to the PICS Good Pharmacovigilance Practice Expert Circle. And finally, we'll have Paul Bellegeron, the Regional Regulatory Compliance and Enforcement Specialist from Health Canada, and he's going to discuss some experiences with the GVP joint inspections. So Claire, I'll let you get started.
Thank you very much for that introduction, Carolyn, and thank you everybody for welcoming me here today. It really is a pleasure to come and speak to you all. I've realised that I have the shift just after lunch, so I'm hoping that I'll be able to keep you awake. And all those in Europe who are joining online, you'll all just be getting ready for your dinner. So hopefully we can make this talk as interesting as possible to keep everybody listening and interested. And I'm going to be talking to you about international collaboration, really from a regulator's perspective. So let's begin. So I think we can all agree that pharmacovigilance is a global connection that we all have. And we all do this role for one real common goal, and that is to make sure that medicines that are available to patients are safe and effective as possible. Well, I hope that's the common goal that we all have in pharmacovigilance. And your systems that you have, your PV systems, they are global in nature. You are collecting that adverse event data from all around the world, and that's feeding into our regulatory systems. And that's really important for regulators to make sure that we are collecting that information on a global nature, one, because we share that common goal for medicines to be as safe as possible, but also in our respective populations to make sure that we have that ability to look at that adverse event data and make sure the benefit risk profile is as favourable as possible. And therefore it makes sense that as regulators we collaborate more. We start to discuss that information between each other. We use that to ensure patient safety, and we also use it so that we can reduce the burden on you as market authorization holders, and that you're able to do your jobs to the best effect as possible without having too much regulatory burden on you. So today I'm going to start with an overview on that international collaboration, and I'm going to start with looking at some current projects, really at a medicinal regulator viewpoint, so not just in the inspectorate. And then I'll be looking at some of the international groups that are around for regulations, and then concentrating on some international agreements. And this really I'm focusing on international agreements that we have in our compliance teams and across inspectorates. And then I'll finish with some working examples and some training initiatives. So hopefully that will be an insight into some of the up and coming steps that we take and the way we're moving. So obviously I'm talking from a MHRA perspective here. And then once I'm finished, what the intention is that Sherry will then talk about some particular aspects of how inspectors collaborate, and then Paul will give some specific examples as well. So I'm going to start off with the Access Consortium. So this is a consortium that the MHRA joined in January 2021. And it's a consortium made up of 5 regulators, so Australia, Canada, Singapore, Switzerland, and the United Kingdom. And the aim of this consortium is to share research and improve timely patient access to high quality and safe and effective medicines. And the consortium really aims to explore different ways in which information can be shared, and it has several working groups. And one of those working groups is looking at how inspection outcomes and reports can be shared and across regulators. They're currently operating under the 2021 to 2024 plan. And one of those commitments that they've made is to look at GMP inspectors, so good manufacturing inspection reports, and how those inspection outcomes can be shared. So hopefully the outcome from that will be favourable, and we can start to look at how we can move that forwards in other GXPs. The second project I wanted to talk about is Project Orbis, and I find this quite interesting. And this provides a framework for the concurrent submission of market applications across a number of regulators. And this is particularly for oncology products. So the regulators involved in this are the same 5 as from the Access Consortium, plus the US FDA who lead on this project, and also Brazil. And this coordinates the submission for review of market authorizations and extensions to licenses for cancer medicines. And the aim is to, for a market authorization to submit that one application, and then it gets submitted to all of those regulators at the same time to be reviewed. The thing about this project is, although those submissions are all put in together, it therefore reduces burden on the MAHs. They just have to submit one submission. It does still allow regulators to make sovereign decisions. And I think that's something that I want to highlight, that although when we talk about international collaboration, I think sometimes we think that we all have to make the same decision. But actually, I think Project Orbis presents an opportunity where decisions can be made in their own right, and each regulator is deciding whether they license the medicine or not. But the actual burden on the MAH is reduced by just making that one submission. And Project Orbis has been successful. I think there's around 20 medicines that have been licensed through this route currently. So just some other international collaborations that I wanted to mention that impact on the inspection teams and pharmacovigilance. So ICH, we always have representation on ICH guideline updates, whether that's through the inspection teams themselves, or we have other delegates from the agency. So sometimes our safety and surveillance colleagues will join those, and then they'll feed back any information and ask for our input in the inspectorate where required. We have input into the CIOMS working group. So at the moment we have colleagues from safety and surveillance in the MHRA who are sitting on the AI CIOMS working group for pharmacovigilance. And then PICS. Now this is the Pharmaceutical Inspectorate Cooperation Scheme. And this is something that I'm very passionate about, and Sherry's going to talk about in much more detail, and then Paul will also. So I'm not going to go into much detail, but this is a scheme that is set up specifically for inspectors. Regulators sign up to this scheme, and it's a way that we can benefit from best practice sharing. We look at hot topics and training, and look at how we can train each other as well. So it's a really good scheme. And I'm deputy chair of one of the working groups there, but I won't go into any more detail on that because Sherry and Paul will talk about that a bit more later. So ICRMA, this is the International Coalition of Medicines Regulatory Authorities. Now this is a voluntary scheme, and it's an executive level. And the aim of this is to bring regulatory authorities together to look at where there may be similar challenges across different regulatory bodies who authorise medicines, and also looking at ways to harmonise. And there's a few pharmacovigilance work streams that sit under this. One of them is looking at vaccines, one of them is looking at increased AE reporting, and the other is looking at big data. And then we have a lot of bilateral agreements. Now, they sit across the MHRA at a regulatory level, but also within the compliance teams also. So I'm going to talk a little bit more about some of those bilateral agreements that we have within the compliance teams. So we have, obviously, I'm here today talking to you at the US FDA, Health Canada and MHRA Joint Symposium. We have bilateral agreements with both the US FDA and Health Canada, and it's an absolute pleasure to be here talking with colleagues from both of those regulatory authorities. Apart from supporting each other at these sort of conferences, we also have regular meetings where we meet on a quarterly basis, and that gives us the opportunity to share intelligence that we may have received. We share our inspection plans, we share inspection reports, we talk about new initiatives which we might have, and we can benefit from training with each other as well. We also have a similar relationship between the MHRA and the Therapeutic Goods Administration in Australia, and we could again share inspection schedules and reports. And we have had Australian inspectors who have come out. Well, they've joined online remotely through some of our inspections, and we've shared best practices. And then through the Northern Ireland protocol, we have a relationship with the European Medicines Agency, and we share our inspection schedules, and we share inspection reports, and that goes both ways. So just wanted to take a minute to discuss why these interactions are so important and why these collaborations bring benefits. Obviously, we communicate significant inspection findings. That means that we can ensure patient safety. We can make sure that any intelligence that is coming through, we discuss with each other and the best ways to move forwards. And we use that intelligence within our own systems to decide who to go and inspect or when we need to take action and when we don't. But one of the other things I wanted to allude to is, but one of our aims is also to reduce the burden that is on market authorization holders. And an example of this was an inspection that the MHRA conducted, and where we found a large number of adverse events that had not been reported into the safety database, and they were residing outside of the safety database. The company had been inundated with a large number of reports in a short amount of time and were struggling to process them. So we issued a critical finding at that time, but rather than just dealing with that ourselves, we communicated that with our partners where we had the bilateral agreements, and we were able to come to a decision together about how best to go forwards with that. And rather than everybody rushing to inspect the company, we all collaborated on what kind of corrective and preventative actions we would like to see. And then that was collated together, and we were able to put one group of those to the company to reduce the burden on them to fix that. And then when it came to re-inspection, rather than all of the regulatory agencies going and conducting their own inspection, the European Medicines Agency led on that re-inspection, and us as the MHRA went as observers. And then we were able to have that assurance that remediation activities had happened without putting extra burden on having double inspection within a short time frame. So just to finish today, I just wanted to concentrate on some of the training initiatives that we have. So I said earlier at the start that pharmacovigilance is a global activity, but it's actually becoming more global. There are more and more regulators who are starting to introduce pharmacovigilance legislation, and because of that, they are starting to introduce more and more inspectors who are looking to make sure that those regulations are complied with. So at the MHRA, we think it's very important that we are involved in that training, and we have a lot of experience in inspecting pharmacovigilance, so we try to aid those trainings where we can. And we have sent our inspectors over to China and trained inspectors from the China National Medical Products Administration, and we gave extensive training on a suite of different presentations to them. More recently, we have had the Ghana Food and Drugs Authority come over to the MHRA. We have safety and surveillance colleagues who look at the adverse event reporting, who have gone over to Ghana to help them implement their systems. And then they've come over to us and we've helped to train their inspectors from everything from who we go to inspect and when, how we're deciding on that criteria, to what we look at when we're on inspection and how we report those inspections. And we are committed members at the MHRA to the African Union Smart Surveillance Programme, and this is part of the Bill and Melinda Gates Foundation. And this is training that is provided to African nations to support their pharmacovigilance safety systems. Our safety and surveillance colleagues have been part of this for a while now, and it's something that we are starting to develop within the inspectorate as well, and we are very keen to support this. So hopefully that has given you a good insight into some of the initiatives we have. So there are a number of international collaborations ongoing. We do already share a lot of information with each other on intel. We don't just keep that to ourselves. One of the aims is to reduce that burden on industry. We are trying to do that, but we do recognise that there are continued efforts required in this area, and it's something that we can keep continuing to push to see how we can collaborate a bit better, how we can use the information that we've got in newer ways, and continue those efforts. So I'm going to pass on to Sherry Booz now, who's going to talk you through the PIC scheme. So I hope you enjoy. Thank you.
Good afternoon. My name is Sherry Bose, and I'm the Director for the Division of Enforcement and Post-Marketing Safety in CDER's Office of Compliance in the U.S. Food and Drug Administration. I'm really excited to speak to you all today about the newly formed PIC's Good Pharmacovigilance Practices Expert Circle. I'll start by giving you a brief history of the PIC's GPV. We'll review the organizational structure of the Expert Circle, the goals of the Expert Circle, and finally we'll review the outputs of the Expert Circle's working groups. In 2014, the PIC's working group on GCP and GPV was formed to facilitate technical cooperation and harmonization practices. This included the information sharing amongst PIC's participating authorities through the Joint Visit Program. In 2021, there was a proposal to dissolve the GCP and GPV working groups and create two separate and distinct expert circles due to their very different technical priorities. And then finally, in 2022, the PIC's committee established the GPV Expert Circle with the purpose of facilitating training and information sharing for inspectors who specialize in GPV. This is our organizational structure. I'm currently the chair of the GPV Expert Circle, and we have representatives from 16 participating authorities, such as MHRA, Health Canada, and EMA, just to name a few. We kicked off our first Expert Circle meeting in February 2023 and established and received approval of our mandate by the PIC Secretariat in November of 2023. There are 3 working groups that fall under the umbrella of the Expert Circle, which include the AI Machine Learning Working Group, GPV Best Practices Working Group, and the Joint Visit Program. We'll discuss these working groups a little bit more in the next few slides. The overall goal of the GPV Expert Circle is to develop expertise by establishing a network of GPV experts, to train, design, develop, and execute a training program for GPV inspectors, to network, to develop a communication system between PIC's participating authorities, and finally to represent, to develop and maintain a common approach and interpretation of international guidelines. The outputs of the 3 working groups are summarized on this slide, and we'll go into each working group a little bit further, but the overall takeaway of the Expert Circle collectively is to establish a formal network of GPV inspectors across the regulatory agencies with the goals of information sharing and harmonization. The AI Machine Learning Working Group's main focus is to design, develop, and execute the Expert Circle meeting to provide training for GPV inspectors to obtain the required knowledge and competence to inspect key topics in a harmonized manner. The GPV Best Practices Working Group's goal is to design and develop documents for publication on the PICS website and PICS Inspectorate Academy. This again helps train GPV inspectors in a harmonized manner, as well as being an excellent resource for PICS participating authorities who are still developing their inspectional programs. And then finally, the Joint Visit Program. This has been a great success in the past, with 32 GCP-GPV groups formed in 2017-2018. It was on hold during the pandemic due to travel restrictions, but the triplicate groups were able to complete the programs remotely as appropriate. The JVP is a great opportunity to learn from each other and harmonize inspectional procedures and techniques across the different regulators by observing inspections in real time. There will be a new call for regulatory volunteers under the new GPV Expert Circle, with opportunity for face-to-face and or remote options due to travel restrictions some countries may still have. In summary, the new GPV Expert Circle was formed with the goals to develop expertise, train, network, and represent. Working groups are formed to establish a network of GPV inspectors across the regulatory authorities, and this will provide an opportunity to compare and harmonize inspectional procedures and techniques across the different regulators. We at FDA are honored to have been chosen to lead this very important effort on behalf of PICS, and we look forward to the success of the newly formed GPV Expert Circle. Thank you.
So, bonjour à tous. Good afternoon, everyone. I'm Paul Baillargeon. I'm the GVP inspector with Health Canada. I'd like to thank the FDA for giving me the chance to come and present Health Canada's perspective on joint GVP experiences. So today I'll be discussing about two different action items that were taken. So as mentioned by Sherry, the Joint Visits Program, the PICS Joint Visit Program, as well as Health Canada's joint inspection with the EMA during COVID-19 pandemic. We will review the lessons learned throughout and as well as summary of these projects. So the first project was the joint visits with the PICS. The objectives were quite clear, so provide training for all the inspectors, harmonization of the different inspections, as well as maintain mutual confidence. To do so, the inspections were carried out by the 3 different health authorities throughout a 24-month period, one in each participating country. We say visit because the host health authority is observing the other members during one of their regular GVP inspection. So this all started in 2018, with the first one being in December 2018, by the MHRA. The next one was in May 2019 with the FDA. And as we know, because of the COVID, the March 2021 was a virtual one, but it still happened during that time. So if we look at what was assessed at the process level, so these visits enabled inspectors to go through each of these areas that are discussed on my slide. So every inspector was able to provide his own health authority structure, regulations and guidance documents that are associated with the GVP program. Each health authority was able to provide the site and product selection process of how they choose. And as well, they were able to go through the inspection process themselves. So just the scope of the inspection, how does the inspectors have pre-inspection activities, the report issuance, CAPA plan assessment, as well as the tool used by the inspectors. A report was presented to the PICS to discuss all aspects of this project. As you may know, as we touched base upon earlier, everyone remembers the COVID-19 pandemic. Health authorities needed to get creative, leverage opportunity, and work jointly with trusted regulators in order to move forward in these periods of lockdown and travel restrictions. So this brings us to our second project, where Health Canada was able to conduct its first joint GVP inspection with the European Medicines Agency. Just to give you some background information, in May 2021, Health Canada contacted an MAH to schedule a GVP inspection just before the start of the summer session. Unfortunately, the MAH informed us that the EMA was also coming just before us. So Health Canada actively engaged the EMA to suggest a partnership for the inspection to see if resources could be joined and efficiencies could be gained for both agencies. Fortunately, there was a positive response from the EMA. Therefore, right away, we installed a pre-inspection meeting so that all inspectors involved in the inspection could provide a game plan. So what we did is we sent the notification to the MAH explaining that both EMA and Health Canada would be coming at the same time. EMA had already provided pre-inspection requests, and they covered most of Health Canada's pre-inspection requests as well. We did provide specific Canadian requests, but those were added to the originally added one. Since the inspection was already initiated by the EMA, it was decided that the EMA would lead the specific sessions and discussions. Health Canada would be present and could ask any questions as well as they wanted. Health Canada also led a couple of sessions that were for specific Canadian topics on pharmacovigilance. The MAH was to receive 2 inspection reports, so one from EMA and one from Health Canada, if any observations were made. The inspection was held virtually, so 10 days virtual. So the EMA shared their video conferencing services as well as their SharePoint access to all the inspectors and the MAH. Regarding time zone differences, all inspections started early in the morning, so around 7 a.m. Eastern Standard Time. And during those sessions, there were predetermined sessions for each of the GVP topic. And during those sessions, verbal requests were made, but also in order to provide efficiency, they were also made in a written format, in the SharePoint service, so that easily the MAH and the inspectors could go back to them and see the responses there. There were some internet issues, but they were very sporadic, so no issues there. We did have daily briefings, so in the morning we would have the discussion for any outstanding issues, and one after the inspection day where we would discuss the possible observation and discussions on the next day topics. Both health authorities provided observation report to the MAH according to their own inspection process. So what happens here for Health Canada is we have an exit meeting at the end. It's where we present the draft exit notice report. This is a possibility for the MAH to clarify any points that they see in the observation. Unfortunately, EMA was not part of that exit meeting. So once the MAH provided a response to those reports, the CAPA was reviewed by both health authorities and discussed amongst ourselves. For the MAH, it was mostly a copy-paste, so there was no really added extra work to provide responses to both of the reports, so that's time there gained for them. As well, Health Canada provided a closeout letter, which was sent when the CAPA plan was deemed acceptable. So Health Canada's lessons learned. So for both the joint visits and the joint inspection itself, we saw that the PV requirements are similar across jurisdictions, and their assessment can be divided amongst regulators. I like the divide and conquer phase. So we saw that for the three parts There, the case reception, case processing, annual summary reports, as well as review of IT systems used in PV. These were quite similar across the different regulations, so they could be combined within the same sessions. Whereas the PSMF file and QPPV portion were more of an EMA portion, but Health Canada did attend those sessions in order to familiarize ourselves with this process. The last 2 points are Canadian-specific requirements, so the unusual failure and efficacy and the notification of foreign action. Again, these were led by Health Canada, but all the different participants were there for for familiarization. So if we look again at the advantages, so as we discussed throughout the day, confidence and collaboration is increased between health authorities. Resources can be shared between health authorities, a reduction in travel costs as well for virtual inspection. It is a more efficient inspection process for the MAH. They do have only 10 days of virtual instead of having 20 days total with 2 different health authorities. We did encounter some technical constraints though. As I mentioned, the internet connection can be difficult for multiple participants from around the world. As well, document sharing application can be tricky, especially when MAHs are sharing confidential documents. So it's important for regulators to provide them with a safe space so that they can share those documents. Virtual inspections take more time though for health authorities. We have to schedule sessions via video conference for specific topics. Outside of those, it's difficult to interact with the MAH to get responses to small interrogations. As well, when you're on site, it is possible to get quick ad hoc sessions to resolve questions directly. The time zone difference can cause delays sometimes. It can start very early or very late for some of the MAHs. And what the inspectors missed the most during the virtual session was the interaction face-to-face. It's very difficult to see the body language of the person that you're interviewing. So that was missed during the inspection. So to recap, our takeaways from these, the joint visits and the inspection. So joint inspection are not only possible, but successful. Technologies as basic as video conference and SharePoint were very useful tools for inspections teams so far apart. All the MAH's PV systems were reviewed by multiple inspector eyes, inspector with different expertise. Health Canada would not have been able to cover so much in the same amount of time. Increasing collaboration and moving towards inspection reliance is also a point that's needed to be considered here. Regarding time, it does not require more time for both agencies besides the pre-inspection meetings that we had, and it will enable all to perform the inspection within the allocated period like we requested. MAH will have 2 agencies at the same time, but to go over the same PV system. SMEs are less solicited and free to perform their regular tasks. A cost savings for sure, like I mentioned, so virtual inspection can be done at home/office for the inspectors. It is important, though, that we identify leaders, like I mentioned earlier, for the different sessions. It will provide a better preparation for each of the inspectors, more time, and then we can focus on one subject instead of multiple at a time. Remote inspections process is promising by allowing remote interviews with subject and matter experts from various locations, software demonstrations, remote navigation, or review of MAH's documents. And like we mentioned, there is a possibility of a hybrid. Again, the invaluable opportunity for inspectors to share best inspection practices were a great training experience for inspectors from all agencies present. The invaluable opportunity to discuss practices and differences in regulatory frameworks, inspection processes, and compliance activities related to PV. Health Canada continued its program review by initiating the review of GVP guidance documents and increasing inspector training opportunities. As well, the last point, so globalization helps align international GV expectations. there are differences in the health authorities' PV regulations versus Health Canada's PV regulations, but still enabling joint inspections. Most companies have global activities requiring multiple input from various sites. Therefore, remote inspections may provide regulators a new way to conduct or collaborate on international inspections. So thank you very much for this presentation.
Thank you, Paul and Claire and Sherry, for that great session about international collaboration. And I'm sure we'll be talking more and working together more in the future. So now I'll invite up the session 5 presenters. We're now going to focus more on what FDA is doing now and looking towards the future. Our first presenters are Peter Dyack and Jenna Stowe, and they're going to be discussing a pilot project that our office performed last year in an effort to explore uses for remote regulatory assessments in the post-marketing safety compliance programs, PAID and REMS. And then next we are pleased to have Chrissy Cochran, the director of FDA's Office of Bioresearch Monitoring Operations, talk to us about OBIMO inspections now and some plans for the future. And then finally, we're going to have Robert Ball. He's pre-recorded a presentation on a very hot topic among regulators and industry. It's the use of artificial intelligence in pharmacovigilance. So Jenna, I'll turn it over to you.
Thank you, Carolyn. And thank you everyone for being present to hear more about how FDA is focusing on innovations and continuously looking for opportunities to improve and streamline processes as we prioritize the modernization of our operations. So regarding the future of inspections, I will be further discussing the use of the remote regulatory assessment known as an RRA. And I believe everyone should be familiar with this at this point. You heard about it yesterday and earlier today. So this segment of the session will focus on RRAs for post-marketing safety programs. And the post-marketing safety programs I'm going to reference are the PAID compliance program. And for those who are not familiar with PAID, that stands for Post-Marketing Adverse Drug Experience. And REMS stands for Risk Evaluation and Mitigation Strategies. The latter part of this discussion will highlight elements of an RRA pilot project conducted by CEDAR's Office of Compliance, Office of Scientific Investigations, to monitor industry compliance and inform risk-based planning conducting RRAs in advance of planned PAID and REMS inspections. So let's start with a high-level overview of the PAID compliance program. To provide context, we follow a compliance program that provides information for conducting PAID inspections in order to monitor industry compliance and ensure FDA gets timely, accurate, and complete post-marketing safety information that we need to effectively monitor product safety profiles. So each fiscal year, we use a site selection tool to identify inspection candidates. The majority of our inspections are domestic. However, we do issue a small number of foreign inspections each year, with the total number of assignments varying based on available resources. And although we have the option to conduct for-cause inspections, the majority of PAID inspections are routine surveillance and generally not deemed to be mission critical. When we find firms aren't compliant, we try to work with them to bring them into compliance. And we can take actions like issuing warning or entitled letters if necessary. So in 2019, a public website was created for the paid compliance program in order to provide a consistent source of information to industry and the public. You can access the link to this website at your convenience using the slides that were provided. Now I'm going to discuss the scope of the paid compliance program. The paid compliance program is informative when considering the characteristics that may make remote assessments a valuable tool for us. We inspect entities required to report post-marketing safety information to FDA. This includes application holders, as well as non-applicants named on labels such as manufacturers, packers, distributors, or retailers. We cover products approved under an NDA, which stands for New Drug Application, ANDAs, which are abbreviated New Drug Applications, or BLAs, which are Biologics License Applications, as well as marketed unapproved prescription drugs and over-the-counter monograph drugs. Paid inspections usually occur at a firm's corporate office or that of a business partner. For foreign firms, the site may be their U.S. regulatory agent who acts as a liaison between FDA and the foreign firm. Once we identify the inspection site, our team issues firm-specific assignment memos to ORA. Then ORA assigns a BIMO investigator who conducts the inspection and communicates with our team as needed for support. During the inspection, investigators assess the firm's pharmacovigilance activities using a combination of interviews, document review, and demonstrations. Because of this, the content they review during paid inspections may lend itself to remote assessments. For example, investigators verify if the post-marketing safety information already submitted to FDA is timely, accurate, and complete by reviewing submissions along with source documents. In addition to past performance, investigators also look at written procedures and current processes to understand how the firm conducts their pharmacovigilance activities. For example, they may do a live walkthrough of the safety database and other electronic systems to evaluate impact on data integrity. Overall, the type of information evaluated in a paid inspection is largely focused on a review of documents as well as the evaluation of processes, procedures, technology, and systems in place to understand how the firm has performed in the past and how it may perform in the future. Now I'm going to discuss the REMS compliance program. FDA conducts inspections to evaluate compliance with REMS. And again, REMS stands for Risk Evaluation and Mitigation Strategies. So we do this to ensure the drug's health benefits outweigh the risk for patients. And note, not all drugs approved by FDA require a REMS, and all REMS are unique. Inspections are prioritized using a risk-based approach with a site selection priority list, and then regulatory actions may be taken if issues found during inspection are not promptly and adequately corrected. Some examples of REMS inspection classifications are no action indicated or voluntary action indicated. And also a note is REMS are enforceable, and some of the possible regulatory actions could be an untitled letter, a warning letter, seizure of the drug, or an injunction. And like with the paid compliance program, you can also find further information on our REMS compliance program website, and that is also linked in the slides that you have access to. And I just quickly want to mention some of the components of a REMS. It is important to remember that all REMS must include a timetable for submission of assessments. Another component could be a medication guide or patient package insert, communication plan for healthcare providers. REMS also come with a TASU, and a TASU stands for Elements to Assured Safe Use, which are required medical interventions or other actions by healthcare professionals prior to prescribing, dispensing the drug to the patient. Another component can be an implementation system and or packaging and safe disposal technologies. Next, I'm going to discuss why paid and REMS are amenable to RRAs. And again, that's remote regulatory assessment. That's a tool you've already heard about, so hopefully this will all come together. If we think back to the impact of the COVID-19 pandemic on resources and safety, you'll understand why ORA had the opportunity to conduct some of the assignments as RRAs. Multiple factors impacted this decision, including whether or not the inspection was mission critical, if there were travel restrictions in place, and the firm's willingness and ability to participate in the RRA. Also, as you may recall, when COVID-19 was declared a public health emergency in 2020, FDA postponed most foreign and domestic routine surveillance inspections. And as I previously mentioned, most paid and REMS inspections are routine surveillance, so they were put on hold. Now let's fast forward to May of 2020. FDA issued a press release informing the public that we may plan to use alternate tools and approaches while routine inspections are postponed. RRAs are an example of an alternate tool that we used. So eventually FDA resumed conducting prioritized domestic on-site inspections using a risk assessment system. And we started seeing a mix of both RRAs and on-site inspections for both programs. And then let's fast forward to summer of 2022. FDA issued a draft guidance for industry that described expanding the use of RRAs beyond the COVID-19 pandemic. It also highlighted the value in the use of remote assessments and opportunity to incorporate them as an additional tool to meet our regulatory mission independent of inspections. The RRA draft guidance describes possible benefits for using RRAs. At a high level, the hope is that it gives us another tool for oversight and allows us to remotely evaluate compliance to advance our public health mission. At a practical level, RRAs may be an effective tool for gaining an overall understanding of the inspected entity, allowing for a more strategic and efficient on-site inspection later. Additionally, just engaging in the RRA may help us identify issues that lead firms to promptly make corrective actions, which we could also look at during a future inspection. In the future, even though RRAs are not a replacement for inspections, they could help us accomplish more work with the potential to reach a wider range of entities, especially those that are lower risk and may not rise to the level of requiring an inspection. The wider reach also ties in with the ability to use our resources more strategically and maintain flexibility when our resources are constrained. Utilizing RRAs may also provide us agility so we can assess specific issues in a timely manner. So after hearing that discussion, I'm hoping you have at least a basic or better understanding of the paid compliance program and the REMS compliance program and how RRAs can be used as a tool. As I had mentioned, we are determined to be innovative and forward-thinking. So we launched a pilot project to further assess the value of RRAs, and now I invite my colleague, Captain Peter Dyack, to further discuss RRAs, the pilot project, and future considerations.
Thank you, Jenna, and thank you everyone for being here. We're going to collaborate on this presentation. So I'm the branch chief for the post-marketing safety branch, and on my branch we have the paid compliance program as well as the REMS compliance program and the post-marketing requirement team. So as you can imagine, during the pandemic, we had a lot of our work put on hold, put on the back burner. So we decided we're going to pivot, and we're looking at these remote regulatory assessments, and we started a pilot where we would take this on in our branch, PSB, and conduct the remote regulatory assessment in advance of an on-site inspection that ORA would be conducted. Over the next several slides, I'll go more over RRAs. I know you've heard a lot about these RRAs already, but these slides will go much more in depth on what we did, what an RRA is, and how it differs from an on-site inspection, and where we see the future of RRAs going. All right. So to start with, we may be able to get a sense of the inspected entity's business operations through conducting these RAs through interviews, reviewing organizational charts, written procedures, contracts, and other documents related to how the entity functions. Next, based on document review, we may be able to verify data that is provided in a regulatory submission. For example, we can compare line listings to summary tables and source documents to submissions. A combination of interviews, document review, screen sharing may also help us assess how the inspected entity handles adverse events. We may gain insight into personnel and staffing decisions, including their qualifications and ability to perform the assigned tasks by reviewing job descriptions, resumes, and training documents. Although challenging as conducting these RAs, we may be able to evaluate certain aspects of data integrity through interviews, screen sharing, reviewing documents like data management plans and audit trails. This goes beyond just data submitted and looks at processes and systems supporting it. Additionally, we may be able to assess activities related to the database, such as validation, reconciliation, and transitions. We may also collect documents supporting communications related to PV activities, and inspected entities should also be documenting compliance activities. So they should be reviewing records related to compliance metrics, performance indicators, deviations, and CAPA management. These would all be informative. And finally, conducting interviews and requesting demonstrations of how their electronic systems actually function can provide insight into how information is managed, how various systems communicate, how records are retained, and other practical uses of this technology. Other content and situations may also be good candidates for remote assessments, but we have to evaluate that on a case-by-case basis to determine if it's an acceptable alternative to an inspection. RAs are conducted following the same compliance programs as inspections, and Jen already mentioned those compliance programs, and you can access those on our public website. The pilot project that we started in PSB, this is in advance of an on-site inspection. The purpose of the pilot is to strengthen our ability to monitor industry compliance and inform risk-based inspection planning. We did this by exploring the benefits, limitations, and utility of conducting RAs in advance of planned paid and REMS inspections. We've heard feedback from our ORA colleagues that these programs can be very document heavy. And in fact, the first few days of inspections are spent requesting and waiting for firms to provide documents and records. As part of the pilot, we wanted to see if obtaining and evaluating these documents through an RA would help focus and streamline that on-site inspection that would happen after our RA. Throughout the process, we plan to collect metrics and feedback to help in evaluating the pilot. Specifically, we're interested in how long the RRA takes to conduct and if it's better able to focus and save time during the on-site inspection. We also want to see if we're able to identify areas of concerns and if in the future this information would help us in using our inspection resources more strategically. As part of the pilot, we also plan to send voluntary surveys to the reviewer, the ORA investigator, and the firm to collect feedback on their experience. Based on these results, we'll determine how we can best use RAs moving forward. The RA, although not a replacement for inspections, we're still being able to use the RAs to assess paid compliance during and beyond the pandemic. It's hard to draw conclusions at this point with this relatively small data set. However, we have noticed subjectively the findings from our RAs so far were consistent with observations made during on-site inspections. The RAs identified issues related to past performance, like failing to submit required reports to FDA, submitting late or incomplete safety information to FDA, failing to identify and process adverse events, and failing to maintain records. The RAs also identified issues related to firm process and impact of future performances, like having inadequate written procedures, having gaps in safety agreement with business partners, not using validated computer systems, and having employees that weren't adequately trained. The depth of what was covered in the RAs varied, but regardless, they were helpful in us understanding the firm's pharmacovigilance processes and potential compliance issues, as well as giving us information that can be used in future risk assessments and inspections. Remote assessments rely heavily on technology, and some of these issues we've seen have had an impact on our ability to effectively conduct them. These things have already been mentioned, but having unstable internet connection, poor audio quality, also sometimes the content, being able to see entities' screen is difficult during technology problems. However, also when we're sharing screens, it can be hard to read the screens. And in order to understand certain processes or verified data, we may need the inspected entity to transfer a lot of information, which can be challenging based on the technology problems that we've encountered. Finally, for all the issues and challenges I've mentioned, not every inspected entity has access to live tech support during these RAs, which further complicates and can cause delays. And from ORA's perspective, they also need framework training and support to use these technologies. However, we have been gaining more experience from conducting these RAs and remote assessments. So hopefully, some of the challenges with technology, conduct, and documents may get addressed as we explore the additional ways and as this program evolves. A lot of pushing, sorry. All right, so the RA, despite the current challenges that we have, remote assessments still provide additional tools for a subset of inspections that help us assess compliance. It could also be an effective tool for gaining an overall understanding of the inspected entity, allowing for a more efficient on-site inspection. Currently, remote assessments aren't a replacement for inspections, but they do help us accomplish more work with the potential to reach a wider range of entities, especially those that are either lower risk and may not rise to a level of requiring an on-site inspection, or sites that are in locations that pose safety risks for the investigators and we can't get there. The wider reach also ties in with the ability to use our resources more strategically and maintain flexibility when our resources are constrained. They could also be a convenient tool to do a focused look at a specific issue, like verifying source data for a particular submission. In a direct sense, it may reduce expenses for both FDA and the inspected entity. But even more, it may provide a pathway to get timely information that we need to make regulatory decisions, support enforcement actions, and inform or preclude the need for an on-site inspection. Collaborations with our foreign counterparts also ties in nicely with the strategic use of these resources. Remote assessments open the door for coordinating joint compliance assessments where there is regulatory overlap. This could be beneficial for both agencies as well as the inspected entity, as it decreases duplicative efforts. It also provides additional opportunity to strengthen the collaboration and understanding of each other's programs. And finally, remote assessments may also be a useful aid in training. With the ability to see and review documents outside of a high-pressure on-site inspection, and with more opportunity to ask questions and discuss the document internally, overall it could be a valuable training tool, and exploring additional remote assessments would be helpful in the training of new investigators. This slide's a list of available resources that are available online. And this is some closing thoughts. If you hadn't listened to the rest of the conversation, this is basically a summary that we're looking to strengthen and streamline our ability to monitor industry compliance and inform risk-based inspection planning. And we're hoping that these RRAs will help us do that. We're going to continue to evaluate our use of the RRAs, the benefits, limitations, and the utility of them, and as a compliance tool in advance of planning paid and REMS surveillance inspections. If you have any questions, we are always open. You can send your questions to our paid compliance inbox and our REMS compliance inbox. These are available in the slides. And I'd like to take a moment to thank all the people on this slide who have participated in the pilot, the RA pilot, as well as helped in preparing these slides. Thank you. And I think next up is Chrissy, who will be talking from ORA. Thank you.
Did that wake people up? All right. It's the middle of the afternoon on day 3 of the symposium, so if you've been with us for a while, you might be getting a little tired. Please feel free, if you need to, get up and stretch. I'll try to keep you awake as much as I can. For those of you who are watching online, welcome. We're glad you're here. And let's talk a little bit more about pharmacovigilance inspections. So today, one of the things I'm going to start with is talking a little bit about an overview of the FDA BIMO program. And so why am I doing this? I get asked these questions a lot. And so I think this is a really great forum to be able to talk about how we, within the Office of Regulatory Affairs, work very closely with our regulatory counterparts in CDER, and how we work and do the work that we do.
We're also going to talk a little bit about the application holder responsibilities. I'm going to focus in here on a couple of really key items that are then going to lead into a little bit of a discussion about the future of inspections for pharmacovigilance. And so really, I want to make sure that those points are the ones that are brought home. So hopefully you'll listen to that part of the presentation. All right, so we're going to start just briefly with our overview. So for folks who are not aware, FDA is run by a commissioner who is a political appointee. He is appointed by the President of the United States and then confirmed by the Senate. So as you can imagine, every time we have an election year, we have a potential for a new leader at ORA. But he's really our guiding light. Right now, we have Dr. Califf. This is actually his second time leading the agency. And underneath his office and all of the things that they do, we have our 6 product centers who review all of the applications that come in. So we look at all commodities at the FDA. Our name may be food and drug, but we also look at tobacco products, we look at medical devices, and biologics, veterinary products, et cetera. So we cover a broad range of topics and a broad range of products. And supporting all of those centers is the Office of Regulatory Affairs. And we do this through inspections, investigations, import operations, making sure that products that are coming into our country here in the United States are safe, and those that are on the market are remain safe and effective. So just a little bit about the organization of ORA. We are led by an Associate Commissioner for Regulatory Affairs. That is currently Mr. Michael Rogers. And you can kind of see down below what some of the things that we do. So we do have a large group that deals with import operations. So looking at drugs, drug components, all those things that come into our country. We also have over on the right side our Assistant Commissioner for Criminal Investigations, or OCI. You do not want them showing up at your door. Those are the folks who are going to show up with an inspection warrant. They're going to come with a warrant from a judge and a gun. So I don't want them showing up at my door, but I will tell you when I'm in a meeting and OCI is in the building, I feel safe. Because they are law enforcement, they're always carrying guns, and so I pick them out and I'm like, okay, if something happens, I'm standing behind him. We also have a large animal and human food inspectorate as well, and then we have our medical products and tobacco operations. So our investigators within the Office of Regulatory Affairs are really organized by commodity. So we have folks who do just animal food and human food inspections. But then under the medical products, we have a group that deals just with manufacturing inspections for drug products, for APIs. So that would be our OPQO, our Office of Pharmaceutical Quality Operations. For example, we also have our Office of Medical Device and Radiological Health Operations that would do inspections of manufacturing facilities for medical device products. OBPO, our Office of Biological Product Operations. So they're going to do inspections of, for example, vaccine facilities or blood banks or plasma centers. And then we have BIMO. So we cover everything we've talked about over the last 3 days here. On the pre-market side, we look at clinical trials, right? So we have our clinical investigators, sponsors, institutional review boards, as well as our non-clinical or GLP inspections. But then we also look at post-market as well. So our paid or pharmacovigilance, as well as REMS requirements inspections. So we have, I think, a unique look in the BIMO program where we get to kind of see everything that's coming in when it's new, right? But then we also still look at things when they're already on the market, and we're looking to make sure that they're still safe and effective. So we get to really look at the whole span of a product's life cycle, and that's really kind of fun and exciting. But in the BIMO program, we do have objectives, and you've heard these a little bit throughout the different talks that have happened over the last couple of days. And really, you know, our main core here is to protect the right, safety, and welfare of our human and animal research participants. That's really what we're interested in. We want to make sure folks who are participating are being protected. So looking at these objectives here, it might not be immediately clear that paid would fall under these objectives. But when we look at our second bullet and we want to ensure the quality, reliability, and integrity of data that's collected, we're not just talking about research data or clinical trial data. We're also talking about pharmacovigilance data. We want to make sure that the information that the agency is receiving is information that we can rely upon to make decisions. Do we need to recall a product? Do we need to put an alert out about something that might be concerning? Do we need to perhaps reach back out to a sponsor company and say, hey, you know, we're seeing this issue in the data. Can you provide us some more information? So we want to make sure that all the data we have coming in, regardless if it's pre-market or post-market, has quality, it's reliable, and we can act upon that data. And so that's kind of where we come in. So the Office of Bioresearch Monitoring Operations is responsible for these inspections. My investigators are spread across the United States. We try to keep folks local to where work is, because trying to travel across the United States can take a long time. You think about it, if I go from the top northwest corner of the United States and I want to go down to Florida in the southeast corner, I can probably get from here to London faster, right? So the United States, we have a lot of geography to cover. And so we have about 100 investigators in my program. They are spread throughout the United States. And to try to help with the organization of work, we have split the country into two divisions. And so we have a Division 1, which is roughly the eastern third of the United States, also affectionately known as BIMO East, because when we originally put this scheme together, I kept getting 1 and 2 mixed up, because we generally read left to right in English. And then the other 2/3 of the country are Division 2 or BIMO West. And so we really use this to try to help with the organization of work and ensuring that we're able to get our work completed and in support of the centers. So in addition to that, when we hire our investigators, in the BIMO program, they're expected to be able to do inspections for every type of inspection that we do. So we don't hire specifically pharmacovigilance inspectors or specifically GLP inspectors. All of our investigators would be expected to do both types of inspections as well as the other types of inspections that we do as well. And so that's part of the reason why we've got them spread throughout the United States. Less travel, is really nice if you can do it. And so we try to get them focused in areas where we know we have work. I do want to talk a little bit about how we get that work and how does it come to us. You heard a little bit from Jenna and Peter about how they are looking at RRAs and how they're looking at some of that work, but how does that get to ORA when they want us to go out and do an inspection? So they do a lot of work behind the scenes. They are looking at our different systems that we have for pharmacovigilance and the adverse events that are coming in. They're looking at previous inspections. They're talking to their center counterparts in the review divisions or also in the Office of Surveillance and Epidemiology that looks at a lot of our adverse event reports. And so they're getting this information and they're combining it together. And they're looking at, if we have any complaints or anything like that. And they're putting it into an inspection assignment. And they're writing up this memo and they're saying, hey, we want you to go and inspect this particular location. And we might have some additional questions or just follow the compliance program. And so that inspection assignment comes to us in ORA. I have a group of folks who take those, they review them, make sure they're complete. They have all the information we need to be able to conduct the inspection. And then they triage it out based on location. So based on where that firm is that we want to be inspected, it either goes to Division 1, East, Division 2, West, or to our foreign cadre, if it happens to be international. And as was mentioned, we do actually do international pharmacovigilance inspections as well. Once that's been triaged out to the division, it eventually makes its way down to an investigator is assigned. They go out, complete the inspection. Once they come back from that pharmacovigilance inspection, they're going to write their EIR, so their establishment inspection report. And then they're going to take that report and they're going to send it back to CDER. And the folks in CDER and the paid group are going to take a look at that for compliance. So we have a little bit of a separation of duties here where we're conducting the inspections and then they're reviewing and assessing for compliance. So they're taking everything we've written in that EIR along with any exhibits we've collected, and they're looking at that information. They're looking at other information with the other groups that they work with in the center to come and make a final determination as to what the compliance outcome of that particular inspection will be. And then they will send you the regulatory correspondence and make the final classification for that inspection. So we get a lot of questions about, well, where should I send information? Who should I contact after my inspection? Well, we do ask, in trying to be very green, that you please send us your response electronically. So if you did receive inspectional observations on a form FDA 483, we will ask that you send that to the division correspondence mailbox based on where that inspected firm was located, and we will ensure it gets to the appropriate contact within the center as well as filed within our ORA files as well. Please keep in mind that if you do receive a Form 483, that's not a final agency determination as to the compliance status of your facility, of your site, of your firm. Okay, so as I mentioned on the previous slide, it does still need to go through a compliance review with the center and with those folks. Okay, so they will take a look at that and then they'll send you out the correspondence. If you have questions about the inspection itself, you can feel free to contact our management, again, based on location. Their email information is on the slides here. If you have a question about the regulatory correspondence or about the compliance status or the final classification of your inspection, you can contact the POC that's located on the letter that you received from the center. So how do we recruit and train our investigators? And so this is my shameless plug. We're always hiring. And this QR code, if you're more interested in finding out about what it means to be an ORA investigator, you can go to this QR code here and read all about it. And, apply if you're interested. So we hire folks from all different kinds of backgrounds, not just from a clinical background, but we look for folks who also might have pharmacovigilance experience. They might have GLP experience. We have doctors who are investigators, pharmacists, pharmacologists, nurses. It really spans the gamut. We love having a diverse group of folks because that really encourages a lot of different thinking, right? Folks bring in very different ways of thinking about things based on their background. We've got folks who are just relatively out of school, as well as folks who have been in industry for 15 or 20 years, who have come to us. I like to ask my staff, why are you here? Why are you doing this, right? Because we work for the government. We don't get paid a lot of money. You're not here to get rich if you're working for the government, right? But, I thought it was really well put one of my investigators happens to be an anesthesiologist by training. And when I asked her that question, I said, why are you here? She said, you know, in the operating room, I can help one person. But at FDA doing this job, I can help millions of people. I can help them. I can ensure that the products we're getting out to them are safe and effective. And to me, that's just like mind blown, right? I'm here to help people. That's why I do what I do. And I'm very passionate about what I do. And you should be too. When we bring our new folks in, they go through basic training. Every ORA investigator goes through the exact same basic training where we teach them about the law, we teach them about personal safety, and how to conduct an inspection, gather evidence, write reports, etc. After that, they go to more advanced training, and that'll be specific for their commodity area that they've been hired into. So for BIMO, we start them with our clinical course, where we talk about clinical investigator inspections, institutional review board inspections, sponsor inspections, because that's really the bread and butter of what we do. And so that's what they start with, and then they'll move on to bioequivalence training, paid and REMS training, GLP training, etc. But really, a lot of the information that we're providing them and that we're training them on is available to you as well. So I highly recommend, if you're not familiar with it already, to become familiar with the IOM, the Investigations Operations Manual. It is publicly available online on FDA's website. It is updated twice a year. And it's really kind of the general overview of everything that we do and how we do it. So how would you expect us to issue a 482 or a 483? How would you expect us to show up to your site? Really here, folks focus on Chapter 5 is for inspections. If you do nothing else, you can read the rest of it. If you can't fall asleep at night, it'll probably help. In Chapter 5 as well, there is a subchapter 5.14 that is specific to BIMO, so take a look at that as well. And as was mentioned earlier by my colleagues here from CEDAR, we do have compliance programs. There is a compliance program specific to paid, so take a look at that as well. So paid or pharmacovigilance, depending on where you're located and what your lingo is. And between those two items, you should be really prepared for an inspection, because the compliance program is going to walk you through the types of documents, the types of information that we're going to ask for while we're on inspection. Now, when we show up at the door, don't just have like a big box with everything that's in the compliance program, you know, that you put in the conference room and say, have at it. Just because it's in the compliance program, we may not necessarily ask for everything that's in there during the course of an inspection. We may ask for other things. The point is that we want you to be prepared with the types of information, and it should be readily accessible. You should know where it is and how to get to it quickly. Okay. So I'm going to move on a little bit. There's a couple of points I want to make in regards to application holder responsibilities, and this is specific to paid or pharmacovigilance. And so one of the things I want to talk about, and if you've been with us for the last couple of days, you may have heard folks talking about in sponsor oversight, control and oversight. And it doesn't change when you get to pharmacovigilance. You need to ensure that you have control and oversight of that data that you're reviewing, because remember, as an application holder, you're ultimately responsible for ensuring that that information is being sent to the agency in a timely manner. Application holders are required to have specific written procedures for how are you going to determine what adverse events are even out there. So what is your surveillance plan? How are you going to go about doing that? When you do find them, how are you going to receive them? How do you take them in to your office, to your program? How are you evaluating them? How do you even make that determination if it needs to be submitted to the agency or not? And if it does, how are you reporting that to FDA? And this is all very important because we have so much electronic data now that we really want to make sure that if we haven't looked back and maybe updated our processes in a while, maybe we've had some new updates to our software, we're going back and taking a look at those procedures and those processes, making sure they're current and up to date. But also making sure that we can have access and that you have access. So remember, as an application holder, you're responsible for the information that's coming in. You should have access to what your subcontractors are providing you or submitting to you. And as FDA investigators, we should have access as well to those systems. Because also, some folks may be aware, some may not be aware, but in the Food and Drug Omnibus Reform Act of 2022, which was in December of that year, there was a change made to regulations or to the law that says that FDA must be provided access to electronic systems. So it's really important you're keeping your data electronically, that we're able to access those systems. Now, I don't want anything other than read-only access. I only want it for the time that we're doing that particular inspection. Okay? But that can also save you some time. You know, we used to traditionally always do kind of the shoulder surfing, where you had to have somebody who was there the whole time, clicking through for us. We'd tell you what we wanted to look at, where we wanted you to look. Yeah, we're finally moving into the digital age now. We're going to start looking at those systems. So we want to make sure that, you know, we can have access to those systems as well. And Congress thought that that was important enough that they actually wrote that into the law. And so that includes not only preclinical or clinical data, but includes post-market data as well and post-market activities. And so just making sure as an application holder that we have access to your systems, but your subcontractors are aware as well that if we come in to do an inspection at their location, that we would need access to their systems as well. I'm not talking about every system under the sun, right? I don't care about your financials. That's not what I'm here for. But just those systems where you keep that pharmacovigilance information. And, you know, and it's really important, right? We have all this information that's coming into FDA now, and it comes in so many different ways. And the majority of this really is electronic data, right? So we have patients and consumers who might be reporting adverse events voluntarily through our MedWatch program, or they might be voluntarily reporting that to the manufacturer. Hey, you know, I had this issue, I have this concern. Once that gets to that manufacturer, you know, really there's that mandatory reporting requirement to have to review that and then submit it to the FDA. And all that information ends up in our database. And the more we can do this electronically, the faster it happens, and the better chance we have of making sure we're capturing things, right? We can review them fairly easily. We also want to make a point here that audit trails, please ensure audit trails are turned on. If you're an application holder and you've never looked at an audit trail, please do so. It really benefits you to look at the audit trails as well. Make sure they're in a human readable format. Because we've gotten audit trails when we've asked to look at them to see how things have been changed that really have made no sense. And it becomes harder to reconstruct what actually is occurring. So again, I did mention safety reports. They do need to be in an electronic format per the regulation. There's a link here to that. And we have 2 guidance documents out there that can help you with that as well. And so those are both linked here within the slide presentation. So please take a look at that, ensure the information is in a form that we can utilize. It does need to be in a format that FDA can process, review, and archive. And so doing that electronically really is the easiest way to do that. So you heard from my colleagues about the RRA pilot, and we're really excited about this because I think it can, have some benefits for us on the ORA side as well. And so we're going to continue that pilot throughout the rest of this year and then do some review and look at it and see, hopefully it's successful, and how we want to continue to incorporate that within our program. Again, I talked about access and inspection access, and we talk about electronic access. We do utilize box.com for a lot of our electronic document requests. We have done some on Teams as well. We know that in a lot of companies, who has a CD drive anymore in their computer? I can't remember the last time I had one. I know USB drives are being locked down. So really being able to transfer files electronically through box.com is great. One point I do want to mention though, if you're sending us a response to a 43 or you're sending us some documents, please hopefully send them as a PDF. They're a lot easier for us to utilize. Please do not send us a link to a SharePoint site that you have at your facility, because we're not going to be able to access that. We won't get past our own firewalls to be able to get there. So if you can send us a PDF, preferably bookmarked, I'm going to put that plug out there. With anything that you're sending us, that just makes it a lot easier for us to find the information. We end up with fewer questions down the road. And really, when we talk about inspection readiness, we've heard the last several days about kind of this, there's been a little bit of a theme, right, about moving things upstream a little bit, making sure you have your risk assessment early, making sure you're doing those things. I would say that inspection readiness for your pharmacovigilance inspection starts before your first subject is enrolled in your first clinical trial. It starts with your risk assessment, with how you build your protocol, how you're gathering that data that's going to go in your label, right? Because then that's going to affect your pharmacovigilance down the road. So really being ready for that first inspection starts before you even start your first clinical trial. And it's just constantly looking at what you're doing. This is an iterative process. It's not a one and done when we first start, but anytime something changes, it's going back, taking another look at that risk assessment and making sure we have things right. So just to summarize, we do work very closely with CDER. We have a lot of communication with our folks and our colleagues in the center, especially around pharmacovigilance. Just remember, application holders, you do have responsibilities for electronic submissions, and there's a lot of great information in those two guidance documents, as well as the regulations about that. And our inspections will continue to evolve. With this RRA pilot, we're looking at ways to perhaps look at some information ahead of time so that, we don't have to spend as long of a time on your site. And hopefully it's going to free up some time for you as well. So thank you very much. I will take any questions during the panel, and I'm going to turn it back over to Carolyn.
Thank you, Chrissy. Thank you, Pete and Jenna. Unfortunately, Bob was not able to join us. He really wanted to be here, but he pre-recorded his presentation. So we'll turn it over to Bob, and then there will be a break afterwards. Thank you.
Hello, my name is Bob Ball. I'm the Deputy Director of the Office of Surveillance and Epidemiology in CDER at FDA. Thank you for inviting me to talk today about artificial intelligence and pharmacovigilance. The opinions in this lecture are mine and don't necessarily reflect the views of the FDA. My only disclosure is that I have a patent on a natural language processing algorithm. Today I'd like to talk to you about what is artificial intelligence, how might AI apply, to pharmacovigilance, then a framework for readiness for thinking about AI and pharmacovigilance, some more general regulatory consensus development activities for AI and PV, and then I'll summarize my talk. So to begin, what is AI? Artificial intelligence has been broadly defined as the science and engineering of making intelligent machines. Many technologies have been placed under the AI umbrella. Machine learning and natural language processing are the two that have been most commonly applied to pharmacovigilance. Machine learning is defined as a technique that can be used to design and train software algorithms to learn from and act on data. And NLP is the application of computational techniques to the analysis and synthesis of natural language and speech. To take a little bit of a deeper dive into these definitions, classical machine learning is divided into two types, supervised and unsupervised. The biggest difference between the two is that supervised machine learning requires a human annotated data set that the algorithms train on. Unsupervised algorithms generally just look to cluster data in interesting ways and based on statistical principles. Deep learning is a type of machine learning that is based on a technique called convolutional neural networks. And this approach has revolutionized much of artificial intelligence work and is the basis for large language models, which I'll talk to in a minute. The key, though, for convolutional neural networks and deep learning is to have large data sets. And this, of course, can be a challenge, especially in a specialized field like pharmacovigilance. On the bottom right of the screen is a Venn diagram that shows how some of these technologies can fit together. You'll see there's the broader blue, which is artificial intelligence in general. ML is machine learning. NLP is natural language processing. Deep learning is a type of ML. And where all of them intersect is where large language models happen. So what are some key concepts about large language models? So first, large language models are designed to predict the next word in a sentence. They do that by training a large convolutional neural network from a massive amount of data. This has largely been taken from the internet, but any text sources could be used. And it's very computationally intensive to do this. Sometimes these algorithms are then fine-tuned. This can be used by providing human feedback, for example, telling when the result of a query is correct or incorrect. Or it can also be used by providing a specialty data set, such as electronic healthcare data or pharmacovigilance data, to make the large language model work better on those data types. The key for interfacing with large language models is prompt engineering. It matters how you ask the questions. So subject matter experts, such as all of you in pharmacovigilance, and AI scientists can work together to design and refine prompts to get the best answers from these models. There's been an explosion of large language models, and this presents many opportunities and challenges. What this graphic illustrates is on the far left, beginning around 2009, large language models began to be introduced. And all the way through that S-shaped black line to the right, newer large language models and bigger large language models have been made available, including GPT-4, BARD, which we've heard about a lot in the past year. And a very important one for our work is the Llama model. Llama is a model that was released to the public domain by Meta, the parent of Facebook. And it's important because it's a relatively small model and can fit on single servers and can be used to process internal data without the massive computational infrastructure that's required. So there's a lot to be learned about these models and a lot of changes that are likely to be coming in the next few years. So exactly how they're going to be impacting pharmacovigilance is yet to be determined. So now shift gears a little bit to pharmacovigilance. Most of the focus in pharmacovigilance with AI has been on individual case safety reports. And why focus on them? Well, they have a long proven track record of identifying new safety issues. They'll likely remain important for the foreseeable future. There's an increasing number and variety of data sources that need to be processed. For regulators, there's a growing volume of ICSRs that need to be analyzed. You can see on the right, that's a graph showing the number of reports over the past 10 years for the FDA adverse event reporting system. And now we're receiving over 2 million reports. And we need some automation to help assist with the processing that. Also, submission of ICSRs is a global requirement. So harmonizing around the approach of applying AI is an important goal. ICSR processing has been happening for at least a couple of years, primarily by the pharmaceutical industry. This slide is taken from an article by Schmidger et al. Describing the ICSR process, which includes intake, evaluation, follow-up, and distribution. What I like about this graphic is it starts with a core of those main concepts, but then shows all the different tasks and subtasks that need to be computed to make this process work. Some examples of ICSR processing include identification of the 4 minimum elements of an ICSR, the identifiable reporter, patient, an adverse reaction, and a suspect product. Additional steps that have been computed include determination of important regulatory categories such as seriousness and expectedness. The next step in the application of AI, though, is ICSR causality assessment. As all of you know, causality assessment is determining whether a drug is likely to have caused the adverse event or not. Right now, that relies primarily on expert judgment and global introspection. You can see on the right, this graphic illustrates, in a manner analogous to the prior slide, some of the core data points that are needed: drug exposure, the adverse event or outcome, temporality between the drug exposure and the outcome, and alternative explanations, with some of the subtopics in each of those categories. But what's important to note is that the assessment of ICSRs for causality relies on information internal and external to the report. And also that exactly how safety evaluators analyze data has not been completely described. It's a complex process. It varies often by the data available. And that's necessary to really understand how best to create a computable application. Many are hopeful that large language models will provide the nuance that will allow this to come to fruition. But when thinking about whether or not an AI algorithm is ready for ICSR processing and assessment, here are some things to consider. Algorithm performance is arguably the most important, but other things such as documentation, transparency, explainability are also needed. And then quality control. Quality control is very important because at the moment, none of these algorithms are perfect. So we're going to need a human in the loop to monitor the output of these systems. But if an AI algorithm doesn't achieve this perfect performance required for full automation, the key challenge then becomes, how do you ensure quality without reducing the efficiency gain from the AI algorithm to make it, to keep it useful? So you could have 100% validation, but then that might defeat the purpose of the algorithm. So some things to think about in how one might approach such a quality system include a risk-based approach in which the effort is proportional to the implications of misclassification on the overall evaluation goals. So if the algorithm is extracting age from a piece of text, for example, that might require less validation than an algorithm that's making a determination that this drug caused this adverse event. It will also be important to incorporate the reliability of the AI algorithm's performance through carefully applied principles of algorithm development and then potentially formal confidence metrics. But the selection of quality assurance techniques is still a bit of an art, and more work is needed to determine what are some of the best approaches. Some techniques include things like sampling or simultaneous independent algorithm application, or incorporating the AI algorithm into a human-led evaluation process that includes other means of quality assurance. So I'm going to shift gears now to some more general regulatory and consensus development activities for AI and PV. You're probably aware that FDA released a discussion paper last year on AI and drug development, including pharmacovigilance. The EMA also released a reflection paper on AI and the life cycle of medicines last year. The FDA-EMA Pharmacovigilance Cluster formed a working group on AI late last year, and Health Canada and PMDA are joining that as observers this year. There's been a CIOMS 14 working group for AI and PV ongoing for the last 2 years, and that working group is trying to develop consensus approaches to how AI might be applied to PV. There's also a pharmaceutical inspection cooperation scheme working group or expert circle focused on how to do inspections for emerging technologies like AI and ML that is forming. FDA's paper had as its goal to promote mutual learning around 3 main core issues: human-led governance, accountability and transparency, quality, reliability, and representativeness of data, model development, performance monitoring, and validation. FDA received many comments and is in the process of digesting those comments with the goal of producing a guidance document over the next year. Some of the work over at FDA includes continuing these collaborations and mutual learnings, tracking the use of AI ML in drug development, including pharmacovigilance, advancing and supporting development of demonstration projects to identify best practices, and probably most importantly, developing a risk-based framework for credibility assessment that is based on work using this approach for models in deterministic systems, such as, for example, pharmacokinetics or medical devices. So there's been a lot of unanticipated challenges with AI, and I suspect those will continue. A short while ago, the Wall Street Journal had this article about entitled, "Does Working with Robots Make Humans Slack Off?" And this is based on a long observed phenomenon in human teams where a small number of the team members do most of the work. This may be something that you all have experienced yourself. And the Wall Street Journal article cited this paper on the right about a test that was run looking at how humans versus a human AI system for inspecting electronic circuit boards performed. And what they found was that the human alone did better than the human and AI. And they hypothesized that the reason was humans relied more on the AI algorithm when it was available to them. So in summary, classic AI systems are being used for ICSR processing and analysis, but uncertainty remains about the best approaches to monitoring quality. Humans in the loop will be important for the foreseeable future. LLMs are very powerful and are likely to change the landscape, but there's still a lot of uncertainty and a lot of technical and policy considerations to think about. And FDA is building a regulatory approach for AI and drug development, including pharmacovigilance based on a risk-based framework for credibility assessment. I'd like to acknowledge my colleagues and thank you for letting me speak today.
Regulatory updates from FDA and MHRA. So up first is Lauren Bateman, who is a senior informatics advisor in the Office of the Commissioner at FDA. And she's going to walk us through the post-market safety reporting requirements for combination products. And then next we have Saranjan D, Deputy Director for the Regulatory Science Staff at FDA, and he's going to take us through the new ICH E2B R3 guidelines for submitting ICSRs to FAERS. And finally, we're going to circle back to Claire Longman to discuss updates to pharmacovigilance requirements at MHRA. So I'll let Lauren.
All right. Thank you, Carolyn, and thank you guys for having me. So I'm going to be talking about the exciting world of combination products and the post-marketing safety reporting requirements for them. So we're just going to be covering the basis today, but with regards to combination products, I'm going to be giving an overview of the 2016 final rule for PMSR requirements for combination products, which is applicable to the combination product applicants and constituent part applicants. So first, what are combination products? Combination products are their own distinct type of medical product. They are a combination of two or more different product types. So this could be a drug and a device and a biologic, a drug and a biologic, or it could be all three. Some of the common examples are a pre-filled syringe, a drug-eluting stent, or a first aid kit that contains both devices and drugs. The drugs, devices, and biologic products that make up a combination product are called constituent parts. And each constituent part has its own mode of action. Combination products are assigned to a center who's responsible for the regulatory authorities associated with that product, and that includes post-market. And that center is called the lead center. The lead center is determined based on what's called the primary mode of action, or PMOA. The PMOA is the single mode of action of the combination product that provides the most important therapeutic effect to that product. So let's look at some examples. So first, we have a pre-filled vaccine syringe. So in this case, you have a vaccine, which is a biologic constituent part, and you have a syringe, which is a device constituent part. When thinking about the modes of action, you have the action of the vaccine, which is to provide protection to the patient. And then you have the action of the syringe, which is to deliver that vaccine to the patient. So in this case, the biologic or the vaccine is going to be the PMOA, and therefore, because the product would be regulated by, a vaccine would be regulated by CBER, the combination product would be assigned to CBER. A second example would be a drug auto-injector. And this is going to be really similar to the example we just talked about. You have the drug and you have the delivery device. The action of the drug is going to be the PMOA. And so if that drug were to be regulated by CDER, the combination product would be assigned to CDER. And then my third example is a drug-eluting stent. In this case, you have a stent or a device constituent part, and its mode of action is to hold open an artery. And that stent may have a drug coating on it. And that coating may be an anticoagulant to prevent blood clots. And so in this case, the action of the device, which holding open the artery, is going to be the primary mode of action. And so this combination product would be assigned to CDRH. There's three types of combination products. Now those products I was just mentioning on the last slide are called single entity combination products. They're physically or chemically combined. Think of them very hard to separate. But another type of combination product is called co-packaged. So an example of this would be if you have an empty syringe that is packaged together with a vial of a drug. Another example would be a first aid kit that contains individual drugs and individual devices. And the last type of combination product are called cross-labeled. They're less common, And these are when they're individual products that are packaged separately, but they are labeled and intended to be used specifically with one another in order to achieve their intended therapeutic effect. An example of this would be a light-emitting device that is intended to be used with a specific light-activated drug. So now let's talk about post-marketing safety reporting requirements for combination products. In general, when thinking about the PMSR requirements for a combination product, the requirements are going to come down to whether you are a combination product applicant or a constituent part type applicant. And I'm going to explain the difference between those. So first, and most commonly, you have a combination product applicant. These are going to be applicants who have the only or all applications for the combination product. So those single entity examples I mentioned, like a pre-filled syringe, if you have that product, you're going to be a combination product applicant. And as a combination product applicant, you have responsibilities for application type-based reporting requirements and constituent part-based reporting requirements. And we'll talk about those in a moment. But first, the second type of applicant are constituent part applicants. These are most commonly going to be in the situations of cross-labeled combination products. So this is when you have one application for one constituent part of the combination product, and there's a second applicant with a second application for the other constituent part of the combination product. Constituent part applicants are only responsible for the application type based reporting requirements. So let's talk about what those requirements are. So application-based reporting requirements, which is along the top row here or orange on my slide, those are going to be your typical requirements based on the application type that the combination product received marketing authorization under. They're not listed on this slide because applicants should typically know what they are, but they are outlined in 21 CFR 314 for NDAs and ANDAs, 21 CFR 600 and 606 for BLAs, and 21 CFR 803 and 806 for devices. Now, moving along to constituent part-based reporting requirements. This is based on the constituent parts that make up your combination product. So thinking about the first column here, if you have an NDA and that NDA includes a device constituent part, you have additional reporting requirements on top of those that come along with the application type. In this case, you would be responsible for 5-day remedial action reports, malfunction reports, and correction or removal reports and records. And while I don't have time today to dive deeply into what each of those are, I do have a resource slide to help point you to where to go to so you can learn more information about them. But I do want to go through an example. So let's say you're a combination product applicant and you have a product that is a co-packaged drug and infusion set, and it's marketed under an NDA. If you determine that a 15-day report is required after a serious, unexpected adverse event that was related to overinfusion of the drug occurred, but you then determine that the infusion set could have caused or contributed to that event, then you also are responsible for submitting a malfunction report. There may be instances when you have to submit a malfunction report and not a 15-day report. An example of that might be a co-packaged combination product in which you have a syringe that the sterile barrier was compromised. If the healthcare professional discarded that syringe before using it, there was no adverse event. However, had that malfunction occurred and the compromised sterile barrier would have been likely to cause or contribute to death or serious injury, then a malfunction report would be required. The other columns in this table are similar in format. So the middle one, if you have a BLA or a device application, and it includes a drug constituent part, then you also must submit field alert reports or FARs and 15-day reports. And last, if you have an NDA or a device application, and that includes a biological product constituent part, then you're also responsible for biologic product deviation reports, or BPDRs, and 15-day reports. So where should you be submitting these? So individual case safety reports, or ICSRs, is the term that's used to include 15-day reports, 5-day reports, malfunction reports, and reports of death or serious injuries. And applicants must submit ICSRs in accordance with the procedural requirements in the regulations associated with the application type of the combination product, and they should follow the policies and the procedures of the lead center. For reports other than ICSRs, so that includes FARs, BPDRs, and correctional removal reports, you would submit those reports in accordance to the procedural requirements and policies associated with the report type. A combination product applicant may submit a single report to comply with more than one reporting requirement if the reports can be submitted in the same manner and the combined report satisfies all applicable reporting requirements, including all of the submission timelines. So for example, if you are a combination product applicant with a CDER-led NDA for a drug device combination product, and you have to submit a 15-day report and a malfunction report, then that applicant can satisfy both of these reporting requirements in a single report that's submitted within 15 days, and that report would be submitted to FAERS. So PMSR reports for combination products must contain all of the information required for the report type under the applicable regulations. And this includes relevant information on the entire product, including each constituent part, regardless of whether it was implicated in the event. So if you have an event that you have to report a 15-day report, and it appears that there's no relation to the device constituent part, you should still include information about that device constituent part when reporting it to the FDA, even if it was not implicated in it. And this slide includes some of the examples of the fields in which you can include this information in each of the reporting systems. So on the left, FAERS, the Drug and Non-Vaccine Biologic Adverse Event Reporting System, there are device constituent part fields. So you can indicate what the device brand name was, its common name, its pro code, and even more. And then on the flip side, in CDRH's EMDR system, which is for device adverse event reports, there are fields in which you can include information about the drug or biologic. So I only had time today to go over a very high level, overview of this. So I'm leaving you with many resources. And I really want to draw your attention to bullet 4, which is our guidance for industry. This is a fantastic resource. It goes into so many more details than I could cover today, but it also has a lot of really great examples to help you think through your products and how these regulations would apply to you. So what do I hope for you to take away today? First, do you have a combination product? And if you do, what are the different constituent parts and what is the lead center for that product? And then second, what are the PMSR requirements that apply to your product and where should those reports be submitted? And lastly, what constituent part information should you be including in the reports for your product? So is there a device in your combination product? Please do include information about the device. And if you still have questions, we're here to help. You can contact the Office of Combination Products at combination@fda.gov. You should also reach out to your lead center if you know them. If you don't, we will help connect you to the right people to answer your questions. Thank you for the time today. And I want to welcome my colleague Saranjan to talk more about FAERS updates.
All right, good afternoon. My My name is Suranjan Dey. I'm the Deputy Director of Regulatory Science Staff and Office of Surveillance and Epidemiology in CEDAR in US FDA. So today's talk will be about updates on FAIRs. So what do we, what are we going to talk about? We'll recognize that FDA will require reporting of pre-market and post-market safety reports in the ICH E2B R3 format to FAIRs. We communicate the implementation status, timelines, and readiness. And FAIRs case processing and analytical modules now accommodates product quality defect and complaint reports. So we have made one system for both adverse event and product quality. All right, so FDA is in the process of implementing electronic submission for both post-market and pre-market safety reports using ICH E2B R3 standards. There'll be separate submission path and business rules for pre-market and post-market safety reports. We have all the technical specification documents that have been posted on the FAIR's electronic submission page. And all communication will be done via the FAIR's electronic submission page on fda.gov. And the link is at the bottom of the FAIR's electronic submission page. So the way we are implementing this, and partly also has been implemented, is we're going to have two separate submission paths, one for pre-market side and one for post-market side. And the reason being that we all know that post-market report eventually gets into the public dashboard, versus pre-market report will not get into the FAIR's public dashboard. So it will not be publicly published. So we have two separate paths, which means if you're submitting through the gateway, you have two separate headers. Now we also have two separate headers in the pre-market side for CEDAR and CBER. And the reason being that we did find that there are some IND numbers which overlap between the two centers. So as a result, we made sure they are separated out. And then once you separate it out in CEDAR and CBER, we also know, that it is for an IND or for a post-market, and the post-market headers are just shown on the screen here. And this is basically a very important table to keep in mind, that if you're submitting electronically using the E2B standards, then for the IND safety reports, you have the AS2 header, you have the routing ID, but within the XML, it also identifies with the N14 saying that it's a pre-market report, and N2R3 says within pre-market it's a CEDAR IND. And similarly for the other three values that you have, of which the third value is for pre-market report, but then they are the IND exam bioavailability and bioequivalence study safety reports. And finally, you have the post-market safety reports. The values of this are basically the same as we did in R2 for AS2 header and routing IDs. So now what you have is you have two options to submit these reports. As we know, post-market safety reports are mandated to be submitted electronically. So these are the two options. When it comes to pre-market report, this will be the two options. Of course, the day we publish to say that we are ready for pre-market, from that point, you'll have 2 years to comply to the submitting of electronically. So option A is database to database submission, which is again the E2B standard. The sponsors submit through the gateway, they get the first acknowledgement, FAIRS processes it, and they get a second acknowledgement which says is it successful or not successful. Option number B, where most of the companies who use option number B who have less volume reports, because you have to submit these reports one at a time, it's a web form, and you submit this report through a safety reporting portal where you have to register first, and then you submit those reports, which then gets into FAIRS. So safety reporting portal intended for sponsors, and also we have seen a lot of CROs who use that, who do not have an infrastructure for the gateway. And these are individual reports only, no batch reporting can be done. One report at a time, can be used for both commercial and research INDs. And right now it's not available for vaccine reporting. If you're a CRO, you need to have separate accounts for your separate customers or license holders. Post-marketing report and pre-market report, they will be upfront. It will ask you in the safety reporting portal that are you submitting for a pre-market or a post-market. It's an online form. If you have any document where you have like a narrative written, I mean, the narrative has to go as typed in narrative in the narrative section of safety reporting portal. We have seen some companies, they would say, see attachment. Yes, you can add attachment to the safety reporting portal, but it doesn't help us with all the AI work that we are trying to do. All right, this is free of cost. And if you are only submitting using the as a post-market reporting, you really don't have to do any actions. But if you are going to be submitting pre-market report, then of course you have to register those INDs for which you're going to be submitting, so that when you go on the online form, it will automatically give you the IND numbers for which you are submitting the report for. And any questions, fairs.esub@fda.hhs.gov. This is the email address where you can ask all your questions. There are some documentation which are available, which are important. So this is the technical specification document. The regional core data elements. This is, if you're submitting E2B, this is a very important document. It lists all the ICH and the regional data elements, all the attributes, the conformance, business rules, and expats and acknowledgement attributes. This is a forward compatibility rule. If you have submitting pre-market report, then you don't have to worry about this, because pre-market report comes through ECTD as PDF. So we've never had E2B R2. But for post-market, yes, you need to look at this document to move from R2 to R3, especially for the, this is for the regional elements. And then finally, we have given some sample XML files, which has different variety of both post-market, pre-market, combination products. And it gives you some sample instances to show you how the XML will look like. So going into IND safety reports, this is an important thing that FDA is working towards, and the process that we are trying to do is to get these gets a submission electronically. We know we all submit ECTD, but now that we will be submitting electronically, so through the XML or through the safety reporting portal. What types of reports for IND to be submitted? The first top three, based on 21 CFR 213, 32A, B, and C. So those are the types of reports that will be submitted through FAIRS. Remaining reports will continue to be submitted through ECTD. How will it help sponsors? So first thing, no need to submit the 1571 or cover letter for the IND safety reports. No separate submission for cross-reported IND. The structure now accommodates the cross-reported IND, so there'll be only one report that needs to be submitted. You get immediate acknowledgement of an ICSR, and some additional benefit is you're submitting directly from your safety database, and I believe most of the companies have to submit through this regulatory affairs. If they submit through ECTD, that also is eliminated. Then you have IND exempt. So a little bit of IND exempt. We have, currently they don't have an electronic rule, so they will be submitted as 3500A MedWatch form to this email address. We also encourage that the systems also will be ready with E2B and safety reporting portal, so you can utilize that to submit. So to prepare your IT systems, you can prepare your IT systems with the E2B R2, or you can go directly to safety reporting portal where you just have to get an account. And then any questions, anything that you may have, you would ask your questions to fairesub@fda.hhs.gov. But if you don't have an account in SRP, you will ask for an account. If you don't have an account in ESG, you will first have to get that account and do all your testing before you submit. For IND exempt, you also have to request through the NextGen portal for a pre-ANDA number, which eventually will become your ANDA. So that number is also important to identify that, say, IND exempt BABE safety report. Where are we with our plans? So we have published all the regional specs. Our tools have been enhanced. The gateways has been set up from our side. System testing has been done. We did some industry testing with getting some of those IND safety reports and the post-market safety reports, all in E2B-R3 format. And with public communication, as we move through, we have actually implemented and published to say that on January 16th, I believe, that we went live with the post-market safety report in E2B-R3 format. So that from that point, you have 2 years to move from E2B-R2 to R3. But if you are safety reporting portal, you're using safety reporting portal, you can continue to do that. And so this is what was done. So right now, we have not still published anything about IND safety reports. So that's why I continued your ECTD format submission. Now, once you have created your E2B R3, and if you're using the gateway, how do you test your XML? So we have FDA also is giving a validator, and it's already published, where you can upload your file. And click on validate. It will tell you the messages, error messages that you may have. You should go back, fix them. And once you don't have any messages, that means you have fixed your systems and you are ready to submit through the gateway now. What changes you may notice once we all start submitting through the E2B-R3 format? So now you submit through ECTD, goes to the gateway, regulatory submission database, and then finally we take that and put it into FAIRS. Moving forward, if you use the E2B XML or you use the safety reporting portal, they all will directly go into FAIRS, and none of these processes, the ones in excess, will go away. And now I'm only talking about the safety reports here. So once we start year 0, which when we go live, and then from that point, you have 2 years for mandatory ICSR submission via ESG or the safety reporting portal. So here is a proposed timeline that we have. So January 2024, we did post-market, we went live. This is another thing which happened after this happened very recently, where FAIRS is now becoming a more enterprise system. It can capture now cosmetic safety reports. Excuse me. So now the voluntary period has started for E2B-R3. Sometime in March, most likely looks like it probably will be first week of April, that we're anticipating the final guidance and the FR notice will come out for IND safety reports. Which we have E2B-R3 format, and for that you will reference all the updates will be provided on the FAIRS electronic submission page. Now once that goes, then you have 2 years to start working to move through from ECTD to E2B-R3, and then once you have moved, you cannot revert back to legacy methods. And finally, probably March 2026 is when companies will now have to comply with submitting pre-market and post-market ICSIs in electronic format or through SRP. All right, so FDA's approach in harmonizing, so we have harmonized the safety and quality databases. This is a big accomplishment in FDA, and that has enhanced our review. Now 2 different offices actually see 1 report, and the follow-ups that come, they also see 1 report. And the analytics module also has been updated. So the analytics module, everybody sees the same report. And so by maintaining the integrity of new data and critical legacy determination, we have really done a good job in putting all the systems together. Some of the references, these are all the documents that we have are references. And in summary, we recognized our submitting safety reports electronically, understand the submission methods and mechanism of different types of reports. We communicated the implementation status, timelines, and readiness, and then finally acknowledge FDA's approach to harmonize safety and quality. So with that, I finish my presentation, and I would like to, and closing thoughts, soon accept pre-market safety reports electronically. And with that, I invite Claire to give her presentation. Thank you.
Okay, hello everybody again. I'm going to be taking you through some MHRA updates. So for those of you who couldn't join yesterday, Steve Vinter gave an update on the compliance team strategy. So if anybody did miss that, I encourage you to go back and have a look at that, because that was a really good presentation on some of the updates that the MHRA are making within the compliance teams and the direction that we're traveling in. But today I'm going to talk you through a bit about regulatory updates in pharmacovigilance. And in order to do that, I want to give you a bit of history in our regulation and the basis of where that's come from. So for anybody who thought they were going to have an easy last session, apologies, I'm going to bamboozle you with EU and UK legislation. Once I finish that, I'm going to move on to our inspection program. We've had a bit of a transition in the MHRA, so I want to talk you through some of our organisational charts and just show you where we fit as a PV inspectorate. And I'll talk you through our inspection program, and then we'll have a bit of a future look at the end to finish. So just to start with, and so we're all on the same page, I wanted to talk to you about some of the major changes to PV requirements in the EU. And the UK was part of the EU for a long time, and therefore our pharmacovigilance legislation is entrenched within EU legislation. And some of the major updates that happened the biggest one being in 2012, came to by the update of Directive 2001/83/EC. And this brought with it the implementation of the GPVP modules. So they are a suite of modules that really go into a lot of detail about how pharmacovigilance should be implemented within the EU. Another update came in 2007, and that brought with it the addition of vigilance. Now, this is a centralised repository where all ICSRs that are required to be reported in the European Union are reported to that one centralised repository rather than being individually reported to EU member states. Then in 2020, the UK voted to leave the European Union, and that transition period ended on the 31st of December 2020. So from the 1st of January 2020, the MHRA became UK standalone regulator of medicines and medicinal devices. And Great Britain, which is made up of England, Scotland and Wales, now operates a legal framework that incorporates EU legislation into the UK law. And MHRA takes the sovereign decisions for medicines authorised in respect to Great Britain. Now, what's important to understand is that the UK is made up of those countries within Great Britain plus Northern Ireland, which makes up the UK as a fourth country. And Northern Ireland and products that are authorised in Northern Ireland are still required to be under EU decisions and under PV requirements, and that's under the Northern Ireland Protocol where that's specified. And that was implemented to prevent a hard border between Northern Ireland, which is in the UK, and the Republic of Ireland, which is part of the EU. So this is where it gets a bit complicated. So I'm going to talk you through some of the ways that products are authorised within the UK. So we have, despite leaving the European Union, a number of products that are authorised in Northern Ireland are still centrally authorised by the European Union. So these are innovative products. And under Regulation 726/2004, they have to be authorised under what we call the centrally authorised procedure. Now, that means that one application is submitted within the EU, And then if that submission, that application is granted, then the authorization immediately becomes effective throughout the European Union, and that includes Northern Ireland still under the Northern Ireland Protocol. Now, we also have products that are authorised nationally by the MHRA. Some of those products are authorised in respect to Great Britain only. As I said, that's England, Wales and Scotland. All UK nationally authorised products are under the Human Medicines Regulation, and pharmacovigilance sits within Part 11 of that. Now, if they're authorised just in respect to Great Britain, they are authorised under Schedule 12A of the Human Medicines Regulation. If they are authorised in respect to Northern Ireland, so these could be products that are just authorised in Northern Ireland, but they could also be authorised UK-wide, then they are granted by the MHRA, but they would be under the Commission Implementing Regulation, and they are those products that aren't the innovative products. So that's very complicated, I know. I have to deal with it every day, so what was the good news is, and this is what I wanted to mention, that the Commission Implementing Regulation and Schedule 12A actually mirror each other. We designed, when we left the European Union, when we drafted Schedule 12A, we ensured that it mirrored the Commission Implementing Regulation. So although there are these differences in practical terms, the pharmacovigilance that needs to be carried out under both sets of legislation are the same. So that's the good news. So I mentioned the good pharmacovigilance practices modules earlier. As I said, these were developed in 2012 under Article 108 of the Directive 2001/83/EC, and these are stipulated within that directive, and that makes them statutory guidance, and therefore any market authorization holder within the EU must comply with that guidance. They are a set of modules that are set up that discuss all elements of pharmacovigilance, from collection and collation of adverse events, reporting of those adverse events, through to signal detection, PSURs, the quality management systems that you need to support your pharmacovigilance system. And they not only apply to market authorization holders, but also to each of the regulatory authorities in the European Union as well, and EU member states. So when we left the European Union, what we did was we decided that we would adopt these into our legislation. And so we are still using the good pharmacovigilance practice modules. And to depict where there are differences between what needs to happen in the UK for pharmacovigilance and in the EU, we designed the exceptions and modifications document, and that is published on our website. And it's designed to be used in conjunction with the GVP module. So you need to read the GVP modules with the exception modifications document, and that will tell you where the differences lie between what's needed for EU products and what's needed for those authorised in the UK. So that's not the end of the story, though. There are more updates to come with the introduction of the Windsor Framework. And this is a new legal agreement which has been drawn up, which adjusts the operation of the Northern Ireland Protocol. And the aim is to address the political concerns in the UK, as there's been difficulties around the movement of goods from Great Britain to Northern Ireland. And so where a lot of the focus on the Windsor Framework is about the movement of goods and includes the labelling and packaging of medicinal products within the UK, there are some impacts also to the authorisation of medicinal products. And what the Windsor Framework will mean is that the UK becomes will authorise all products within the UK. So where I was talking about there are that subset of innovative products that are authorised by the EU under the centrally authorised procedure, that will no longer happen. They will all be authorised by the MHRA, and therefore they will sit under the Human Medicines Regulation, Part 11 for pharmacovigilance, and then under Schedule 12A of the Human Medicines Regulation. So where that should make things more simple, that we are the sovereign regulator, we are authorising all products, there will still be those non-innovative products that sit outside of the Windsor framework, which although we will authorise, they will be under the Commission implementing regulation still. So unfortunately, doesn't make things as simple as we would all like, but it is a step towards that. So that's all the complicated bit done. I can take a breath. I'm going to move on to something that's a lot more easier to explain now. And I'm going to talk to you a little bit about our inspection programme and where we are. So we inspect under the Human Medicines Regulation, Regulation 327. And this here explains a little bit of our corporate structure. We sit under the Healthcare and Access Division and in Standards and Compliance. We have two compliance teams. And that contains all of our GXP inspectors. So Compliance Team 2 is our good manufacturing practice inspectors and our good distribution practice inspectors. And then we have Compliance Team 1, which is headed up by Steve Vinter, who spoke earlier, who opened the Pharmacovigilance Symposium this afternoon. And that compliance team consists of our good clinical practice inspectors, good laboratory inspectors, and then our PV inspectors as well. So we have 6 PV inspectors, or we should do when we're at full remit, and then the head of PV is Sophie Radiker. So you'll notice that we have 2 vacancies at the moment, which we should be filling by mid this year. And then I sit just outside of this in the expert circle, so we have an expert for each of the GXPs as well. So moving on to our inspection programme, we in the pharmacovigilance team, we operate a risk-based inspection programme. That means we don't inspect on a cyclical basis. We choose who to go to and where we go and the timeliness of how often we go by risk. So we use a number of factors. We look at things such as the product. Is it a product that's on the additional monitoring list? They're the newer innovative products that we're looking at. Is it a product that we know has associated risks with it? Does it have additional risk minimisation measures, for instance? We'll look at the MAH or the applicant themselves to look at, do we have any intelligence? Is there something that's leading us to believe we should go and inspect that entity? And then we'll look at the PV system. How many products does that PV system have under it? Has there been significant changes to that PV system, any mergers or acquisitions or updates to that PV system that we know about? And then we'll use our compliance history as well. What do we know about the compliance history of that organisation? Have there been significant critical findings in the past? Have we received any intelligence from any of our international collaborative partners regarding that? And there's 4 different inspection arms which we can inspect under. So the main bulk of our inspections are conducted under our routine pharmacovigilance activities inspection arm. And this includes the main elements of pharmacovigilance. So everything from collection and collation of safety data, reporting of adverse drug reactions, signal detection, writing and authoring the submission of PBRAs and PSURs, the quality management system, and all of that comes under that bracket. We then have our routine management, risk management and safety communication inspection arm, and this is looking at all of those risk management, routine risk management that has to be mandated as part of having a licence within the UK. So here I'm talking about summary of product characteristics, which is equivalent to the US PI, and the patient information leaflets in packs. And we'll look at, are those documents up to date with the latest scientific knowledge, and have they been implemented within the timelines that are required? Then we look at the additional risk minimisation measures inspection arm. These are for a subset of products where the benefit risk profile is deemed to be a little bit more unfavourable, and there needs to be some risk minimisation put in place to ensure the safety of that product. And we will conduct those on a product-specific basis. It may be that our assessors within our safety and surveillance team have asked us to conduct inspections, and they can be pre-launch inspections or post-launch, and we'll conduct a specific inspection just looking at that particular additional risk minimisation measure. And they're usually reserved for the more complex additional risk minimisation measures, such as pregnancy prevention programs, controlled access and controlled distribution. And then finally, we have our non-interventional post-authorization safety studies, and they're looking, again, they are very specific. They might be specific to a product or certain studies that we'll choose. They're usually the mandated studies, so category 1 or 2 PAS. And we come into the MAH, we'll all look at from a sponsor, from the MAH perspective, and then we'll also choose a couple of sites to go on to the investigator to have a look at how AE data is being collected. So they're a little bit of a different type of inspection, and for companies, they're a new way of being inspected by the MHRA, because it means a collaboration between not just the pharmacovigilance departments, but the clinical teams as well. And then very briefly, we're going to have a future look and look at regulatory updates. So some of these have already come into effect. So the international recognition procedure, this came into effect on the 1st of January 2024. And this is a process in which the MHRA is recognising market authorisations that have been approved in other regulators, and there's certain regulators that are on the list where this will be recognised. There's 2 ways that market authorisation applicants can put in their applications, dependent on how long that market authorisation has been approved, for instance, the type of product that it is, whether there's any additional risk minimisation measures associated with that product. And it means that it should be a swifter process for approval within the UK, and less burden on our assessors. The early access to medicines scheme, this is something that we laid a statutory instrument for this in 2022. But the reason I want to mention it today is because we're starting to implement some guidance on this, and hopefully that will be published this year. And this is where there has been identified an unmet need for a medicine that is used in a life-threatening disease, and that the company can submit an early access to medicine scheme application, and that means that the MHRA assessors will give a scientific opinion on whether that can be used in the UK, and if so, that scientific opinion will last a year. And as I said, we're implementing some guidelines which hopefully will come out to support that this year. Point of care, this is the introduction of a new innovative regulatory framework. This is looking at manufacturing techniques. So traditionally, products are manufactured in a centralised basis, but with more and more innovative medicines, this is becoming more decentralised, to the point where some medicines need to be manufactured at the point of care, at the patient bedside. And this regulatory framework will help to support those medicines, and will be an update to some of the pharmacovigilance requirements around those as well. And then lastly, the Windsor framework, this will be implemented on the 1st of January 2025. I'm not saying very much about this now, and I can hear all those QPPVs in the UK shouting at me for not saying more, but we will be saying more at our own symposium, and this is a shameless plug for that. It's on the 28th of February 2024, in a couple of weeks, in Hammersmith. So please do come along to that, or log into the webinar where you can hear more on the Windsor framework. So just to summarise very quickly, because I've gone over time, the current UK PV requirements are changing with the Windsor framework. We are getting back up to speed with the number of inspectors that we have with our inspectorate, which is great news for us, bad news for all of you, because you'll see us more regularly. We operate a risk-based inspection model. So for some of you who don't come out at the top of the inspection model, we may never come to inspect. For others of you who hit a lot of those risk criteria, we'll be seeing you more often. And look out for legislative updates. We do put those on our websites, and we are going to be updating our guidance shortly to incorporate the Windsor framework. So please do look there. We also publish our inspection metrics. We have our inspectorate blog where you can find out information, and we try to keep that as up to date as possible. And one last plug, we have our symposium on the 28th of February in Hammersmith, and you can join online. So thank you very much for listening. I think we're going to take a 5-minute break, and then we'll be back for the panel session. So thank you very much, everybody.
Hey, Carolyn.
let everybody take their seats, and I'm going to invite my session 4 presenters to the stage. Paul, Claire, and Pete is going to take the place of Sherry for us. Okay, we'll get started. So, Claire, I have a question for you. So for MHRA, in your opinion, can international collaboration efforts ever lead to full reliance among regulators and negate the need for duplicate inspections? And just a part 2 to that question, will there be harmonization soon on PV regulation as there's increasing collaboration across the board?
Well, that would be utopia, wouldn't it? We'd all love that, I'm sure. And it's something that I think we would love to strive towards. I don't think it's something that we will get to in the short term, but I think what we are aiming for is to harmonise a lot more and to use each other's inspections and the intelligence that we gather to really input into each other's risk assessments and to recognise that we could all start to risk assess companies in the same way and use the same processes in order to do that. And then when we've identified issues, that we can start to recognise, is that something that we need to incorporate into our risk? You know, if we're looking at it from an MHRA perspective, US FDA or Health Canada have been to this company, they didn't find anything that was particularly significant, therefore we can lower them down our risk assessment and use in that way. And I think the other thing that we want to aim for is to not be duplicating efforts. We want to get a lot more harmonisation on where each other are going. We know that having inspections puts a lot of burden on companies, and we want to try and reduce that. So recognising where each other are going, the types of things we're looking at, and what those outcomes are of those inspections, I think is something for the short term. And then looking to harmonise regulations, that would be wonderful in the longer term. So something to aspire to in the future.
Definitely. Thank you. So Paul, I think this is related to your presentation. Are there any opportunities to have access to collated joint inspection reports? And part 2, would the collaborative inspection outcome be available for other MAHs for learning and understanding?
Absolutely. So for now, on Health Canada's side, we can only provide what is publicly available. So there is a website in which you can scroll through the different GVP inspections that were held by Health Canada, and you will find the ones, unfortunately, you won't know exactly which one would be a joint inspection, but you could see the findings there. So for now, there's not a lot of other resources that were put in order for MAHs to have more information on that. But we do invite you to scan the Health Canada website for all those reports to see where we're going and the different MAHs that we've seen. So that's pretty much what I have for now.
Let's see. Okay, Claire, for Project Orbis, what countries are included?
So for Project Orbis, there are 8 countries included. So that is the MHRA, Health Canada, the US FDA, Singapore, Switzerland, Australia, Brazil, and Israel. I remembered them all.
And Paul, why do virtual inspections take more time for health authorities, and can this be avoided?
So in general, it would take more time because as we are planning, and again, this is on Health Canada's side, we have to plan sessions in advance. We need to make sure that all the SMEs are present during the inspection. So it's a lot of organizing. Whereas when you go directly on site, you just provide the agenda of what which topics you'll be discussing. And then when you get there, it's easier to just discuss with the different SMEs present. Whereas when you're virtual and you stop the session, then if you have like smaller questions, then you need to perform another ad hoc session and then get the information that you wanted. So that's where we want to, that's what I intended of saying when it's taking more time. It's more of a generic way of building the inspection itself. Whereas when you go on site, it's much easier for everyone.
Okay. I have another one for you, Paul. Can any country request training, and what is the process?
Unfortunately, there isn't any specific training done. We invite everyone to consult our guidance documents, our regulations. We invite everyone, as Claire mentioned, there is the great webinar that will be held from the MHRA, where you can get information, sessions like these where we can get training. So these are the ones that you are encouraged to go to and assist, but unfortunately we don't, at least again, as the Health Canada way, we don't have anything formal for you to apply.
And then I have one last question to kind of wrap up this discussion for session 4. Will this group do any dedicated symposia on pharmacovigilance in the future?
So I think I'll answer that one. I'd say absolutely. We definitely have plans, or that's our goal, is to continue this collaboration and continue to put on pharmacovigilance conferences like this. We want to continue to be transparent, get the word out, and hopefully everyone in the audience enjoyed the presentations and the training, and you're interested in this topic, and we can continue putting on symposiums like this.
Okay, thank you, session 4 presenters. Okay, we'll now invite session 5 presenters to come up. So that's Peter, Jenna, and Chrissy. Okay, first question. Do regulatory agencies expect evidence of corrections with their responses to inspection observations?
So I'll try to answer that. We expect, we would request a response as quickly as possible, within 15 days. If the response can show proof that the correction was made, we recommend that that's included. It doesn't have to be. Sometimes it can take longer to make that correction. But our goal is to get into the company to get into voluntary compliance, and they can include that information as part of their response that we will evaluate at the center level if it's received in time.
Thanks, Pete. Chrissy, I think this is one for you. The BIMO program for sponsors and CROs was updated in September 2023. How are BIMOs conducted since the update? What is the difference in terms of the approach that FDA, investors, auditors are taking?
Yeah, thanks. So our inspections are conducted the same, even since the update to the compliance program. What we really did with the compliance program is not so much update the approach, but ensure that we were covering new ideas or items that had not been incorporated since the previous revision. So for example, we looked more at electronic systems and updated that information within the compliance program. You know, what would we be looking at? How would we be looking at it, for example? So it's not so much that the approach has changed, but some of the areas that we might be looking at, we've updated just to be consistent with what is occurring in industry at this time related to clinical trials and other types of sponsor inspections.
Okay, is there a difference in type or frequency between PV inspections for generics or new drugs?
So again, I'll try to answer this question. I think it depends. FDA uses a risk-based approach. We don't look particularly just at that one element, if it's a NDA or a generic. We look at many data points, the risk of the product, and we also look more globally at the firm. We're not necessarily looking at just one product, but at the firm. How many products do they have? What type of products they have? And the risk of those products and other information that we've received. So I think it's hard to answer that question just based on a comparison of generic versus NDA.
Chrissy, we have a couple questions related to recruitment and hiring. So does FDA recruit candidates from the UK? Or is it possible for a foreigner based in the EU to become an international FDA inspector?
Wow, I would love that. That would be fantastic. Unfortunately, to be hired with the US government, you do have to be a US citizen or a US national, unfortunately. So if you want to come here, that'd be great. But no, you know, somebody who is, you know, a foreigner in the EU, unfortunately, we cannot do that. So thanks for thinking of us.
I have a question related to the RRAs. Will RRAs be planned or a surprise?
So again, the RRAs will not be a surprise. The RRAs that we've conducted as part of the pilot were voluntary on the parts of the firm. So we were reaching out, I guess, requesting their participation in the RRA. So it was not a surprise. And once we got back word that they agreed to participate, then there was a further time given before we had the kickoff meetings and requested information. So also our inspections are also pre-announced for paid inspections as well. So they're not typically surprise inspections as well. So similar.
We received a few questions about audit trails. Is there guidance, re-audit trails for electronic systems, something that we can use with our IT folks? And then another question, can we say more about performing risk-based audit trail reviews of GCP, GBP systems? Then the rest of the question says that it's kind of a challenge for some of the people involved in that. So I think they want to hear from you again about audit trails.
Yeah, absolutely. So, you know, audit trails are really important. And here's where I'm going to show that it's really important. I'm taking out my glasses because I wanted to make sure I got the citation right here. And so we do have a regulation that deals with audit trails. So if you refer to 21 CFR Part 11, and this is specifically 11.10E, it does say in there that the use of secure computer-generated time-stamped audit trails to independently record the date and time of operator entries and actions that create, modify, or delete electronic records. So that's the requirement. Changes shall not obscure previously recorded information. So I think quality assurance professionals who are hopefully in this audience. We understand that, right? And that this audit trail documentation shall be retained, so not must, but it shall, for at least as long as that required for the subject electronic records, and shall be available for agency review and copying. So if we're coming out to take a look because it shall be available for us, it would behoove you to take a look at it before we get there. So I would really recommend that you take a look at your audit trails, make sure they make sense, that they're capturing the information that they're required to capture per the regulation. And then somebody wanted us to say it louder for the people in the back. So, hey, the people in the back. Yeah, we don't have we may have a published guidance. We have so many guidances, it's hard to keep track, but it is right there in the regulation. And so that should really be enough for your IT folks to say, yes, we do actually need to comply with this because it is in a regulation for that pertains to FDA in regards to electronic records.
So thank you. And then I'll just do one more question for you, Chrissy. Does the expectation for electronic systems access include EHR, patient health histories, and trial management systems, management of clinical and/or safety operations, for example?
Yeah, it does, to the extent that you can, right? We recognize that for some systems, you may not actually be able to give us access. Right? So in that case, we're going to do some shoulder surfing, right? Have somebody sitting there accessing the system while we're looking at it. But to the extent that you can, it really does make things easier on your staff and on our investigators to be able to have somebody show us, here's a patient record, here's where you can find the information that you're looking for. And then we can go through at our own pace and take a look at it. And those folks are free to go do other things that they need to do, right? We know everybody's incredibly busy. So to the extent that we can get access to your systems, fantastic. We know sometimes there are limitations though, and we'll work with you on that if that's the case.
Great, thank you. Thank you, everyone. Thank you, session 5 presenters. And now I'll invite the session 6 presenters up. That's Lauren, Saranjan, and Claire. All right, thank you, everyone. Okay, so Lauren, we'll start with a question for you. If you're unable to find a pro code for a product, say a spray or pumps for topical application, what should be used?
That's a great question. So if I wasn't sure what pro code to use or product code for the device constituent part of my product, the first place I would go to is, unfortunately, it's on my slides. There's a website that has a list of the common device types in NDAs, ANDAs, and BLAs, and check there first. There might be one that fits nicely. But if not, there is a pro code database that's searchable for both common device names and pro codes. That's also on my slides. So do check out those links. But, you know, if you just are looking for help, I would recommend reaching out to CDRH's Division of Industry and Consumer Education, or DICE. It's DICE at fda.hhs.gov, and they can help to identify a pro code that aligns with your product. Or you can reach out to the Office of Combination Products, and we can help you as well.
All right, thank you. Saranjan, will pre-market vaccine INDSRs be submitted via R3 to CBER IND and not EVARs? EVAERS.
So when it comes to vaccine reporting, especially for pre-market IND, when we publish our guidance, the binding guidance, then yes, you would be submitting to FAIRS. So, but the post-market one still goes to EVARS. So post-market reporting is still to EVARS. The pre-market one would come to us, and then it'll eventually be shared with CBER. Of course, during the period of time while you are implementing R3, you will be still submitting through ECTD. We have a mechanism to get that into FAIRS, but once you're ready with electronic submission, then the safety reporting portal as well as the gateway. Safety reporting portal will have an option to say, are you submitting for a CBER IND? And it'll take you to the appropriate form to fill it, which will come to FAIRS, and E2B-R3 will also come to FAIRS. Now, again, just to repeat, it's all for the IND safety reports. Thank you.
Lauren, I have another combination product question for you. Please expand and clarify determination of the lead centers.
Sure. So determination of the lead center or the center who is assigned to the regulation for the combination product. I mentioned that this is based on the determination of the primary mode of action, which is the single mode of action that has the greatest contribution to the overall intended therapeutic effect of that product. And my examples were very straightforward, and it does get more complex than what was on my slides. So there are situations in which that is not so cut and dry. Perhaps there's two independent modes of action. And so in that case, the lead center is determined based on the center that regulates other combination products that have similar types of safety and effectiveness questions, or if that center, there isn't a center that has that, there's, it will be based on the center that has the most expertise that is relevant to the most significant safety and effectiveness questions of that combination product. And if you are not sure, you would like guidance from FDA, you should look into submitting a request for designation or RFD, in which FDA will determine, which constituent part has the primary mode of action and the lead center, or a pre-RFD, which is an informal way to receive that determination. Thank you.
Saranjan, can you tell me more about submitting complaints to FAIRS?
Submitting
complaints?
Oh, complaints to FAIRS.
Yeah.
So, so we have the product quality side of FAIRS. So you know that product quality side, you have the FAR reporting, you have the BPDR reporting, and then we also have from the voluntary side, we have the consumer complaint reporting. So they all come into FAIRS. So the FARS reporting actually has the FARS 3331A form, I think. So that's the form which is filled and submitted to, I believe, a mailbox, and then that report is then taken into FAIRS. And then you have the BPDRs, and they also then are submitted through the mechanism that they have, which then comes into FAIRS. And the consumer complaints also go into FAIRS. So they all are in one database. And we hope someday that we will be able to have a similar type of a portal that we do for adverse event reporting, which then can be utilized to submit the FARS BPDRs, and then also utilize the same portal to be able to provide the consumer complaint that we have got, which has, I believe it's a priority one. It is kind of sent, a copy of the report is sent back to the sponsors for their due diligence on that complaint. And hopefully that portal helps us to do that. That's it. Thank you.
Okay. All right, one last one for you, Saranjan. Do we have separate FDA email contact for IND-SR pre-market product?
Now this question is such there could be multiple answers, but when it comes to IND safety reports today are submitted through electronically, but a PDF format of the MedWatch through the ECTD format, through the gateway. Now, if you have questions about that reporting of how it is to be reported, of course, you will write to esub@fda.hhs.gov about anything concerning with the ECTD. When we move for those safety reports from ECTD to FAIRS, then these reports will have to be either submitted through the safety reporting portal, which is again a web form, or through the electronic submission gateway, which will be the E2B, ICH E2BR3 format. They will all go into FAIRS. Now, if you have any questions then your questions will be directed to FAIRS esub@fda.hhs.gov. Now, those questions are mostly questions we would like those questions to be more on the technical aspects of things. Now, if you have anything to do with the regulatory aspect of things, then the best thing is to contact with the regulatory project manager in that therapeutic areas and ask that questions, or I think there's a mailbox to ask any kind of regulatory questions relating to, should I be submitting this? What about these types of report? And what about this condition? So that would most likely should go to the CDER's Office of Regulatory Affairs, who can answer those questions. Thank you.
All right. Thank you, guys. That was great. A big round of applause for session 6 presenters. And now I think, Ray, oh, there you are. Okay, Ray's going to come up with some housekeeping items.
Thank you so much. We have a special gift for our in-person attendees. Please feel free to take the display copies of the guidances out on the SBI exhibit table with you as you leave today. And this leads us right into our symposium wrap-up and closing remarks by Dr. Laurie Muldowney, Deputy Director, Office of Scientific Investigations, CDER FDA. Please join me to welcome Dr. Muldowney.
Thank you. All right, let me move this up just a little bit. And Ray, I have to say, I love your voice. It's an awesome voice, right? I wish I had that sort of like, it's great. So, wow, it's been a full week and a bit of a whirlwind. So I really appreciate you all sticking with me till the end here. Again, my name is Laurie Muldowney, and I have the pleasure of wrapping things up this week. I understand that I'm the only thing likely between happy hour, your next meal, or traveling home, so I will try to keep it brief. But before I jump into some closing remarks, I do want to take just a few minutes to thank those who made this workshop or this symposium possible. First, I want to thank Brenda Stoddart and the team at CDER's Small Business Industry Assistance Office. I think I got that right. SBIA handled all the logistics, right? They got the conference room, they handled registration, they handled the audiovisual, really everything to make the logistics possible. And if you aren't familiar with SBIA, you really should check out their webpage on fda.gov. It's all free, and it's all intended to be resources, educational resources. They provide live and on-demand information to industry, so definitely check it out. I want to thank the Planning Committee. Planning Committee was comprised of not only staff across the FDA, but also we had Stephen Vinter and his team from MHRA, and Hossein Abid and the team from Health Canada. And the planning team really has been working for the better part of the past year, trying to develop the agenda, identify presenters for this workshop. Many of whom were also serving double duty and presented, which kind of leads me to my next thank you. I appreciate and thank our awesome presenters, our moderators, and our panelists. I think we had a lot of excellent presentations. Thank you for answering all of these questions at the end each day. It was really tremendous. And thank you to the audience for not only providing these questions, but for really staying for this. 3 days is a lot to take out of your busy schedules. We get that, but we also understand that this is a topic, and these are topics that are really important to all of us. And again, thank you to those who are actually still in the room. And finally, I want to thank our fearless workshop leader, Lakeisha Williams. Thank you, Lakeisha, in the back. Lakeisha handles so much of the planning behind the scenes. It's like herding cats is one of my favorite little sayings, but trying to get everything she needs from the presenters, from the planning committee on time in order to make the workshop possible. So Lakeisha, we could not have done it without you. So thank you again. So today we covered a variety of topics. We heard a lot of really interesting bioequivalence case studies this morning that highlighted some of the potential pitfalls in BE studies, both with clinical and on the bioanalytical side. And these case studies really underscore the need to have available the data and the records to reconstruct all of the study activities, to verify if subjects received the right sequence of drug treatments, and that the drug concentrations that are reported to our agencies are reliable. This afternoon, we heard about international collaboration in GPV, the future of pharmacovigilance inspections, and we heard safety updates from each of our agencies. But I want to try to step back and highlight a couple of the common threads from the entire workshop. So we covered 3 broad topics in 3 days. We covered good clinical practice and clinical trials, the conduct of bioavailability and bioequivalence studies, and good pharmacovigilance practices. But there were a few themes that were repeated throughout, and I'm sure these are probably not going to be a surprise to anybody. And some may sound very familiar if you heard Dr. Grandinetti's closing remarks yesterday, but I have been told that repetition is the mother of learning and the father of action. So lots of quotes here today. So perhaps being a bit redundant is not a bad thing. But first, I want to reflect on the impact of innovation and technology on both industry and regulators. On the regulatory side, we heard about leveraging technology and software to facilitate remote and on-site inspections as well as compliance activities. You know, we know that remote evaluations were essential to maintaining oversight during the COVID-19 pandemic, and they continue to be explored by all of the agencies who were represented across a variety of compliance programs. We did hear a bit about some of the challenge that regulators experience when they're conducting these remote activities, as well as some of the successes. And I think it's really great to have an open dialogue and discussion about our experiences with this new tool and how it's evolving. I think it's fair to say that we're continuing to explore how to best use this tool. I do expect that over time we're going to gain efficiencies and that we're going to better understand where the appropriate place to use this tool in the toolbox, as we like to say, is. Sorry, I have dry hands. On the industry side, during our GCP sessions, we heard about digital health technologies. We heard about clinical trials with decentralized elements, real-world evidence, and adaptive design. We're seeing increased numbers of data sources. We have complex data flow, and we're seeing the increasing use of these digital health technologies. And with all of these complexities and opportunities, I like to think, I think the most important takeaway from this entire week, potentially, is the importance of applying a proportionate and risk-based approach to the entire clinical trial lifecycle. So it's really about thinking and documenting your thinking and not using a one-size-fits-all approach. So you should incorporate quality into the design of the study, identify and appropriately manage those aspects of the trial that are critical to the protection of research participants and to generating reliable data that's essential for decision-making. And this includes, of course, through the use of risk-based oversight and monitoring approaches that are tailored to the potential risks of the specific study and that are revised as needed. It's not a start at the beginning and then just run with it. You need to continue to revise and look at the information that you're gathering and continue to update your oversight and monitoring processes. In pharmacovigilance, there are also increasing sources and volumes of data. Both industry and regulators face challenges with timely and efficient collection, processing, and evaluation of the vast amount of safety information that comes in. And advances in emerging technology that we heard about earlier, including artificial intelligence and machine learning, have the potential to address some of these challenges by creating more efficiencies in pharmacovigilance systems. But again, all of this needs to be done in a risk-proportionate way, focusing on important identified risks as well as important potential risks. And we as regulators recognize the need to understand industry's use of these technologies and continue to develop approaches and policy as this landscape really evolves. The other common thread that I hope was really obvious throughout the week is the importance and really fundamental need to collaborate. The pharmaceutical industry, again, it's increasingly global and complex with multi-regional trials that are pretty standard, greater number of clinical trial sites across the globe. After approval, products are used around the world, making patient safety and pharmacovigilance a global endeavor as well. And at the same time, we've already covered some of the increased complexity that we're seeing, but I do want to mention one other area that's been raised a few times and I think is really important, and that's the growth of third-party providers who are conducting trial and post-marketing pharmacovigilance-related activities. So collaboration, including international collaboration between our regulatory agencies, is really more important now than ever. We heard over the last 3 days about the many ways that FDA, MHRA, and Health Canada collaborate, but also how we collaborate with many other foreign regulators, some of whom may be in the room or on video, and how we, as regulators, collaborate with patient groups and industry as well, and really the value that these collaborations bring. We heard a bit about ICH efforts to harmonize regulatory requirements across regions to the extent that that's possible. But even when there still may be differences in processes or even in specific legislative authorities, we're really focused on the same principles. And there's quite a lot that we agree on, as I think everybody heard this week. Regulators are leveraging international collaborations like PICS and ICMRA, as well as bilateral information sharing agreements to try to develop common standards, harmonize inspectional approaches, and exchange important information, including from inspections. Our international collaborations have improved our ability to identify data reliability and human subjects protections concerns. And importantly, they help us to optimize our use of limited regulatory resources and to extend our oversight capacity. So, and of course, meetings like this provide an excellent opportunity to promote compliance, hopefully with a pretty shared voice, which I think is awesome. This is our 4th collaborative workshop over, I think, the last 6 years, and it certainly will not be our last. So please, as you leave today, don't forget to complete surveys. Let us know if there are topics that you would like to see covered in a future workshop on GCP, BE, and PV, or if you have other recommendations that you think might make this workshop more valuable. And for those of you who have been here all week, you will understand why I felt compelled to come up with a Disney quote to send us on our way. Thankfully, Marvel is a subsidiary of Disney because I'm actually an action film girl myself. So I leave you with a quote from Black Panther, which is, "Just because something works doesn't mean it can't be improved." We're in the midst of a time of fairly rapid change, and really in everything we do. And we should embrace this thoughtfully, recognize that not all of our efforts to innovate are going to be a success, and that's okay. But we should always be looking for ways to improve the way we do things. And with that, I say thank you, safe travels to those who are traveling, and we look forward to our next time together. So thank you.